हेलो 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 हेलो
Good morning, everyone. I, Lakshmi Narendra Kumar, with Agila Kumari Pragasam, feel privileged to welcome you all on behalf of the organizing event of one-day conference on human microbiome in health and diseases and 2 days workshop on next-generation sequencing and data analysis. May I request all of you to please keep your cellular devices on silent mode, and for those joining online to please put yourself on mute as we will be starting the program now. To begin the event, I would like to first call upon our Honorable Chief Guest, Professor Rajesh S. Gokhale, the Secretary for the Department of Biotechnology, Government of India, to the stage. Welcome, sir. Next, I welcome our Executive Director, Professor Pramod K. Garg, to the stage. Sir, please. Next, I invite Professor of Eminence, Professor Shinjini Bhatnagar, to the stage. Welcome, ma'am. Next, I invite Dr. Bhabatosh Das, the organizing secretary of the event, to the stage. Sir, please. To begin with, we will have our traditional lamp lighting ceremony. Light is a symbol of brightness and prosperity. Let us begin the event by seeking the blessings of Goddess Saraswati by lighting the lamp of knowledge and wisdom. For seeking the choicest blessings, I invite our Honorable Chief Guest and the dignitaries on the dais to kindly come forward and light the lamp. Sir, ma'am, please.
Thank you, sir. Thank you, ma'am. Now I call upon Professor Chinjini Bhatnagar, Professor of Eminence THSTA, for the welcome address. Ma'am, please.
Good morning, everyone. On behalf of THSTI, a very warm welcome for this program and to our beautiful campus. So it's a proud moment for us and me particularly to share that our human research program on microbiome has reached great heights, and particularly to share that it spans work from basic science to applications which will go into clinical practice, into applications, which is particularly the mandate of this institute. And that philosophy is reflected in the multidimensional program today and the multidimensional faculty and audience that we have for this meeting. So a very warm welcome to all of you to enjoy the meeting. A very special welcome to Professor Gokhale, who is not the Secretary of Department of Biotechnology, so all his support, but particularly as a scientist who's done fantastic and impressive work in this domain. So a very warm welcome to you and thank you so much for coming. And to Dr. Garg, who's Professor Garg, who's despite the 2 years has been so hugely supportive of all these research programs, which are actually now we are seeing applications, and which is really very, it's very satisfying for THSTI. So Bhaptosh, great work, and we hope that this conference is a big success. Again, a very, very special and warm welcome to all of you.
Thank you, ma'am. Next, I call upon Dr. Sushmita Chowdhury, Assistant Professor at THSTI, for initiating the proceedings with the objectives and expected outcome of the conference and workshop. Ma'am, please.
A very good morning to all of you attending today's conference here and online. We are going to start the main program of the conference today, which is on human microbiome in health and disease. So we have kind of tried our best to present various carefully curated sessions for you today, where we have the opportunity to listen to insightful presentations from renowned scientists across the globe. They mostly work on human microbiome and host genome. So we sincerely hope that the participants will benefit from newer scientific ideas and new replication methodologies. In addition to that, we would also like to take this opportunity to celebrate a few achievements of THSTI in this field. After this conference today, tomorrow and day after, we are going to have a workshop on next generation sequencing and data analysis. As we know, NGS or next generation sequencing technologies have revolutionized the world of microbiology. And, we can do very thorough, insightful analysis of compositional and functional profiles of the entire microbial communities now. So this hands-on workshop is aimed to train a young researcher pool on sequencing, the preparing sequence libraries, monitoring sequence data quality, learning specialized algorithms for analyzing the huge and complex pool of data. So we hope that we are contributing a little to create the next generation of scientists in training them. We really sincerely hope that all of you have a very productive time and you are able to nurture and exchange ideas. And thank you. Let's start with the main program.
Thank you, ma'am, for elaborating on the importance and impact of the topic. Now, I call upon our distinguished chief guest, Professor Rajesh S. Gokhale, to share his thoughts regarding the conference and workshop. Professor Rajesh S. Gokhale is currently the Secretary for the Department of Biotechnology, Government of India. He was previously the Director of the Institute of Genomics and Integrative Biology and is an excellent scientist. The thematic focus of his laboratory has been to elucidate the complex interplay between metabolic reprogramming and immunity in the context of the pathogenic diseases, tuberculosis and autoimmune skin disorder vitiligo. He is a recipient of multiple awards, including the Shanti Swarup Bhatnagar Prize for Science and Technology, one of the highest Indian science awards for his contribution to biological sciences. It is an immense honor to have you here today, sir. Please.
Namaskar. Good morning to all of you. Dr. Pramod Garg, Dr. Shinjini Bhatnagar, Dr. Bhautosh Das, Professor Partha Da, my old microbiome collaborator, Dr. Shamila Mande, and lots of my friends here, DBT colleague, Jyoti Logani, and all my friends and colleagues. I think it's a real pleasure to be here today. Early morning for Delhi. I know that it's Delhi always warms up a little bit later in the day, but it's an occasion that I thought that I should come and share some thoughts and ideas that the way the Department of Biotechnology wants people to move on in their lives and careers, actually, such that make impact in the country's future of perspective, actually. And therefore, I thought rather than talking about human microbiome, which is obviously one of the key subject of how technology and how biology is going to influence human lives and careers, actually, I thought I'll put together this thought process of how biology will build the next technology revolution, actually. And I think many of us should in fact start believing it, that it's going to happen sooner than later. Because if you don't do that, then obviously you don't carry forward the burden of making it to happen, actually. I think that the key aspect that we need to learn here is how has technological changes profoundly affected markets and societies, actually. And these two are very intricately linked, because as the markets grow, the economic dividend that you get reaches to the society. And even though, sometimes there are differential, in the way the differential, actually the burden is shared, but you still get value for every individual to be able to carry forward ahead, actually. This is a nice slide that actually gives you a perspective of how from the 17th century onwards, the technology has in fact shaped how our society should function. In fact, in 17th century, the industrial revolution changed the way everything has to happen in this world. Actually, the coal and then the steam engine and the railway started to come in the 18th century. And then there was an era of steel and electricity and heavy engineering, which led to the changes in the way automobile industry moved on, the mass production of it. And then you move to an ICT, which is in the late 19th century. And now, you can see the rapid pace in which the technological inventions are happening, you can actually feel for yourself. And now this is going to be Industry 4.0, which is going to make the whole world probably different in the way it really interacts with us. But it's also important to look at the y-axis actually. And why this is important to understand is because y-axis shows you the GDP in 2011 in the way the US dollars actually all across the countries. For the countries who have these technologies, and the countries who do not have these technologies, and you can see the GDP difference that is likely to be have if you don't adopt to Industry 4.0. It's going to be amazingly very, very different than what you had years back actually. So this is just trying to tell you the fact that if science and technological prowess in the country doesn't grow to and expand to a level that we all expect it to happen, there's going to be a tremendous amount of challenge in terms of the economic value that you will get. And if the economic value is low, obviously there is going to be an equal challenge in terms of variety of sources of funding that you will get for your own research actually. So this is what is the real aspect to look at. So this is what I thought to give you just a perspective. All of you know about it. But centuries ago, when physics and engineering came together, you can see all these got invented actually. Radios, telephone, televisions, aircraft, radars, nuclear power, internet, and host of digital tools actually. And I think you cannot imagine life without it actually. And this is not very long back. I think you can see just about a century back, everything has happened now. And now what is going to change, I mean, the whole of the world is going to be what people talk about now, is going to be living machines actually. And which is the convergence of biology and engineering that's going to create the completely different new world actually. And I think the thoughts and ideas that we have right now in terms of how we do biology and how biology in terms of convergence with engineering is going to change, it's going to be quite remarkable, I think, and it's going to be really exemplary. For example, how something can happen and something can dramatically change. So what are the problems that confronts us potential disaster consequences actually? If we really don't if we don't really put together our thoughts and ideas together in this process now. And if you really look at, there are really 4 areas that really we need to secure our country if one wants to make a larger and larger impact actually. The first is agri-food, which is obviously one of the green revolution that really changed the course of the population growth actually, because the population growth was happening at a very rapid pace. If you didn't have food for everybody, then you would have really a challenge of looking at how it would have been today actually. The energy is obviously one of the central component of it, and clearly there are different, the oil and the gas sector has completely dramatically changed along with the coal, which initiated early on. And if you really look at the oil bust that happened in 70s, where the Gulf War and other had started actually, there's a new era that is again going to start about, which is actually now happening with the geopolitical changes that is happening, is that the energy crisis is going to brew up in a very different manner all of again. It's also very important to understand when would these eras go up and down actually. So for example, if you look at the Stone Age and how Stone Age to the oil era in fact have emerged, you'll see the time that changes happens in technologies is actually getting reduced every now and then. And that's why it's even more challenging for us to learn to adapt and establish and be able to take up to these kind of activities. The third one is healthcare actually. And I think many of you would have realized during COVID how healthcare systems can just fall apart without actually even having all of us to be able to manage to look at it. For a country of 1.4 billion people with having 12 or 14 All India Institute of Medical Sciences in the country, how difficult it is to manage the entire systems actually. And I really believe that the healthcare systems, rather than being a reactive one, where you start looking for doctors as you start having to, will actually convert into a proactive one in about I think it may not even take more than a decade to happen, where people will have wearables actually, and their data may be embedded into one single soft database centers from where the data could be in fact be valued and various different activities could be done. So to do all these things, and the fourth one is obviously climate change, which is associated with all of them. The center of all of this is going to be the data and the digital world actually. And it is this concept with which that the world is looking at India, because the amount of data that Indians can actually acquire and generate right now for a variety of diseases, a variety of systems as to look at, is going to be very, very immense actually. And this has to be connected with three technological changes that are, for example, I've listed big data and artificial intelligence, communications and networks, and the mech tech, for example, drones or AI, robotics and other these kind of activities. So many of these, if we are able to encompass ourselves, we'll be able to solve problems which are associated in the present, which seems like challenging, but they are very, very, very quickly will be conquered in various ways that we'll be able to look at it actually. And I think the biology is going to play a major role. For example, I mean, already people are talking about viruses, which will be energy storage functions for how energy will be stored in viruses actually, such that they can be utilized in a way. A cluster of viruses will become kind of an energy storage mechanism for the way in which the future of the world would be actually. And I thought, the title of the workshop, which is Cultivating Your Own Microbiome, actually is a very, very trendy one and very, very appropriate for the way to look at it actually. And of course, if we look at our own traditional practices, this is a book that I just wanted to look at it actually. Both Ayurveda and Chinese practices for healthy gut and clear mind has been a very, very long-term activities of thought process and ideas. And it's now maybe we are trying to say gut-brain axis, and we start talking about all sorts of parameters which are directly efficiently looking at it. But I think what is really interesting is how should we approach these kind of microbiome related problems, in fact. And I'm one of those who believe that, if we have missed some part of technological changes, we should not go back to the older ones. So for example, do we really have to look at mice as a microbiome to be able to understand the human microbiome systems, or we need to figure out our own mechanisms and ways to be able to look at it actually? I mean, I was very, or we need to generate our own synthetic microbiome such that we start understanding how the dynamics of this occurs in a population-based systems actually. And of course, all of you are experts in this area. I've kind of dabbled around in this area a little bit actually. But I was very stark to see one of the synthetic microbiome paper that appeared in Cell about, I think, in September or October of this year, actually, from a friend of mine called Michael Fishback, actually, from Stanford University. And he and I used to work together on a common area of polyketide and non-ribosomal peptide synthesis. But I think he had this consortium of 100 microbiome that he transplanted in mice. And I think it's a rather intricate story where he then utilizes fecal transplant to be able to see, once we have colonized these 100 microbes inside the intestine, how many more can in fact survive and make it into it actually. In fact, he got a number of about 119 microbes then, which he says are a consortia of microbiome that you can now analyze and study as a population dynamics of looking at it. And of course, we all now know that even the commensal bacteria under the conditions become virulent. And so, the whole idea of trying to look at, like, for example, some of the work that we did along with Sharmila, who is here actually, is one of the early work in looking at skin microbiome actually in patients with leukoderma. And what we wanted to compare was between the lesional and non-lesional skin of the same individual, how is the microbiome differing and what kind of way differences can you look at. And one of the benefit in looking at skin microbiome is that you get samples that are from the same individuals, which are localized and which are from different regions, and give us some really interesting vitiligo-specific microbiota, which is now validated by several others. Of course, this was ribotyping story actually very early on in science areas, and we would have loved to do a little bit more. But you can see actinobacterial species becomes a central regulatory node in non-lesional species belonging to Firmicutes, which dominates the lesional sites. Now, the whole concept is this is maybe an acquired syndrome actually, because the lesional sites in leukoderma patients have heightened amount of oxidative stress, and this oxidative stress leads to an altered microbiome, which maybe in a way is able to maintain a homeostatic condition that makes it very, very important for it to survive and to be able to carry out. But you would look at acne as many of these propionibacteria. I mean, there are your commensal bacteria, but when they trapped in the mucosal layer in a way that they start to become virulent actually. So we really need to understand these dynamics of biology and really make it useful for getting rid of several diseases and several concepts. And my real interest is to try and talk to you all of them. Is that Department of Biotechnology's role is to really do this actually, is to make the mechanism cycle useful for all of you to get these activities to function. But what I would really like you to look at is that many of these, in fact, can be done in institutions like these fantastic institutions that are like PHSTI, where Dr. Pramod Garg and several of you are doing fabulous stuff, actually. And of course, the COVID response to THSTI was absolutely outstanding. And I think congratulate all of you. But really, we need to run through this entire cycle to be able to make these activities happen. The innovation, which is actually happens, is strengthening and aligning the science and technology innovation ecosystem. Of course, the R&D budget becomes the center of it. And then the industry has to acquire the harness frontier technologies. And how do you, make them, incentivize them? How do you get them to work better and better to be able to look at it? And then you build a workforce, transitions and capacity building, because that's a constant change that one has to look at. And then you need regulatory reforms, which is institutions and instruments to synchronize. And finally, if ever the technologies have to come to the market, the societies has to be even grown up to the level to be able to accept them, actually. And so social protection becomes one of the other important aspects to look at, actually. And I thought that's why I put together this slide, because I think THSTI is a magic mix of resource, people, and ethos, actually. And of course, with Dr. Bhan's, ideas of setting up this institution, there's a tremendous amount of positive energy that is there in THSTI. And my only sincere request to each one of you is that it must achieve something that no one have done before you have accomplished, actually. And that's the ethos that we need to look at and develop with the scarce resource and limited funding. It's the innovation that has to grow up at a faster pace. We cannot keep on duplicating the stuff, because you have to understand the GDP of our country is not what GDP of USA is today or the GDP of UK is. However, there's a tremendous amount of change in which the attitude of people, if you look at the way younger people want to approach the problems, actually. And I want some of the elder ones like me to mentor them to take high risk problems, actually. And that's where I think it's a real challenge for us to really look at it, actually. Incidentally, there is also one of the major challenges to look at how transition in workforce has to happen, actually. And I think that has also got stuck in 19th century. In fact, this article in Nature very recently again caught my attention. It says PhD training is no longer fit for its purpose. It needs reforms now, actually. And this is an editorial that was published in Nature 2023, Jan issue of it. And I thought it raised some really stark and interesting problem, actually. It says PhD candidates are inadequately prepared for cross-disciplinary work and large team characterizing cutting-edge science today. And I can almost everybody would agree that if I have to write a cell paper today, there's almost no way a PhD student can complete a cell work even in 5 years' time, actually. It's just so cross-disciplinary, so much of data, so much of amazing things you need to embed. There's a chronic lack of support and poor quality supervision in senior researchers, rarely trained in mentorship, because everybody runs through their own cycles, actually. We live in our world. We never think of how the futuristic world is going to be, and it's not harder to change. And all of us come trained with something. And I think many of us continue to be trained in only what we have done it, actually, and not continue to train ourselves into new areas of sciences, which is very, very critical. Given the pace in which the innovations are happening in the world, I think we need to continue to learn even while being a PI on the right. It's also a master apprenticeship where safeguarding and advancing knowledge in the individual discipline is far more thought to be. And this is a classical 19th century Germany PhD. You can look at where PhDs really started in, evolved in the Germany. And the work is still evaluated for a PhD as a single author dissertation, which has to be published for whatever, with all sorts of predatory publishing, all sorts of crazy things can happen. So really, this is a big challenge, actually. And I think this is a slide to show in 2016. You can see how many PhD students do we create across the world these days. There are close to 70,000 was that number that time. Now it is close to 80,000 PhD students. India is about 26,000, and now it is about 40,000 PhD students actually that we produce every year. And I think China produces about 80,000 as well. So it's large number of population of people that who are PhDs actually. And what are we doing with them actually? And I think the challenge is we are creating PhDs, but we are not training them to be real PhDs of value. It's actually going to be a real challenge. I mean, you're going to create unemployment PhDs who have no value to bring to actually. And so therefore, I really think that this is something that we need to embed into our PhD programs. And I'm hoping that DBT's new PhD program that we're going to start as part of a BRIC would in fact have this 4 facets of learning. And this is very well said in earlier literature, actually, if you really look at the 4 key things to your learning process. One is Shastra, Tantra, Vidya, and Kala. And what we do very well is Shastra, actually, which is specialized knowledge. You all come with the learned knowledge. You come with the specific thoughts and ideas, and you want to, you convert into. So Tantra is what your PIs taught you about doctrine and principles and embed with you this. And Vidya is the deep knowledge and reasoning that you acquire with time, which is about 3 years of time when you start to acquire. However, you never convert into performance or a skill effective manner, actually. We never taught what I'm supposed to do after that. And I think this Kala is what all other forms of, you can look at arts or various other, where the Kala is what is given more precedence over just about looking at Vidya, actually. And in our scientific areas, we rarely put any attention on Kala. We in fact put all the attention on Vidya, actually. And therefore, many of us are ill-trained and ill-adapted to actually, in fact, to solve problems that either country wants to solve, or your institution wants to solve, or your people wants to solve, actually. And I think somehow we need to change very, very quickly and very rapidly if we want to modify and get into these areas, actually. Just lastly, I think what Department of Biotechnology is looking at future of this country. And obviously the future belongs to bioeconomy. And as I said, bio is what going to be the world of science. And because this is a chemical world, one of the things that happens that India produces about 70,000 to 80,000 chemicals, chemical industry, that is, and all of it comes from a bulk of petrochemical industry. In fact, a large program is being envisaged on around this of how biomanufacturing of biogenic products is going to be the future. And many of you, if you're looking for some problems to look at, I think it's a great area to start your careers on, adapt very quickly. You can see how technologies will dramatically change the world, and it's going to make with deep science and deep knowledge. This is the only way that one can see the future of this country. Thank you very much.
I'm actually compelled to speak out of turn for a fantastic talk that you have given. And you can see how an excellent scientist is transforming into an administrator and a policy maker, enlarging your canvas. So this is such a beautiful sight, how your own experiences are going to now shape the biotechnology sector and help all these institutions such as THSTI to carry out their mission. And thank you for your kind words for this institution. Thank you.
Thank you once again, sir, for the excellent briefing and for the deep insights. Now, I request our chief guest, Professor Rajesh Gokhale, sir, to launch our product, Biome Life, a universal specimen transport media developed at THSTI and technology transferred to Rivenel Biomedical. So please.
Welcome to Rivenel Biomedical. Biome is a universal rubber media for specific protection, preservation, and transport. It can preserve a wide range of bacteria, fungi, and viruses, whether aeros or anaerobes. Depending on the temperature, the sample can be stored for 48 hours at temperature, 7 days at 4 degrees Celsius, 90 days at minus 20 degrees Celsius, or 1.5 years at minus 80 degrees Celsius. It keeps the microbes viable, preserves the tissue, and maintains high DNA and RNA integrity. Whether it is genome sequencing, metagenomics, metabolomics, proteomics, or functional genomics, BioLife can assist you in making groundbreaking discoveries. Rivenel Biomedical is a DSI recognized company with more than 12 patents and 10 trademarks. It is internationally recognized as a deep tech pioneer by Hello Tomorrow, Paris, France. Connect with us to know more.
Thank you, sir. We believe that our product will be of great use and help for all the clinical researchers over there for the improved transport and storage of the specimens. Now we have the announcement of the THSTI Lifetime Achievement Award. The THSTI Lifetime Achievement Award is being presented to Dr. G. Balakrishna Iyer. Professor Balakrishna Iyer is an eminent Indian scientist who has made several groundbreaking discoveries in the field of microbiology. Professor Nair was a former executive director of the THSTI Faridabad. Before joining THSTI, he was working at the National Institute of Cholera and Enteric Diseases as a director. He has also served as a director of the Laboratory Sciences Division at the International Center for Diarrheal Disease Research, Bangladesh. In 1998, he received the prestigious Shwanti Swarup Bhatnagar Prize for Medical Sciences for his contributions to the discovery of the new cholera-causing serogroup, now globally known as Vibrio cholera 0139 Bengal. During his tenure at THSTI, he established a distinct culture of gentle reshaping of scientific minds towards translational research. He led the Center for Human Microbial Ecology Center at THSTI. In appreciation and recognition of his outstanding contribution to the field of human microbiome, the THSTI fraternity would like to honor him with the Lifetime Achievement Award. Dr. Nair was unable to join us here today. I invite our chief guest, Professor Rajesh S. Gokhale, sir, to join us to witness the moments of honor when the THSTI family presented the Lifetime Achievement Award to Dr. Nair at his residence. Sir, please. Thank you, sir. Next, I would like to call upon Professor Pramod K. Garg, the Executive Director of THSTI, to deliver the keynote speech. So please.
Good morning, ladies and gentlemen. It's my pleasure to welcome once again Professor Gokhale, who has graced this occasion as the chief guest. And I'd like to welcome all the distinguished guests who have come from all over the country. And a special welcome to all the delegates who are going to participate in this conference and workshop. Bhaptosh actually asked me to give this talk, and I said I'm not the right person to give it because although I'm a gastroenterologist, but I don't really work on microbiome. But he kind of forced me because he wanted to get himself out of it. So, but it's okay. So I was making the slides last night till about 1:30. So please, if you have any questions, of course, you have to direct to Bhaptosh during the tea break, not to me. So when we talk about microbiome, I mean, it's all over the place now. Whether you are a biologist, you are a microbiologist, you are a doctor, you are a physician, you are surgeons even, or anybody, everybody talks about microbiome. And I think it is very important for all the scientists to know a little bit about microbiome, even if you're not directly working in this area. So some of the things that I'm going to cover would be what is microbiome? Is there something like normal microbiome? What are the factors affecting it? When we talk about microbiome in disease, is there an association or causality? What are the functional consequences? When you have to design some studies in microbiome, what are the key considerations and where we are in terms of therapeutics? So most of these aspects will be covered during the day today, and also the workshop participants will have in-depth discussions on these aspects over the next 2 days. So the reason I'm doing this is just to give a primer on microbiome in health and disease, and also a little bit about the various technical aspects of this. So you all know that healthy human microbiome, and microbiome is all over our body, inside our bodies and all over our bodies. And Dr. Gokhale mentioned about skin microbiome, for example, and it forms about 1 to 3% of our body weight. When we talk about microbiota, they comprise of bacteria, archaea, fungi, protists, and algae. And if you look at the gut microbiome, oropharynx actually has a lot of these bacteria, and you go down, it decreases, and then towards the colon, it increases. And there is a specific reason why that happens, why the upper gut is kind of sterile, kind of, not really, but kind of sterile. And you go down, then you have a lot of these microbes in our intestine and in other places in the body as well. One term which is often confused by students is microbiota and microbiome. Microbiome is the gene pool of the microbiota, which is the living organisms in our body. And there are four major, when we talk about microbiome or microbiota, basically it is largely confined to gut that I'm going to talk about, although now we have respiratory tract microbiota, and during COVID, there are a lot of work being done on that. And we have, skin microbiome, which is also very important. So in the gut, we have four major types: the Firmicutes, Bacteroidetes, Acinetobacter, and Proteobacteria. In Firmicutes, you have these three major phyla, and in Bacteroidetes, you have two, Bacteroidetes and Previtola, and Acinetobacter is Bifidobacterium. Proteobacteria is something which could be, not really, not always, could be pathogenic, like E. coli, Shigella, and Salmonella. And I'm sure all of you know about their infective diseases. Most of these bacteria are anaerobes and very difficult to culture. And that's the reason why study of microbiome takes a different dimension, and we have to rely on other techniques. So these are the dominant bacterial taxa in the gut of healthy Indians. Healthy is, of course, in inverted commas. And these figures that I've given here, and this slide has been given to, many of these slides have been given to me by Bhaptosh. So thank you, Bhaptosh, for that. These figures will vary. So Firmicutes, for example, says 62%, but this may be 40%, 50%, 60%, 70%. So these are variable and across populations, different populations. And diversity is also different, although you find that diversity is also different in rural areas and urban areas. In fact, my colleague, Dr. Ahuja, brought some samples from Leh, which Bhaptosh actually analyzed and showed that there is a difference in those areas as well. So there is nothing like normal or healthy in a strict sense that you have to have this composition to say it is normal or healthy. It actually varies. And one of the important things that we need to realize that our gut microbiota, or generally microbiota in general, is established in the early childhood. And there is a tremendous influence of the maternal microbiota. And also the mode of delivery has an influence on that. But during the early childhood, it establishes itself and remains stable in adult life. Now, when I say stable in adult life, it has a very important therapeutic implication. Now, therapeutic implication in the sense that if you think in a person, this microbiome is not that healthy, you want to manipulate it, change it, you can only do so for a short period of time, because it comes back to its original state. It may change, but it comes back to stable. And the therapeutic implication thereof is that you have to continuously try and alter that to the state that you want it to be. It may change temporarily, which is also known as plasticity, and major influence, as you know, would be diet, which is so variable, geography, which has possibly some influence of genetics and ethnicity and diet and other lifestyle behavior. Infections, acute infections, chronic infections can alter your microbiota tremendously. Drugs, and two important drugs. One is antibiotic, we all know. The other we may not know is acid suppressor. And when I ask many patients what are the medications you're on, they will never tell me they're on proton pump inhibitors. So for example, a drug like Pentoside, or no, I take that in the morning, like breakfast, So that has a major influence on your gut microbiome. And then exercise has a very, very positive influence on healthy gut microbiome or microbiome in general. And many beneficial effects of exercise come through its impact on microbiome. So Dr. Baptosh has actually done many studies, and I'm going to show you some of them. When I talk about variability between populations, you see here how Indian and Japanese gut microbiome are different, both for bacteria and fungi. In fact, it tells me that in the Indian gut, we have more candida than the Japanese gut. And this could also be having some influence on our health and disease. And similarly, he also compared Indian and Danish population and showed a significant difference. So when we talk about microbiome in health and disease, there is a symbiosis between us and bacteria or microbes in general, and this is known as homeostasis with regard to the microbiome. If there is dysbiosis because of many factors, and these factors I've just told you could influence your microbiome, could lead to various disease states. So a normal healthy microbiome may look like this. You have more firmicutes than other phyla. And when there is a dysbiosis, it changes. And it changes, and this change is somewhat specific to different disease states. You may have a different bacterial microbial signature in diabetes or heart disease or inflammatory bowel disease or other disease states. So they are different, and that is why the study of microbiome becomes very important in specific disease states when there is dysbiosis. And now n number of diseases affecting every organ in the body now has been associated with dysbiosis, whether it is GI tract, which is the obvious first choice, and then other liver diseases, cardiovascular diseases, neurological diseases. And now we know even, disease associated with old age, like dementia, may be originating somewhere from dysbiosis. And that is why there is a lot of emphasis now on therapeutics in that area. So some examples here, Dr. Shalimar is here from AIMS, who works on liver disease and NFLD, and he and Baptosh have collaborated on NASH patients. You know there is a bidirectional communication between gut and liver, very important, and they have shown that in NASH patients there is lower microbial diversity, and one particular bacteria is increased, that is Collinsula. In fact, they have shown and there are other bacteria taxa which are also predominant in these patients. And mechanistically, Dr. Baptosh has also shown that Collinsula in animal models could also induce fatty liver changes. In IBD, my colleague Dr. Ahuja and Saurabh Kedia, who are going to be here today, they work on IBD and have shown in both ulcerative colitis and Crohn's disease, there is significant alteration in gut microbiome. In diabetes, again, there are different specialized signatures of a microbiome which differentiate between euglycemia and a diabetes status, again, emphasizing the role the gut microbiome might be playing. The question is, how do these microbes influence our health and disease? So one important thing is that we have now realized that bacteria actually ferment a lot of complex carbohydrate, digest and provide us more and more calories, including short-chain fatty acids, which have a major influence on colonic health and also maintain gut permeability. And they also have an important role on immune regulation. So, some people will say that I don't eat much, but my weight keeps on increasing. Others will say I eat a lot, but my weight doesn't increase. And now it has been shown there is a lot of difference between obese and lean people. So it is quite possible that someone who doesn't eat much, but still weight goes up, it is the microbiome which is making sure that everything that you eat, even in small amounts, is actually utilized to its best potential. And you get more and more calories. So that is something, and this has been shown, experimentally also. You transfer the microbiota from obese mice to a lean mice, and vice versa, and there is a tremendous change in their weight. Bacterial metabolites, we know a lot of metabolites are produced by these bacteria, and they can also influence your health and disease. Many of these have been associated with disease states. And I'm happy to tell you that at THSTI, we do this metabolomics, and there you can also identify bacterial metabolites if you know them. One of the important things was trimethylamine, which converted to TMO, and now it has been shown that it correlates with the prognosis of many cardiovascular diseases, and there is a potential that you can manipulate that. And of course, you know that microbes produce a lot of vitamins. No point taking supplements of vitamins. You feed your bacteria healthy food, and they will produce natural vitamins for you. The major question that arises here is, are these mere associations, or there is a causality here in the sense that it is the microbe dysbiosis causing disease or disease causing an influence on that? And it could be both ways, bidirectional rather than saying unidirectional. But how to establish that causality? Somewhat difficult, but now we need more and more animal studies to have mechanistic insight into this association. We have to have transcriptomics to understand the bacterial products, how they affect our physiology and involved in pathophysiology, and then therapeutic effect. You modulate your gut microbiome and see the effect on the disease outcomes. These are areas which are being worked upon currently, and I'm sure more and more information will come on. If you have to study, and there are a lot of students here who are going to be attending the workshop also, if you have to do studies on microbiome, which are very important, what are the key considerations? The key considerations would be study design. What kind of study design you have? What are these selection of your subjects? Not only the cases, but also controls, because you have to compare with something. And there are a lot of information is available. How do you select your controls? How do you select your patients? How do you calculate a sample size? How many people to study? You say, okay, I have only 10 lakh rupees in my kitty. I can only study 50 patients, because Bhaptosh will say, no, I will charge you more. And Dr. Gokhale will say, sorry, we have only this much. We can't give you more than 10 lakhs. So what do? But you have to have a scientific basis, a rationale for calculating a proper selection sample size before you embark on that. You can do a pilot study first, then go into a bigger one, but you should understand that you just can't do in 20 patients, 30 patients, or 10 patients, or whatever. Then collection and preparation of sample is very critical, because you may collect a sample in a manner which is different from someone else, and you find the results in the same disease are completely different. Then DNA extraction, and Dr. Bhaptosh had just launched this product, which kind of maintains the proper DNA of the microbes, and then you can have a better DNA quality. Then what kind of techniques you use for identifying the bacteria, metagenomics, very important technique, sequencing that we have. And most important is, once you have a lot of data output, how do you analyze that? How do you find out what is the composition of that microbiota? So how do you study microbiome? Simple answer is sequencing, because most of these bacteria are non-culturable. So I'll tell you a story. This was in 1991. This is an editorial published in a journal called Annals of Internal Medicine, and by Harvey Alter, and Parthoda is smiling on that because he knows this story. Hepatitis C virus, which is now on the verge of elimination, wasn't known, but we knew at that time that there is another virus which causes chronic liver disease or cirrhosis of the liver, and also hepatocellular carcinoma. We knew about hepatitis B virus that had been characterized in 1960s and won a Nobel Prize for that. But hepatitis C wasn't known. So what they did, they sequenced the samples. They didn't know anything, and they come out that there is a special virus which is there in patients with liver disease, and they named it as hepatitis C virus. And that is why this title of this editorial, very catchy: Descartes before the horse. I clone, therefore I am. The hepatitis C virus in current perspective. And this is something similar, which also happened in the field of microbiome. Professor Carl Woese, who devised the 16S RNA technology and also discovered RK, he was, I think, the main figure behind the whole technology of how microbiome is studied today. So there are two major techniques, 16S RNA and metagenomics, and I'm sure tomorrow and day after, you are going to have a lot of information on that. 16S, as you know, is a prokaryotic ribosome, and 16S component of that is the one which is generally used in sequencing of microbiome. And of course, we are very happy that when you talk about ribosome, there is an Indian connection, and a Nobel Prize has been given to one of our own scientists. So this is something which is ubiquitous in bacteria, very special, very conserved, and has both conserved and variable region. And studies have shown that by doing 16S RNA sequencing, you can easily identify most of these. And again, you have to go through a series of, sample collection, DNA extraction, primers, and sequencing before you come out with results. And next generation sequencing is the one that we generally use. There are multiple platforms of doing this. And I'm very happy to tell you that at THSTI, with the help of DBT and all the support, financial support and other supports, we are able to now have the complete platform at THSTI. And as I said, bioinformatics is something which is very, very important. And when you analyze these samples, you have to talk about alpha diversity and beta diversity. And tomorrow you are going to have some lectures on that. One term that I like to introduce to or highlight to the students here is shotgun metagenomics. People always confuse between 16S RNA sequencing and shotgun metagenomics. Shotgun metagenomics is untargeted, that is shotgun sequencing of all means meta, microbial genomics is genomics. So this is not, but scientists obviously use very fancy terms sometimes. You don't know what is shotgun, this kind of a gun or a short or a long gun. So collective microbial genomic content of a clinical or environmental sample is a sequence in this, and all extracted DNA is then shared and desired fragment size and then sequenced to find out what all are there. So now we know there are fungi, there are bacteria and viruses and other things. And this is obviously a culture independent technique. So it profiles all kinds of taxonomic composition and also tells you the functional aspects of those microbes. So functional is very, very important. And there is a whole sequence of events, steps which are required for metagenomics. Which one to use? I think it depends basically on research questions. If you have to do species specific, you go for metagenomics, and functional status, of course, you have to do metagenomics, but depends on research questions. Coming to therapeutics, we are in an era now that we are using these probiotics and prebiotics, which are somewhat helpful, not really, but something which has been shown to be very effective is fecal microbiota transplantation. And what Dr. Gokhale was referring to, that now is an era of synthetic microbiota also, some kind of combination of this. So they say that, why fecal microbiota? The sum of parts is greater than the whole. So you have multiple bacteria coming in, you want to give 10, 20, 100 or whatever, but it is possible there are so many others we don't know, and therefore fecal microbiota transplantation may be more helpful. This is already a success story in C. difficile infection and also IBD. My colleague Dr. Ahuja has recently shown in a randomized trial that fecal microbiota transplantation is very effective in them. And I think major advances in future would come from bacterial metabolites as drugs, because this is something that going to come in next 10 years or so. At THSTI, we have almost a complete solution. We have all platforms as sequencing, bioinformatics, and also now a kind of training school. Bhatosh has been involved in that earlier also, and now we are doing it regularly. So I would request all of you who have come from other places to come and collaborate with us. We are very open, and we have all the complete solutions for this. So I like to conclude and summarize my talk by saying that microbiome is omnipresent. It is actually today difficult to define what is self. Self, who am I? I am my own self, or I am plus all the microbiome. So maybe that is all the complete self. You have to take care of your microbiome. Two things, simple: healthy diet and exercise. If you do that, you can keep all these diseases away. Best medicine is diet and exercise. We have to work on association to causality. More mechanistic studies are required. Therapeutics is already there, but I think we have a long way to go to get into real mechanism, and microbial metabolite based therapy could be the future. Thank you for kind attention and for this opportunity.
Thank you, sir, for the very informative talk on the role of microbiome in various disease conditions and maintaining health. Now, I request Professor Pramod K. Garg to felicitate our chief guest, Professor Rajesh S. Gokhale. Thank you, sir. With this, we are concluding our inaugural session. I request the dignitaries on the dais to kindly take their seats in the audience. We will now begin with the first scientific session shortly.
We now begin our first scientific session on microbiome in health and metabolic diseases. I humbly call upon the session chairs, Professor Subir K. Maulik and Professor Bipin Kumar Nair to the stage. Sir, please. Professor Subir K. Maulik is presently an emeritus scientist at the Indian Council of Medical Research, New Delhi. Prior to that, Professor Maulik was a professor in pharmacology at the All India Institute of Medical Sciences. Professor Maulik has been actively involved in both basic and clinical cardiovascular pharmacology research and mentoring programs for the last 30 years. His research involves scientific validation of the pharmacological effects of medicinal plants. Thank you, sir, for accepting the invitation and joining us here today. Professor Bipin Kumar Nair is the professor and dean of School of Biotechnology, Amrita Vishwa Vidyapeetham. His research focuses on studying the role of quorum sensing and biofilm formation in bacterial pathogens and identification of natural product inhibitors that block virulence and biofilm formation in bacteria. Additionally, he also works towards development of low-cost biomedical devices and diagnostics in the management of diabetes and chronic wounds. It's an absolute delight to have you here with us today, sir. I also humbly request our next scientific session chairperson, Professor Sudhanshu Vrati, to kindly come upon to the stage. Sir, please. Professor Sudhanshu Vrati is serving as the Executive Director of the Regional Center for Biotechnology. The major research focus of his team has been on understanding RNA virus replication, in particular the Japanese encephalitis virus. He is also a visiting scientist at the Pasteur Institute, Paris, and an executive director of Bharat Immunologicals and Biological Corporation, a Government of India undertaking to manufacture vaccines and immunizers. Thank you, sir, for coming over to grace the event. In this session, each talk will be of 20 minutes. After the talk of all the speakers, there will be a 10 minutes long question and answer session. I request the audience to kindly reserve their questions until the end of the session. I now request the session chairs to introduce the speakers and call them over for their speak. Thank you, sir.
Good morning, everybody, and very warm welcome to the very first scientific session of this very important deliberation. The first speaker for this session is a very distinguished person, Professor Partha Pratim Majumdar, and I have great pleasure in inviting him. Professor Majumdar is currently a distinguished professor at the National Institute of Biomedical Genomics, Kalyani. He is also an emeritus professor at the Indian Statistical Institute, Kolkata. He was the founder director of National Institute of Biomedical Genomics, NIBMG, Kalyani, and an honorary director at TCG ISI Center for Population Genomics in Kolkata. He is a recipient of several prestigious awards. His research work is in the direction to develop statistical methods and to innovatively apply these methods in the understanding of the genetic basis of human disease, such as oral cancer. I have great pleasure in inviting Professor Majumdar. Please.
Good morning, everyone. It's really a pleasure to be called upon to give a talk in this important event. Actually, last night I went to bed at 11:30 and I was very anxious at that time. And the reason why I was anxious is that, title of my talk was given by the organizer himself, Dr. Bhavatosh Das. And I also knew what the title of the executive director's talk was, and I wasn't sure how much of overlap there would be. Today, after I heard the executive director, Professor Pramod Garg's talk, realized that there is some amount of overlap, but not very much. Like Professor Garg said, some years ago when work on microbiome started, those of us who were thinking about or working on the areas of health and disease, we just couldn't let that pass by us. We thought that we should understand what the microbiome is, and especially for people who work in the area of genomics, where you really needed to study the microbiome using sequencing and all of that. We couldn't let it pass by. So about 10 years ago, I started doing some work on the microbiome, but and initially started working on the skin microbiome. But that work has now been taken to much greater heights by my colleague, Dr. Shobhik Mukherjee, who will present a talk later in the afternoon. So I am not going to touch upon the work that I have done, again, collaboratively with Shobhik. He's going to present all of that. So essentially, like I said, that the title of my talk was provided by the organizing secretary or the main organizer of this event. And essentially, I'm going to borrow some examples from published literature. I do have one slide of the first work that we have done on the skin microbiome, but essentially it's going to be a more general talk because Dr. Das said that there will be lots of students who have come essentially for the purpose of participating in the workshop tomorrow and the after, and it's really an introduction to for all of them. Again, like I said, that when I went to bed last night at 11:30, I was anxious, but that's when Professor Garg started making his slides, and fortunately, there is some amount of overlap, but not very much. Microbes are all over us, like he has emphasized, and there are millions of microbes per square inch of our body, external and internal. There are thousands of different species on the skin, and they thrive on different environments on the skin, for example. Dry and humid areas of the skin. And it's estimated that there are more microbes in the intestine than the human cells in the body. And like Professor Garg essentially concluded by saying that, we need to know who we are. Are we the microbes or are we humans? Essentially, that's what it is, primarily because there are more microbial genomes in our body than our own genomes contained in our own cells. The word microbiome, just a little bit of history, the word microbiome was coined by Joshua Lederberg, and this was not so long ago in this century, essentially, or in the last century. And he defined it as the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have all been but ignored as determinants of health and disease. So it's very important symposium and the workshop that's been organized by THSTI and by Dr. Das, that these are ignored as determinants of human health and disease. And therefore, it's important for us to actually consider their contribution to our health and also to the diseases that we have. There is a huge amount of diversity of these microbes. And again, Professor Garg has actually pointed out, if you look at any part of our human body, you will find that there are multiple pie charts. The colors are distributed unequally, and you can see that there is a lot of diversity. Each color represents one particular kind of microbe, and so there is a lot of diversity in microbes across our body. Again, this is something that overlaps with what Professor Garg has said, and that we have microbes that belong to archaea, bacteria, and eukarya. Some of them have no nucleus, and others have some amount of some nucleus. Again, we need to understand all of these different kinds of domains of these microbes to be able to understand how they might impact on our health and disease. And again, like I said, that I'm going to give you one or two examples of that. If we look at microbial diversity across individuals, even in one particular organ, let's say the human gut, and if you look at two individuals, what you see is that this is, I'm not even going to read out what these colors mean. They are actually different kinds of microbes. But if you look at two individuals and if you look at their gut, all that you can see is that these two pie charts are not the same, essentially meaning that there is the proportions of the same microbe in the two guts of these two individuals are quite different. And why does it happen so? Again, various kinds of factors may actually play a role in the gut, diet being one of those key elements that plays a major role. So in the gut, the bacteria play some useful role. They convert what the carbohydrates in the gut and they allow us to absorb, but they also release hydrogen. And then there are other kinds of archaeobacteria in the gut who will, the hydrogen that's released is not good. They will absorb the hydrogen and convert it to methane, which is then expelled from the human body. So essentially, these bacteria or the various kinds of microbes that there are in the human body, they play synergistic roles in maintenance of our physiological systems, and any kind of change in those physiological systems might actually cause disease. So we have these interactions of microbes within the gut, and that ranges from early in life. And again, the microbes themselves teach our body, teach our immune system how to recognize between self and non-self. What are those components of the human body that belongs to the human, and what are those that do not belong to the humans, non-self or invaders? And the microbiome actually teaches the immune system exactly that. Throughout our lives, these microbes secrete compounds, and Professor Garg has said that we are now beginning to exploit those compounds for therapy. So these microbes actually secrete compounds that regulate immune cells. For example, the T cells in the human body, they produce various kinds of chemicals such as cytokines, chemokines, and other chemicals that are important for the maintenance of health and for warding off various kinds of non-self that's invaders. And this is all essentially done early in life by the microbes that are present in the human body. I wanted to give you a couple of examples. Again, short-chain fatty acids have been described or at least mentioned to you by Professor Garg. The relative proportion of these short-chain fatty acids that are produced by these different kinds of microbes or bacteria in particular differ between individuals who have who are healthy versus individuals who are known to be non-healthy or suffering from some disease. So here are a bunch of healthy adults and a bunch of individuals suffering from colorectal cancer. And if you look at so one of these bars, it belongs to the healthy individuals, this one, and the other belongs to colorectal cancer individuals. And as you can see, the proportion of individuals with producing different kinds of short chain fatty acids are quite different between the healthy individuals and individuals suffering from colorectal cancer. And all of these are, of course, produced by or influenced by the microbes that we have in our human body. So essentially, it belongs to the health belongs to or is an interplay between individual genetic background, and I will give you an example of how the individual genetic background actually impacts on the composition of the microbes that we have in our body. So the individual genetic background, the genomic background of the microbes that there are in our human body, and what we eat primarily, this kind of interaction, and if there is homeostasis in these interactions, that defines health, which is essentially various kinds of health definitions. We can low risk of allergies, infection resistance, and you can name a variety of other traits that define health. But when there is perturbation, then we are often prone to allergies. There are metabolic syndromes that we suffer from, obesity, various kinds of infections that we suffer from, etc. So it's basically the maintenance of homeostasis which is key, and any kind of perturbation actually leads from health state to a disease state. What we really need to understand, like our human body, like I said, that and others have said, Professor Garg has said, and Professor Gokhale has said that we are not us just alone. We carry along with us a large number of microbes that's defined as the second genome. The genomes contained in the microbes is called the second genome, and the first genome is our own genome, our own cells, human cells. That the genomes contained in the human cells is our own genome, and that's the first genome. We need to understand what's the relationship between our genome and the second genome that belongs to the microbes. If you look at variability across, I've already given you one example of variability of gut microbiome in 2 individuals. If you look at a whole bunch of individuals, and this is from our own data that was published some years ago, if you look at relative abundance of various kinds of microbes, what you really see is that there is an enormous amount of variability across individuals on the skin, across the variability of these proportions of these various microbes on the skin across individuals. Now, why does this kind of variability happen is what we really need to understand, whether this kind of changes or variability makes a bunch of individuals susceptible to some kind of skin disorders or not. This is what we need to really understand. But right now, the last 10 years or so, people have been trying to understand how much of intrinsic variability is there and to correlate the intrinsic variability to the susceptibility to various kinds of diseases. So if you look at, even if you compare 2 studies done in 2 different places, what on the same organ using a reasonable number of individuals, what one sees is that there is a lot of difference between the 2 studies, because these individuals have been sampled from 2 different environments. So essentially, the composition of the microbes in the human body will depend on the environment, and one needs to understand what's the commonality and what's the difference between the environmental factors that actually determine the various kinds of composition of the various kinds of microbes on the human body. Again, I'm not going to belabor this point because this has been very well explained by Professor Garg. We use sequencing techniques, and we use 16S, which is in part completely non-variable and hypervariable in some other regions. And then right now, in order to do even better, we do metagenomic sequencing. Again, you will learn all of these in great detail in the next 2 days. If you look at, so I come to the last 2 slides of my talk, 2 or 3 slides of my talk, essentially talking about the relationship between the first genome and the second genome. If you, the reason why we think that first genome and the second genome genomes interact is because if you look at the monozygotic twins versus the dizygotic twins, monozygotic twins will have, of course, essentially the same genome. I mean, it's like a clone. So they have exactly the same first genomes, but dizygotic twins do not have exactly the same first genome. There are differences. And what you see is that if you look at the microbiome or even the microbiota in the gut of monozygotic twins and compare that with the extent of difference between in the gut between two dizygotic twins, you will see that one sees that there is a decrease in similarity. This provides us with some cue that it's quite possible that the first genome is actually determining how much of variability there will be in the second genome. This was actually brought about in quite clearly, and there are now many more examples. This was one of the one of the examples that I would like to give. This concerns a disease called Parkinson's disease, which one does not, it's really in the brain, and one does not imagine that the gut microbiome will have something to do with Parkinson's disease. At least during the early periods when these kinds of studies were being done, nobody actually imagined. But now we understand why there is such a relationship. And so essentially, the Parkinson's disease is because of a misfolded alpha-synuclein gene, which is really the hallmark of Parkinson's disease. And this is seen to form early in disease. The misfolding is forms early in the disease, and it's shown to propagate in a prion-like manner from the gut to the brain in animal models. So that's the reason why we think that there is now a lot of reason to believe that the gut microbiome actually impacts on these so-called neuronal diseases such as Parkinson's disease. And this happens with these short-chain fatty acids that are produced by the microbiota. There is an interaction between genetic variants in the alpha-synuclein gene and dysbiosis of the gut in Parkinson's disease. And if you look at, so this is a busy slide, but I will tell you what it's all highlighted. So essentially, these are individuals with Parkinson's disease and a bunch of individuals who are controls, 511 Parkinson's disease patients and 291 control individuals, normal individuals. And what is being done is 3 different loci, polymorphic loci in the alpha-synuclein gene are being looked at. And those polymorphic loci are named as 356, 229, 100. So these are three different polymorphic loci that are being looked at. At the first locus, there are 2 alleles at that position. T and C are the 2 alleles. So you have 3 genotypes, TT, the heterozygote TC, and the other homozygote CC. And the C allele is the minor allele in this particular case at this locus. If you look at if you look at one particular kind of microbe, and if you look at the relative risk of this particular microbe in its contribution to Parkinson's disease, and if you look at it overall, then the overall this particular microbe is actually actually enhances the relative risk from 1 to 1.6, and that's significantly different from 1. So it significantly elevates the risk of that of Parkinson's disease if there is a perturbation in this particular kind of microbe. But that's also interesting that if you subdivide the Parkinson's disease patients and the controls by the genotype at that particular locus in the alpha-synuclein gene, then among the TT individuals, they have no enhancement of the disease, or no enhancement of relative risk to susceptibility of Parkinson's disease. If you look at the heterozygote, it significantly enhances to 1.9. And if you look at the minor allele homozygote, it becomes 2.5. In other words, as you move from the major allele to the minor allele homozygote, there is a significant gradation, significant increase in the relative risk to having Parkinson's disease. And this is the second genome. This is really the TT, TC, and CC are characteristics of the first genome. And what we are looking at are characteristics of the second genome that's influencing our own disease by elevating the relative risk. The same thing happens if you look at Prevotella, for example. It moves from 3.5 to 1.8 to 1, In this particular case, what you see is that the minor allele is protective, the major allele actually enhances the probability of disease or the relative risk to the disease. The only thing that we are trying to, I'm trying to establish to you through this example is that the first genome and the second genomes interact. The interaction comes in the form of enhancement of relative risk to a specific disease in the human, which is in this particular case, Parkinson's disease, but we can provide many more examples. The second point that I want to make from this particular slide is that one never imagined that microbes in the gut can actually impact on brain function. And we now also understand what's the relationship between the various kinds of chemicals that these microbes actually, the various metabolites that these microbes produce and how it impacts on the brain. So there is Professor Gokhale has mentioned about the gut-brain axis. So did Professor Garg. And so that's the gut-brain axis that we now understand. And these are very clear examples of how the two genomes, our first genome and the second genome, interact. This is more detailed. If you look at those regions where these polymorphic loci in the alpha-synuclein gene are located, and if you saturate that region with more markers, the entire story still continues to hold. So it's really not just an association, it's probably a causality, and that's what we are all trying to grapple with right now. And Professor Garg has actually emphasized that. Is it really an association, or is there some amount of causality? And understanding the relationship between the first genome and second genome can help establish that, as we have seen in terms of the Parkinson's disease example. So this is really a complex ecosystem containing predators, prey, and all of that. And we need to understand this entire ecosystem and the interactions within our human body in order to understand. This is my last slide. It really also tells us about the various kinds of interactions that we must consider and understand. And it has environmental factors, the microbiome, and the host genetic factors that I gave you an example of. That's the first genome. And then we look at the gene regulation in the host perturbed by these various microbes and the host phenotype, which is ultimately what we really are interested in, because the host type is the disease that we're talking about, and we are very self. We really need to understand ourselves, and we ourselves in relation to the other organisms that live within us, commensals or people that come from time to time, infect us and go away. I'm not going to read this entire thing. I'll just leave it there. There are different kinds of possibilities to actually examine in this network of interactions. And these are 4 hypotheses that we are all chasing, trying to understand how the host genome and the microbiome genomes actually, and the microbiome actually interact with each other. Again, like I said, from a very selfish point of view, to understand how our disease is maintained by the microbiome in interaction with our own genome, and how perturbations actually inflict disease in us. So this is a very fruitful area, and it's a very timely, well, more than the meeting, I think the workshop is very, very timely, and I see a large number of students who are going to attend this workshop, and microbiome is really becoming a very important field domain of study in the humans. And I'm saying it is becoming because there are more and more people coming into this area trying to understand, how these different axes. I mentioned about the gut-brain axes. There are other kinds of axes that are developing, and whether or not it is only an association or it's a two-way causation between the humans and the microbiome, and what the those causal elements are. This is what we are all trying to understand, and therefore there are more and more people joining this particular research domain, and that's the reason why I think that this workshop is very, very timely. And THSTI and Dr. Bhavatosh Das have played a major role in the national scene in catalyzing this these various efforts in this domain. And I thank THSTI and Dr. Bhavatosh Das and Professor Pramod Garg for organizing this meeting. The discourse today and the workshop in the next 2 days. Thank you very much for your attention.
Thank you very much, Parthada, for very nicely introducing the second genome. This was something not known to me, somebody who is new to this field. So thank you very much. I think we'll take questions towards the end. There's a panel of discussion and question answer at the end. And to introduce the second speaker, I will hand over the mic to my co-chair now.
Thank you very much, Professor Partha Majumdar. Actually, you are highlighted peaceful coexistence, I think, that we have to exist peacefully. And I have the pleasure and honor to introduce our second speaker, Professor Sharmila Mande. I don't think she needs much of introduction to this august delegate. Professor Sharmila Mande was a former distinguished chief scientist at TCS Research. She has more than 38 years of research experience in computational biology. She joined TCS in 2001 to establish TCS Bioinformatics and computational biology research and development activities. She has created a world-class group focusing on fundamental and translational research in niche areas of microbiome and metagenomics. She has more than 100 publications in peer-reviewed international journals. Her immense contribution to the field of computational biology has been recognized with several prestigious awards. And I would like to invite Professor Sharmila Mande to deliver a talk entitled Importance of Our First and Second Microbiome Informatics towards Translational Applications. Professor Sharmila Mande, please.
Good morning, everybody. Thank you for the kind introduction. I would like to first thank THSTI, the organizers of this conference and the workshop, Dr. Bhabatosh Das, for inviting me to give this talk. What I'm going to do is that I'm going to talk about some informatics, the power of informatics, what in the field of microbiome. And this has been already highlighted by Professor Garg and Dr. Rajesh Gokhale and Professor Majumdar as well about our microbiome, our second genome and our genome. And I will, in short, we know that we have many more cells than our own cells, and there are many more microbial genes that is the micro contributed by the microbiome, and which are very important. So what I am going to talk about is, before I actually go to the main topic, I just want to mention so I started this particular work almost like a decade and a half ago when we started developing algorithms for analyzing the complex data which are generated. One of the points which was mentioned by Professor Garg, that one of the important tasks is the bioinformatic analysis. So we started this for the development of the algorithms for analyzing the large volume of complex data. And then over the years, we started working on. So I'm actually very fortunate to have the first microbiome study from India in collaboration with Professor G.B. Nayak. So I'm really glad that he got the Lifetime Achievement Award. So that's what we started. And we have been doing a lot of work with THSTI, with Bhabatosh being there in some of the, most of the projects, the malnourishment project, the diabetes project, the gastric microbiome project, and so on. So these are the ones which I'm really happy that we have been working with the THSTI. Dr. Rajesh Gokhale has already mentioned about the work which we did quite a while back on the vitiligo. And this just slide shows the journey, what we have done in the microbiome field at TCS Research. I'll talk about a little bit on, I'm not going to talk about all of it, but in the next 20 minutes, what I'm going to talk about is that you all heard that there is an association between the microbiome type and the diseases and disorders. And then so there's an imbalance between amongst the different kinds of microbes which are there, and then these microbes are commensal, pathogens, and the symbionts. So in the disease case, there is an imbalance. So the question is, can we capture these imbalances? That's the question we asked. So can we identify signatures from the microbiome data, which actually makes it go from a balanced state to the in the disease state? And who are the key drivers? These are some of the questions which we asked and tried to understand from the data. Okay, and what are these signatures and where and how do we search? So in the next 15 minutes or so, what I'm going to talk about is, I'll not give the example in details, but we can discuss later on in after the session. The paths for answering the key question, that's the who, I mean, where are the signatures? How do you identify the signatures? There are two types. One is looking into taxonomic. Okay, so the taxonomic signature is one which we can look into, and there are different paths which we can follow. The first is find out the basic signatures from the taxonomic using basic statistics. You have the alpha diversity, beta diversity, identify the signatures based on using the basic statistics. That's the first path where one can look for. The second is a more complex signatures, which you may not find using the basic statistics. I mean, looking into just the alpha diversity or the beta diversity, you may not get those signatures out in certain cases. You'll have to apply the machine learning methods to identify those complex signatures. What you get is the combinatorial signature. I'll give an example of that as well. And the third path is systems level. So these microbes, when you look into the microbiome, they don't, so you can look into each individual microbes and check which are the microbes which are there in the healthy state and the disease state. But they live in the community. Like the humans, they also live in the community. So the community structure changes when you go from a healthy to a disease state, for example. Right, so you had to look into the systems level signatures. You find out the community interactions, how they are changing, and basically you will have to look into the network of these microbes to find out those signatures. So these are three paths one can look into. One will have to look into for taxonomic signatures. Now it's important that one has to understand that these microbes are there, but they are performing some functions. They are generating metabolites, which were mentioned earlier, for example. So these functional signatures are as important as the taxonomic signatures. So going beyond the taxonomic signatures, you can find out which are those functional signatures which can indicate the dysbiotic state. And these functional annotations, you can go take the basic signatures, more complex signatures, the same as what I have shown in the first three parts. Those you can look into from the functional part. So let me give you some examples of these. So the basic taxonomic signatures. So basically what you have is, when you have the metagenomic sequence data, or your 16 days based sequence data, what you have is you have the different taxa. In first stage, what you get is the basic taxa. You have the taxa 1, 2, 3, and so on, and in different samples. So what I have shown in the colors, the blue is say one type, that is healthy. It could be healthy. The green part is something to do with one disease, and the red could be another disease. So you have this taxa information. Now you want to get the signature out of it. So let me give you some examples. So we had developed we identified a microbiome-based biomarker, identified the signature for the preterm risk of preterm delivery. So the preterm delivery, I think some of the groups here in THSI is working on that. So we all know that about 1 to 1.5 million babies die every year because of the preterm complications, and 50% of them who survive, they have some problems or the other. Now what happened is, So what we did was, at the time when we did this work, there were close to 2,000 samples, which were 2,000 data sets which were available from the public domain, and which had the samples from the term and the preterm. And I mean, in each of the studies, they showed that there are differences between the taxa, between the term and the preterm cases. So what we did was we combined all of them together, and all the data sets together, all the 4 data sets. And if you plot the gestational week, which is on the x-axis, and the diversity on the y-axis, you can see clearly that in case of the preterm, the diversity was very low. They were low in the first trimester. Compared to the term cases, there are distinct differences, but up to a certain particular point. And if you can, so if you see that, so in the first trimester is the difference which is very obvious, whereas in the second and third trimester, the diversities are very similar. So you cannot use this as the marker. So when we looked into the first trimester, the differences, we tried to understand whether we can capture in some way these differences. But if you see this, that you cannot use this particular the Shannon diversity as a signature, because there is an overlap between term and preterm, right? So our question was, how do we amplify this signal to create a clear boundary between the term and the preterm? Is the question which we started with. So so what we did was we actually we actually got some clue from the the economic inequality called the Gini coefficient, which is being used in to capture how the nation's wealth is distributed across. So here, if you see the cumulative share of people in the lowest to the highest, and this is a cumulative thing. And then this has been used in economics very well. So we took the clue from here, and what we did was, here we have plotted the taxa curve in the rank from the increasing. This is the lowest, that is the least abundant ones, and that's the sparse one, and this is the most abundant one. So if we plot this and the cumulative abundance, if you plot it, so you get the similar kind of wealth distribution what you have. And what we did was, when we use the Gini coefficient, which is used in the economics, you get a good, it's better than Shannon. But we actually, for capturing this dominant versus sparse taxa, that is capturing these, because see these sparse taxa which are there, the ones which are very less in number, they are also doing some work, right? So what are the roles? The roles will have to be captured. So the capturing the dominant and sparse taxa, which are the tail here, we captured in the, these are all there in the paper, how we captured that, and we came up with a metric called as the taxonomy composition skew metric, called as the TCS metric. And this is already there in the publication, and we already got the patent granted from 7 different countries. If you evaluate these using the Matthews correlation coefficient, this taxonomy composition skew, that is the TCS metric, it performs very well in the first trimester, and then the performance goes down as expected in the second and third trimester. And this is way better than all the other indices what we had. Now, how do we validate this? You might ask that, how do you validate? We had 4, when I said that we started with 4 different data sets, 3 from US and 1 from Chinese. And what we did was we trained on one and then tried tested on the others, and they all perfectly matched. So this indicated that the microbiome can be used as an early diagnosis. So basically this is from the vaginal microbiome one. So swab can be taken to the hospital, and then you taken for sequencing, and then based on the taxonomy composition skew matrix, you can say whether the patient is as a high risk or low risk, and so on. We have other biomarkers which we have, and this particular one, we are validating this particular one with the Lady Harding. The preterm birth risk, we are already doing work with Lady Harding in Delhi. There are other ones which I'll not go into. The second one, which I said the part 2, is capturing the complex taxa signature. And this is the example which we did, which I'm putting from the pre-diabetes, which was done along with Babatosh was one of the PIs in that particular project. So if you look into there's no observed diversity because this was done on the 450 cohorts from India and from 450 from Denmark. And we don't see any major signature, but when we applied the machine learning method, we could capture the signatures, but I'll not go into the details of that. The third level, I said that you can capture the community level signature. And here is one example. If you look into these two, and this is just a small example of the two community, they look very similar, right? If you consider each of these nodes as a microbe, then you will think that these two are very similar. But if you go closely, you'll see that the members who they are sharing with are very different. So what we try to we developed a method called as the net shift from the microbiome data. What we do, we try to, what this net shift methodology or the algorithm it does is it identifies the rewiring, how the rewiring happens, the microbial community rewiring happens to come and quantify the network rewiring, identify key drivers which take from one state to another state. And this also, the details are all in the paper. And this has been used by others, and they showed some key markers. These are from the literature. They have used this methodology to identify key drivers which are responsible for some of the diseases. Now I'm coming to the functional insights. And as was mentioned earlier by Dr. Garg, Dr. Rajesh Gokhale and Dr. Majumdar, that what is there in the gut also influences other distal organs like brain, which was mentioned, liver, which was also there, liver, cardiovascular disease. So the what is there in the gut health is very, very important, because what is there in the gut microbiome that influences several different diseases which are linked to different diseases and disorders, which affect not only just the gut, but also distant organs. So the function is very, very important. One of the important functions of this microbiome is that what it does is that, there are some of these microbes, they actually assimilate the undigested carbohydrates and the undigested proteins. So the undigested carbohydrates get into the short chain fatty acids. Undigested proteins go through the putrefaction pathways, leading to some of the harmful products, releasing ammonia and so on. So, so it's very important to understand which bacterial groups are doing this. And what we try to do is that we first looked into the microbes of all different microbes, not the microbiome, and all the bacterial genomes, and try to understand the functional potential of each of these the bacteria. And what we identified, so we looked into, in this particular case, we have looked into the butyrate pathway and the putrefaction pathway, and identified 5 key pathways which are differentiating in the pathogens and in the commensals. And with that, we came up with a gut health prediction scheme, which involves actually these 5 different pathways, and came up with a store. Again, these are there, all the details are there in the paper, and which can be used for checking. I mean, if you have 2 different time points or healthy and disease cases, you will be able to see whether the health of the individual is getting better after treatment or after therapy, or between the healthy and the disease cases. How do we validate this? Because the gut health score, which we had, we checked out with the 500 different data sets which were publicly available on different diseases. As you can see from here, there are diseases which affect our gut, there are diseases which are affecting even brain and liver and so on. So all these data sets were taken, and each of these we looked into the gut health of the healthy cohort, which were published there, and the gut health of the disease ones. And in all of them, we could see the gut health was better, much better in the healthy compared to the disease cases in each of these diseases. And this again, the details are there in the publication, and the patent is already granted in one of them. To end this talk, so what I have given you a glimpse of what are the potentials of using these microbiome informatics to get the signatures for which has applications in the entire life cycle of the human. So if you look into the the microbiome's role, for example, I just showed you the early diagnosis before the baby is born. We can predict whether the baby is going to come out preterm or term. And once the baby is born, and these are some of the things which we have been working on, which I've written here. And once the baby is born, and then during the course of the time, to check the risk of different diseases and disorders, these markers can be used. And the middle ages, of course, not just the risk assessment, but also wellness monitoring, the gut health, what I was talking about, the gut health score, which we have used, can be used during the entire cycle. And this all basically, when we are talking about the future, we are talking about, you can take any sample, it could be saliva, stool, in case of the preterm, the vaginal swab, and then from the sequencing data, we'll be able to find out these markers, which I was talking about, the signatures, and based on if somebody is at a risk, the therapies, personalized therapy or diet can be recommended. So like any health equation, any equation, you have a constant and a variable in a health equation. The constant being our own genome, that's our first genome, and the variable being our second genome, which can be changed based on the diet and therapy and so on. With this, I want to thank my entire team who have been involved in the microbiome work. This is the existing team who have been involved in the microbiome work. And thank you for your attention.
Thank you, Professor Sharmila, for a nice talk, which has got tremendous public health importance, especially in maternal and child. Thank you very much indeed. For the next speaker, I pass it over to the.
Thank you. It's a real pleasure to have the next speaker, Dr. Manny Toralba. It's a good fortune that Dr. Manny got his visa yesterday morning and flew in here this morning. Dr. Manny started his research career at the Henry Jackson Foundation, looking at genotyping the GAG region of HIV-1 and treatment of naive patients using Sanger sequencing. Then continued on at the Craig Venter Institute, broadening his experience with genomics and proteomics technology. He currently serves as the Director of R&D projects in the Office of the Chief Scientific Officer at Thermo Fisher Scientific, where he focuses on using omics approaches. He's co-authored more than 70 publications in genomics research, ranging from studying the development of various cancers and autoimmune diseases, microbial communities, and so on. So welcome, Dr.
Thank you very much. Great. Yes, and yeah, just a quick story, my visa was approved yesterday morning and I left last night from right outside of Washington, DC. So I arrived late last night. So I am very jet lagged, but hopefully I'll be able to give this talk the way I wanted it to. So first and foremost, thank you everyone for attending this morning, and thank you especially to THSTI for the invitation, particularly Dr. Babatosh Das. And I'd like to give my talk today regarding industry perspectives on microbiome research and applications. So everyone is probably aware of Thermo Fisher as a company. What a lot of people aren't aware of is that Thermo Fisher actually invests over $1 billion US in research and development. We're not just a company that provides all of you wonderful people with pipette tips and plastics and containers and tubes. Last year, I believe we invested about $1.4 billion in research into product development, as well as providing solutions for researchers to make the world healthier, cleaner, and safer. Quick overview of our R&D. We have well over 7,000 employees that work in a number of different areas. That is organized into 8 different groups and 28 divisions and over 50 businesses, all working on various fields in healthcare, life sciences, applied markets, and pharma and biotech. We were very pivotal in the US response to COVID-19, as well as COVID-19 surveillance. Our network is pretty broad. We have a number of sites here in India, particularly in Hyderabad and Bengaluru. I am based right out of Washington, DC. My colleague Abhijit Afzalpakar is here as well. He's based out of our San Diego location, or our Carlsbad location, rather. We are a worldwide company, and we have well over 130,000 employees, and we cover scientific solutions in a number of different areas, from sequencing to proteomics approaches, as well as designing new types of enzymes, new workflow solutions to make your lives easier when doing these research. Okay. So as the introduction was said, I am a director of research and development in the CSO office, or the Chief Scientific Officer's office at Thermo Fisher. And what we do in the CSO's office is basically support our global R&D network. So we support all of our researchers at Thermo Fisher in doing a number of different supporting their research, essentially. We focus on 6 different areas, which includes connecting our R&D community, investment in the future. We are very much focusing on green initiatives and sustainability initiatives. And we also do a number of things where we try to support internal innovation with crowdsource funding, standardization of internal processes, facilitating communications with our corporate leadership to ensure that our research resources are going to the right programs, as well as improving R&D efficiency. So how do we support our internal innovation? We do this in a number of ways, where we promote research and development. That's one of the key goals that I have as an employee at Thermo Fisher. We promote research and development by allowing people to use various mechanisms for promoting their R&D programs. And that includes a My Idea platform, which is our idea bank, and a number of internal funding programs that we have, and they vary in different size awards. What we are looking into right now is promoting those internal funding programs to include more contributions from external parties. So if you are interested, definitely feel free to reach out to me when it comes to collaborations. So a little bit about myself. I have a PhD in cellular and microbial biology from the Catholic University of America. My background is primarily in microbiology, molecular biology, and genomics. My research experience ranges from genotyping the GAG-POL region of HIV-1 all the way to being involved in some of the first human microbiome projects there were almost 10, 15 years ago. I have an extensive research experience, which includes well over 70 publications, and my contact information is here. Like I said, if you wanted to reach out to me regarding collaborations with Thermo Fisher, we definitely welcome that. So the microbiome, you're probably going to hear a lot of the same points about it. What is it? Essentially, it's a collection of microorganisms living on the human body, and it's essentially in all body sites. From your gut to your oral cavity, your genital, skin, nasal, and even hair. I was heavily involved in the Human Microbiome Project from 2007 all the way through 2012, and that was an NIH-funded project that that focused on two different phases. One was to isolate and characterize microbial isolates that were found in the human body and create a catalog. Right now, that catalog is still in existence and has been referenced thousands of times. The second phase of that project was to essentially use metagenomic approaches to studying microbial communities and understanding what they are doing. Eventually, this research did expand into host microbiome, host microbe interactions, as well as host functional interactions. And all of this was possible through the evolution of sequencing technology. So why did this research expand so rapidly? Well, sequencing technology, as you all may know, has expanded rapidly just within our lifetimes. I started out in grad school or even parts of my undergraduate program where I was pouring those huge acrylamide gels to do Sanger sequencing in the very old school way. From that, it evolved into capillary-based Sanger sequencing, which is still being utilized today, and quickly evolved into what's now known as pyrosequencing. That started out with Roche 454 systems and eventually evolved into other technologies such as Illumina, as well as the Ion Torrent platform systems that Thermo Fisher supplies. These are very important high throughput approaches because they're able to get a lot of data in a very quick way. And you can make a number of different conclusions, or you can get a number of different conclusions from those large data sets. This can be done in a number of ways, where you can look at loci-based metagenomics analysis, and that's also known as 16S sequencing, or if you're looking at eukaryotes, you can look at 18S, fungi, you can use the ITS as a genetic marker for those analyses. Fairly recently, more so shotgun methods have been utilized to understand what these microbial communities are doing. And this essentially gives you a comprehensive view of what the microbial community is not only doing, but who is there, but also what they are doing. So the microbiome and microbiome sciences, a lot of this is going to be repetitive because this is a very specialized conference. You can look at it, you can study the microbiome in a number of different ways, but I wanted to focus on a couple of things here where we're looking at loci-based metagenomics in the rest of my talk, but there are a lot of other methods right now, including shotgun metagenomics, metatranscriptomics, and more recently, metaproteomics. There are a number of different approaches to studying this. You can look at 16S, metagenomics, metatranscriptomics, and proteomics, and there are a number of different advantages and disadvantages between each one. For the most part, a lot of the science is looking at both 16S and metagenomics, and because of the relatively ease of generating the data, as well as the cost effectiveness. The unfortunate situation with those large data set approaches is the not only the data storage, but also the data analyses. The one challenge to proteomic analyses is the database use, and it's a very tedious process and slow to medium throughput only. So those challenges with data analysis, there's a lot of limitations with big data. Most metagenomic runs can produce hundreds of gigabytes of data. Some instruments right now can produce several terabytes of data from just one run. So you have to understand what to do with all of that data. And there are a number of analysis tools out there. Most of them are open source platform. There are several examples here of open source platform systems that you can utilize. and customized to your own specifications. But the other challenges with big data includes accessing that data, securing it, storing it, most importantly, computing power. This is not something that you can just run on your MacBook. So a lot of larger companies or just companies in general look at cloud-based systems for analysis services. And that in itself costs money. And most importantly, these computing services take time. So examples of microbiome research, I'm just going to go through 3 different studies that I published on while I was at the J. Craig Venter Institute. And this is just a large range of different areas that we focused our microbiome studies on. One of which is the astronaut microbiome. I had a 5-year program with NASA to study the microbiome in 9 astronauts that lived on the International Space Station. And what we found was that, and actually, let me take a step back. What we did was essentially taxonomic analysis of both the astronauts, their microbiomes, as well as the microbiomes of the different surfaces on the International Space Station, as well as the water tanks that they were using as a source of drinking water. What we found, we took 4 samples before flight, 3 samples during flight, and 3 samples after flight to see exactly what the changes were in the microbial composition. What we found was that it was really interesting to see that all of the body sites that were exposed to the environment on a consistent basis, and that includes the skin, the nares, and oral cavity, all seem to stay pretty consistent. The microbial taxa did change while in flight to more gram negatives, but overall the microbial diversity was pretty consistent. What was really interesting was what happened with the gut microbiome. During flight, the gut microbiome diversity drastically reduced. And that's pretty consistent given that this is a microgravity environment as well as a heavily irradiated environment. They don't have the protection from intercellular radiation that the ozone layer provides for us. Essentially what we also found was that the astronauts that stayed on the station longer ended up having skin microbiomes that appeared to look like the International Space Station surfaces. So there is some cross colonization that's happening there. Another study that I published on not too long ago was on using NGS approaches to look at potential or identify potential biomarkers in oral squamous cell carcinoma. What we did was we used NGS approaches to characterize the microbial communities in tumor and contralateral oral tissue from oral squamous cell carcinoma patients. And we used 16S and metagenomics analysis. And what we did find was that there are key bacteria that could potentially be utilized as biomarkers in the development of oral cancers. These key bacteria were highly upregulated in certain, in tumor tissues only. And that includes what I call the usual suspects, which are oral pathogens, gram negatives that are known to cause inflammation, such as Fusobacterial species and certain Parvomonas species. What we also found was that there are a number of virulence factors that are highly associated in the saliva of people who have oral squamous cell carcinoma, as well as the tumors that were extracted from people who have oral squamous cell carcinoma. And these were mostly adherence factors that are in Streptococcus gordonii. But essentially, the paper resulted in potentially using oral pathogens as biomarkers for oral squamous cell carcinoma diagnostics. And this is significant because most people who are diagnosed with OSCC are diagnosed in stage 3 and 4 of the disease. Lastly, this is a microbiome project that is not really conventional. This is a project that I had going on with the University of Florence, where we were looking at using NGS approaches to study and identify bacterial species on artwork surfaces. And what is important about this is because most art conservators, and just to take a step back really quickly, art conservation is a $16 billion business. Most art conservators only focus on fungal species when it comes to preventing decay of these highly valuable pieces of art. What we found was that there are certain species of Cellumonas bacteria which metabolize cellulose, which is the main component of canvas paintings, as well as wood. And there were also Oceanospirillus species that are capable of metabolizing oil and organic-based paints. And both of those were highly detected in artwork that was going under rapid decay. And this is somewhat a pivotal moment in the study, because there are a number of artwork pieces out there that are essentially protected from the environment, but are still going under decay. So potential outcomes from this type of these microbiome research studies. You can identify microbial markers and use them to predict health and disease. Biomarkers are getting more and more common these days to determine whether or not they'll be viable for diagnostic products. Thermo Fisher as a company, we have a number of biomarker diagnostic products right now, one of which is FDA approved. There is a therapeutics potential here as well, where you can combat antimicrobial resistance. Fecal microbiota transplantation was mentioned earlier, and this is another component that we are looking to address as well as live biotherapeutics products therapy. And you can also determine this drug efficacy when studying studying the microbiome. So we are an industry leader in microbiome solutions, and the some of our main goals is producing or establishing a end-to-end workflow solution. We have a number of product offerings for microbiome research support. So feel free to take a look at any of these at your leisure. But we do focus on a number of different areas, particularly with automation in these areas here, from DNA RNA extraction, all the way through data generation. So one product I would like to highlight during this talk, and it was great to see all of the different sequencing technologies that are available here at THSDI. But one thing that I think would be wonderful for your institute to integrate into your already established workflow is our Ion Torrent system, particularly the Ion AmpliSeq Human Microbiome Health Research Kit. If you're not aware of this kit, because I definitely wasn't prior to coming to Thermo Fisher, but this is an AmpliSeq, this is AmpliSeq technology. Is that my time? Oh, yeah, I'll get through this really quickly. So essentially this is a two-pool system, and that will cover species level identification as well as microbiome profiling. It's very robust and accurate and accessible as well. And it's a comprehensive end-to-end workflow solution. In Thermo Fisher's perspective, how long should 16S sequencing take? It should take 3 days. And that is in an automated system. So this is a workflow that includes sample preparation, library preparation, and sequencing and data analysis, all of which is automated and can happen end-to-end in a 3-day time span. You get your microbiome analysis profiling with the data analysis suite, and all of this is integrated into one system. And all of this is available online, and I believe this has been updated since. So quickly, just to summarize Thermo Fisher is here to address challenges to microbiome research, and that includes solutions and areas of opportunity, from study design, using the appropriate technologies for data generation, all the way through using NGS approaches for improving point of care. And with that said, I'd like to thank all of you for your attention.
Thank you, Dr. Traorkar. And we go on to the last speaker in this session, Professor Shalimar.
Moving on, I have great pleasure in inviting the next speaker, Professor Shalimar from All India Institute of Medical Sciences. Professor Shalimar is currently working at Department of Gastroenterology and Human Nutrition at AIIMS, New Delhi. His area of research include liver failure, non-alcoholic fatty liver disease, and Budd-Chiari syndrome. He has published more than 100 articles in reputed journals. He has long been associated with THSTI in understanding the role of the gut microbiome in NAFLD. I invite Professor Shalimar to give his talk. The title of the talk is Gut Microbiome Signature in NASH. Professor Shalimar.
So I'm a clinician, so I'm a gastroenterologist. So I see patients. Most of the work is done in collaboration with Dr. Babatosh at THSTI. So what I am going to take you today out here is on a disease known as non-alcoholic fatty liver disease. So the outline of my talk would be, I'll take you through what is the burden of this disease globally and in India. Why are we really talking about this disease in this symposium? I'll briefly touch on the gut-liver axis, how this disease progresses from non-alcoholic fatty liver disease to clinical problems in terms of complications, and what is this role of gut microbiome over here in this liver disease. So this is a global map which shows you that the prevalence of non-alcoholic fatty liver disease is very common. Very common means 25% of the world population has non-alcoholic fatty liver disease. So a disease which is so common, we do not have a drug for this, so no treatment. So it's a very good disease for you to take up as a carrier. So for all of you young researchers who are sitting over here, you want to work on a disease. So this is one disease which you should really look forward to working on. I'd like to introduce you one term, which is body mass index. Body mass index means it's a simple calculation mathematical equation which includes your weight in kgs divided by height in meters square. So if your BMI is within the range of 18.5 to 23, that is what is the normal range of BMI for Asians. For Asians, if the BMI goes about 25, we define as obesity. Just remember these terms, and I would like you to calculate your BMI during this talk. One, the reason being, if you got a BMI which is in this obesity range, you got comorbidities like diabetes, you're a very high risk of developing non-alcoholic fatty liver disease. So some of you may be having non-alcoholic fatty liver disease, which you do not know of, and you do not have any problems. So let's come back to a term which is known as lean NAFLD. That means non-alcoholic fatty liver disease in those individuals whose BMI is within the normal range. Globally, this is quite a common phenomena. Even in our country, almost like 20 to 30% of patients with lean body weight may have non-alcoholic fatty liver disease. So it is not a disease which is associated only with obesity. Even those individuals who got normal BMI, or say those who consider them to be normal, may also have this disease. So this is how common this disease is. Let's come to our country. Where do we stand? This is a meta-analysis which we did, in which we included 50 studies published from India. Non-alcoholic fatty liver disease in Indians is very common. What this data suggests you that 1 in 3 adults and 1 in 3 children have non-alcoholic fatty liver disease. So now coming to the point, every 1 in 3 of you have non-alcoholic fatty liver disease, which you do not know, you do not have any problems, but disease is so common. You go to north, south, east, west, the prevalence remains essentially the same. Coming to the gut-liver axis, you heard about this since morning, and you've seen this cartoon also. Just to reiterate out here, you like eating food. In the morning, you were served with breakfast, which had lots of delicacies. You ate, your gut wanted it. You had lots of appetite, you had it. So what goes into your intestine is what your taste is, you like it. But who feasts on it is all the bacteria sitting in your intestine. So they are going to have the real feast. So once they have the feast, they're going to absorb all those things. And you heard the lean mice will get it, obese mice will get it because they've got some bacteria which cause them to absorb more or less absorb. So what is absorbed from here goes from their intestine through the veins into the liver. So who's your real pachower of thing is your liver. So liver pachows everything. That's why we heard about this. The liver is really the organ which you're looking at metabolism of all things what you're going to eat. Over here, you heard predominantly about a term called as, which I'll say the microbiome. You heard since morning about the bacteria. I'm going to take you something, all the other constituents of this gut microbiome includes the virome and the microbiome also. So in this talk, I'll highlight on these points also in relevance to non-alcoholic fatty liver disease. So this communication between the intestine, what you're going to eat and what's going to be absorbed and goes to your liver is a bidirectional thing. You in this generation are using Bluetooth, Wi-Fi to communicate. This is something which happens without any kind of transmission of any kind of a real matter. But what is this communication happening between your liver and the gut is through small metabolites. These may be produced by the intestine because of the microbiome sitting over there. That leads to some new products being formed, which go to the liver, affect its function, and liver in turn produces something because liver what helps you in is digestion. It also produces bile. Bile has lots of bile salts. They help in your metabolism. So this two-way communication is very important. And then what you heard in the morning is gut, liver, brain axis. So we're just focusing on the gut, liver axis in this talk. Okay, so when we talk about non-alcoholic fatty liver disease, I said it's so prevalent, 1 in 3, but then it doesn't cause you disease in everybody. So it may progress in some individuals from non-alcoholic fatty liver disease, just a simple fatty liver, fat in the liver, to a disease state, what we call as non-alcoholic steatohepatitis. This is a term which we are a little worried about. This may progress, this may even come back. So it's a bidirectional again, as your gut liver communication is bidirectional. You may progress to NASH and you may reverse back to NAFLD also. And if the disease progresses, you may get cirrhosis and cancers in the liver. So there are multiple factors which affect this transformation from no disease to a disease state, and in which the microbiota is one player. It is not the only player. So there are lots of other factors which play a role. Now, this is how steatosis progresses from inflammation to fibrosis and complications. Now coming to our gut microbiome. You heard there are lots of bacteria, billions of them. They account for almost 1.5 kgs of your body weight. They produce lots of things. You heard there were 4 major bacteria, what Professor Garg mentioned in the morning. So some may go up, some may go down. So it's like a balance between the good and the bad bacteria needs to be maintained to maintain a state of homeostasis. If this is not maintained, there are a lot of changes which occur, which lead to eventually in terms of insulin resistance, your gut immunity goes down, and there are some products which are produced, which eventually lead on to a disease state in the liver. So when we talk about these, what are these molecules which are being produced by the bacteria? These include LPS, lipopolysaccharides, lactate, ethanol, TMAO, and a reduction in the bile acids. These all will lead to progression from a normal to an abnormal liver. So that means when these changes occur, you're going to progress to a disease state. And when the good things are produced, you reverse back to a healthy liver. So this balance is very important. So now the microbiome, or so-called bacteria, are producing some substances. How are they affecting the liver cells? They affect the liver cells through multiple pathways. We talked about the production of short-chain fatty acids, acetates, ethanol. These are going to be causing more toxicity to the cells. So the cell is getting injured, right? Apart from that, the bile acids which we talked about are also going to produce a state through the effect on Kupffer cells, which are other cells which are present in the liver, to a pro-inflammatory state. So now what is happening in your liver cells is more damage, more injury because of these substances which are produced by the gut microbiota, leading on to more complications in terms of progression of the disease. The gut microbiome is also affected by multiple factors. You had a good food in the breakfast which was served. You like eating out. There is an increased consumption of alcohol these days. You don't like doing exercise. So there is a complex interplay of factors which are going to play a role in the gut microbiome diversity, and what we heard in the last talk, how it is measured. This slide you saw in the morning also, just to reiterate, I want you to focus on only the point which has been, the area which has been highlighted in the red box. Indians, Japanese, healthy adults, study done by Professor Bhavatosh Das and colleagues, shows that Indians have more Prevotella. This is the bacteria which is more higher in the Indian gut. Look at the fungi in the feces. Between the healthy adults, Indians and Japanese, we have more Candida. Keep this point in mind. I'll come back to it in the later slides. Why is this of some relevance to us? So as I talked in the initial part, I'll be dividing this next part of my talk into three distinct subgroups. First, I'm going to talk about the gut microbiome. That's a bacteriome. So what does the available data suggest? There is lots of studies, but I've just tried to highlight only a few of them which have an importance in this talk. One is this study compared the gut microbiome diversity in the whole cohort. And because I told you that NAFLD can occur in obese and non-obese, depending upon the BMI cutoff, which I gave you. Now, if you look at the comparison of the diversity in patients, or say when we look at the whole cohort and those who got non-obese and obese, so this diversity is significantly reduced in those individuals who are non-obese as compared to those who are obese, or when we look at a complete analysis of the whole cohort. And this also is shown in the family and genus level comparisons of the gut microbiome. Again, when we look at non-obese versus the obese non-alcoholic fatty liver disease. So there is also evidence in the literature to suggest that when you combine these changes in the gut microbiome with some changes in the bile salts, you can come up with a signature which can help you in differentiating the different stages of fibrosis. So this could be a marker of what is really going on inside the liver. So this has some relevance in terms of looking at or differentiating the severity of the liver disease in individuals. And this also performs more better in non-obese. So looking at some of the data in the Indian context for non-alcoholic fatty liver disease and NASH. So this data, though it's unpublished, is from the patients which Dr. Bhavatosh looked at in his lab and included a comparison of 17 obese and 10 NASH and 21 healthy controls. When the comparison was done between obese and healthy, there was no difference in the richness index. So essentially, they were similar in their richness index. Whereas when we look at the NASH population as compared to the obese, the richness was significantly reduced, suggesting that there are some changes which occur in this group of patients, which are a little more advanced liver disease as compared to those who are not having this disease. When we look at the diversity again, this is also reduced in patients with NASH as compared to those who are obese and those who are healthy. And this is what you heard in the morning also, is this bacteria is Collinsella aerofaciens, which is seen in a more higher proportion of patients with NASH as compared to those who are healthy and obese, suggesting potential role of this pathogen in the pathogenesis of this disease. So what is this Collinsella? So it is a gram-positive obligatory anaerobe rod. It's quite common and ferments glucose to ethanol and forms short-chain fatty acids, and it's present in almost 90% of the individuals. Sorry, the slides have gone. Can you? Yeah, take your time. So just to reiterate, in the time the slides come back, what has been shown is that in the non-obese individuals, definitely there is some differences in the gut microbiome. Yeah, this one, please. No, I'll move the slides, not an issue. Okay. Okay, yeah. So talking about the over here, yeah. So there is also available literature from elsewhere suggesting that individuals who got lean NASH have a lower abundance of Faecalibacterium and Ruminicoccus, whereas those who are obese NASH have higher lactobacilli and reduced Bifidobacterium. So going a step forward, we've talked about the gut microbiome. Let's look at what happens to the hepatic microbiome. It's a very interesting study, shows that in individuals who are lean and obese, they did liver biopsies. What they have shown is that there are higher amounts of bacterial DNA copies in the liver tissue obtained from obese, which were predominantly NAFLD, and lean individuals who are essentially healthy. And what this also showed is, this study showed that there was a lower alpha diversity in the obese population and a higher proportion of proteobacteria was seen in these individuals who had, that is, the obese have higher proteobacteria and lower alpha diversity. So in nutshell, if you look at the literature, most of the studies would suggest that yes, there are some distinct signatures which are there, which can help you or which can give us some idea regarding what is happening in terms of the changes in the gut microbiome in patients who got non-alcoholic steatohepatitis. Moving gears, now coming to microbiome. So what this data would, this small study suggested is, when they looked at the fungal microbiome composition, is that similar to what we talked about in those individuals versus like comparison between non-obese versus obese, there were significant differences in the non-obese patients with NASH or those who had advanced fibrosis as compared to those who were obese. Again, highlighting the point that possible role of microbiome in lean non-alcoholic fatty liver disease and its complications. There was a trend towards, what this study also showed is trend towards lower richness in non-obese patients, similar to what I had shown you for the bacterial the microbiome on the bacterial aspect. And though over here, the authors couldn't show a significant statistical difference. So here comes our candida, where I showed you that in the small sample size analysis, which was done in Indian patients, Indians have more candida. And candida is also seen more in patients with NASH. Probable. Could there be some role of fungal microbiome in Indian population, which is lean, which has lower BMI? This needs to be explored more further. And also, this study showed that there were differences in the ratios, and these were more significant in non-obese, again highlighting the fact that probable role of fungi in the lean non-alcoholic fatty liver disease. The authors in this study, whose data I have shown you, did an interesting experiment. They used the mice who had who had a fecal microbiome, which is in the humanized mice, treated them with amphotericin B. And they could show that the steatohepatitis, which was induced by the Western diet, could be ameliorated over here. So over here, when these mice were treated with Ampho B, which is a drug to kill fungi, these mice didn't show the changes of non-alcoholic steatohepatitis. Coming to the last part of my talk, that's the virome in non-alcoholic fatty liver disease. Again, what this data suggests is a significantly reduced viral diversity in those patients who got NASH versus those who do not have NASH. There is a lower proportion of bacteriophages in those who got NASH compared to controls. And similar changes are seen in those who got fibrosis, suggesting there is less diversity as your disease progresses in terms of steatohepatitis or in terms of fibrosis. So to conclude, NAFL pathogenesis is multifactorial. Gut microbiome possibly plays a role in the pathogenesis. And the differences in the gut microbiome are more prominent from the available literature and suggests a role possibly in lean non-alcoholic fatty liver disease. The most important point is we need still more data in the microbiome differences across population and across comorbidities. And in the end, I'd like to thank my collaborators, Dr. Babatosh Das, Dr. Sanjay Banerjee, Dr. Yashwant, Saurabh Prasanjit, and Dr. Rajni from our department, and my mentors, Professor Acharya, Professor Madhu Dikshit, Professor Garg, and Professor Ahuja. Thank you.
Thank you, sir. Since we are running behind the schedule, we will be just taking 2 questions from the audience. I request the invited speakers to kindly come over to the stage to take the questions. So please, please.
Well, looks like, people want to have tea now and they don't want to ask any questions. So we will conclude over here. On behalf of, my colleagues chairing this session, I would like to thank all the 4 speakers for giving this wonderful talk, these wonderful talks, and for nice session. Thank you very much. Looks like there is a question finally. Yeah, Sunny, go ahead, quick.
Hello, my question to Sarmila ma'am. I'm Somenu Mahapatra from Institute of Life Science. So my quick question is how you decide that you want to use Gini coefficient in this study? There are other alpha diversities also, and how we will decide in our study which alpha diversity index give us in proper input.
Okay, good question. I don't know whether you noticed that we compared with all possible diversity matrices in that, and you will get that in the paper as well. You will see that what we found was the Gini coefficient why we used is this distribution, right? The wealth distribution pattern and the distribution pattern what you see in the microbiome from the sparse to the most abundant ones. If you see the distribution patterns were very similar. So we use the Gini coefficient to check how it is compared to all the other known diversity matrices which are normally used in the microbiome. So if you notice in that particular table, you will see that the Gini coefficient was performing better than the other diversity matrices which are generally used in the microbiome field. So what we did was that taking that cue, so we started based on the Gini's one and then came up with a matrix which performs better than the Gini coefficient, which you will get it in details in that. But we did find that because of the distributions which are very similar, we said, let's see how it works. So we checked that, and I'm sure there might be other, from other fields, there might be other kind of matrices which people use in other fields, we don't know. But it just happened that we saw that the distribution, when we use the Gini, which performs better than the Simpson, Chow, and all that thing. So that's why we use it. Nice question. Thank you.
Hello, my question is regarding the network connection, as you shown, no, in between different micro. What was the basis of establishing that connections? Over to you, ma'am. That during the network analysis, you were showing the difference between the networks. What was the basis of the linkage between different nodes in the network?
Okay, so you're talking about the network, right? I mean, the network. Your question is, what's the basis of? Okay, see the network. So the basis of how do we construct the network is the co-association network. So if you see in a disease case, certain microbes occur consistently, okay, across all the disease subjects. That means they are some kind of, it's basically a correlation network, okay. These microbes are correlated. So they are interacting in some way, not directly physical interaction, but they have some association in some way, right? In case of healthy, you will see that, again, the same thing. If you see in the healthy, you see all the, microbes which are there, certain microbes which occur all the time that they are higher in case of the, say, healthy, right? So they are in some way. So it's basically a correlation based the abundance correlation across the samples, and that's how you come up with it.
If there is no other question, I think there will be many questions that can be discussed. We're closing the session for this. Thank you very much indeed.
All the session chairs and speakers for the exciting and informative session. Now I request the session chairs to kindly felicitate the speakers. Now I request Professor Sudhanshu Vrati to hand over the token of appreciation to Professor Partha P. Majumdar. Sir, please. I request Professor Subir K. Maulik to hand over the memento to Professor Sharmila S. Mande. Sir, please. Now, I invite Professor Bipin Kumar Nair to hand over the memento to Dr. Manny Toralba. I again request Professor Sudhanshu Vrati to hand over the token of appreciation to Dr. Shalimar. Now, I humbly request Professor Shinjini Bhatnagar to kindly come up onto the stage to felicitate the session chairs. Ma'am, please. To Professor Sudhanshu Vrati. To Professor Subir K. Maulik. And to Professor Bipin Kumar Nair. Thank you, ma'am. Thank you, ma'am. Thank you, sir. We now break for tea. I request the conference volunteers to assist the dignitaries to the area for the tea. We request the participants who are presenting the posters be present near their respective posters for evaluation. Since we are running behind the schedule, I request everyone to kindly assemble back by 12:15 p.m. sharp for the start of the next session. Thank you.
Me, 12:15.
I request the invited speakers of session 2 to kindly share their presentations to Dr. Ilango, who is near the stage.
May I request everyone to please be back to the auditorium? We can begin with the next session, please. Welcome back, everyone. After a very exciting and informative session and some brainstorming over tea, now we begin with the second session for the conference on microbiome and health and inflammation. For this, I humbly call upon the session chairs, Professor Sharmila Mande, Professor Shalimar, and Dr. Jiva Shankar to the stage, please. Dr. Jiva Shankar is working as an additional professor in the Division of Neonatology, Department of Pediatrics at AIIMS in Delhi. Earlier, he worked as a medical officer in the Department of Child and Adolescent Health and Development, WHO Geneva. His primary area of research interest is neonatal sepsis. He coordinated the multicentric study on sepsis involving around 15,000 neonates in 4 large tertiary care hospitals of Delhi. Welcome you, sir. In this session, each talk will be of 20 minutes, followed by a 10 minutes long question and answer after all the speakers finish their talk. I request the audience to kindly reserve the questions until the session is completed. Since we are running behind the schedule, I request the speakers to restrict their talk to the given time. A buzzer will be given at the end of 20 minutes. I now request the session chairs to introduce the speakers and call them over for the talks.
It's my pleasure to introduce the first speaker of this particular session, Professor Shaw. I would invite now Professor Saurabh Datta. He's currently a professor in newborn unit, PGI-MER, Chandigarh, India, and is adjunct clinical professor in Department of Pediatrics, Manchester University, Canada, neonatal sepsis, probiotics, and retinopathy or prematurity are some of his main research areas. He has published more than 150 publications in reputed journals. He is also a member of many national and international professional organizations, including the American Academy of Pediatrics, the Pediatric Research and Education Society of India, among others. Professor Saurabh Datta, please.
Good morning, and thank you for that kind introduction. I would like to thank my good friend Bhabatosh and my also another good friend, Dr. Shinjini, for this very kind invitation to speak about breast milk microbiota of Indian mothers. Now, I'm a clinician. I'm a newborn specialist in PGI, and some of these definitions about diversity, etc., get very complex for me. So I'm going to start off with a very simple definition on my first slide. And that is, what is the definition of a baby? So a baby is essentially an alimentary canal that is very demanding at the upper end and has no sense of responsibility at the lower. With that, let's get on to this understanding that most of the gut colonization of a newborn infant or an early infancy is because of microbes in the breast milk of the mother. That's a thing that has been understood and established over the last 10 or 15 years. That was not the case when I was a resident. We were always taught that the mother's breast milk is completely sterile. And it was always a mystery as to how gut colonization took place in the first few weeks of life. So how does human gut colonization proceed? So this is normal colonization in a breastfed term infant. This is the oversimplified diagram. So basically after birth, a whole lot of aerobes first get into the gut, and they create an anaerobic environment. In that anaerobic environment, then a whole lot of other anaerobes get in, bifidobacteria being the most important, probiotic organism in early infancy, which is later replaced by lactobacilli as the predominant lactic acid bacterium. And subsequently, you have Bacteroides and Enterobacteriaceae making their entrance. And it is only well beyond 2 years of age that the gut microbiome of infants actually gets well established. So from where do the first bacteria get into the neonatal gut? It was way back in 1975 that this very simple experiment was done. Carlson et al., they went to the delivery room. The baby's mouth was swabbed in the delivery room, and they also swabbed the mother's rectal and vaginal swabs, and they cultured it for lactobacillus species. And they found, to their not to their surprise, it was kind of expected, that there was a very high correlation between the oral cultures of the babies and the rectal and vaginal cultures at birth. They grew the same strains of lactobacilli. And they also found that the source of the vaginal lactobacilli is actually the mother's own intestine. On day 6 after birth, they found that the oral cultures had become negative, but the neonatal stool cultures were now growing exactly the same organism. So they concluded that these were bacteria got during the process of birth, with the baby ingested, and which later on colonized the baby's intestine. So the message was that the first inoculum of bacteria came from the mother's vagina and the perineum. Although this has now been challenged because we have increasing evidence that the fetal gut is probably getting colonized even before the baby is born. So
the next question that was asked was, is breast milk a source of bacteria for the neonatal gut? And this was a landmark study done by this group in Madrid, Rodriguez et al. They published it in 2003. So this is all information which has got only in the last 20 years, actually. But prior to that, we always thought it was sterile. So about 20 years ago, they studied 8 mother-infant pairs. They did cultures and rapid PCR of breast milk collected aseptically. They used a whole lot of precautions. They also collected cultures of the breast skin, vaginal swab, neonatal feces, and the neonatal oral swab after feeds. And what they found was, at that time, revolutionary. They found that breast milk was not sterile. It had a huge number of bacteria. There was a very high concordance, identical profiles of the neonatal feces and neonatal oral swabs with the breast milk. And there was actually no concordance with the breast skin flora and the vaginal flora. So they concluded that the breast milk was a huge source of organisms for the neonatal gut, and it was actually colonizing the neonatal gut. So the message was the breast milk has bacteria that colonize the neonate's gut. So then the question was, which was the more important source? Was it the vaginal flora at the time of birth, or was it the breast milk later on? And from where on earth were these bacteria actually getting into the breast milk? So initially the hypothesis was that the baby's mouth was first getting colonized, and because the baby was sucking at the time of feeds, the breast milk was somehow getting reverse colonized from the baby's mouth. Because unlike a whole lot of other organ systems which get colonized, like the GI tract, the respiratory tract, the genitourinary tract, the human breast doesn't have that kind of an access to the outside environment that to get colonized with outside bacteria. So this was a puzzle for a long time. And that puzzle was finally solved in 2007, where this paper was published. They collected breast milk aseptically from 5 mothers with elective cesarean section. LSCS is lower segment cesarean section, with no rupture of amniotic membranes. So that means these babies had no contact with the outside world, outside environment, when this breast milk sample was taken. They took 5 mothers who had delivered vaginally, and they took pre-colostrum even before the baby had delivered. And they extracted bacterial DNA, and they did all the usual stuff that people do in microbiome research. And what they found was there was no difference in the breast milk microbiota between the LSCS, means the breast milk composition, between the cesarean and the vaginally delivered babies. So that means these babies who had cesarean borns did not get a chance to actually go through the birth canal and get the oral colonization done. And despite that, the microbial colonization composition was almost the same in the breast milk. Breast milk, of course, was replete with bacteria, which many authors had shown by that time. And it was present even in the pre-colostrum before delivery. So no question of the baby sucking and then it getting reverse colonized. So then the question was, if there are so many bacteria, why don't they cause disease? I mean, if it's thousands of bacteria. So in 2005, this group published a paper in which they checked one of these species for pathogenicity. After that, many authors have checked it for pathogenicity. And so they looked at Enterococcus faecium, which is otherwise known to be a deadly organism causing sepsis in newborn infants. But they looked at virulence determinants, genes involved in plasmid conjugation, resistance genes, etc., etc. And they found that these bacteria grown from human milk had no virulence genes, none of those things that actually make organisms virulent and cause disease. So they concluded that possibly, all these bacteria in human milk are avirulent, and they have to be, because otherwise it would be a disastrous for the baby if there are virulent bacteria in human milk. So then this question was asked, okay, if it is not by reverse colonization from the neonate's mouth, how do the bacteria get into the breast milk? And this is a unique hypothesis that has been put up. It's been widely accepted. There's a lot of experimental evidence, although the final word on it has not been said. There exists a gut mammary axis in mothers. That means bacteria get transported from the mother's own gut during the end of pregnancy and lactation to the mammary gland, and that is what colonizes the breast milk. So the hypothesis is that highly specialized immune cells, probably dendritic cells from the gut, pairs, patches, and lamina propria, somehow home in on the mammary gland during late pregnancy and lactation, and they actually act as vehicles or carriers of probiotic commensal bacteria from the mother's own gut. And so that means the mother's gut and the baby's gut are very strangely intertwined in a way, because bacteria from the mother's gut are finally finding their way into the baby's gut. So the evidence for the gut mammary axis for bacteria was provided by Perez in this absolutely wonderful study published in 2007. It's really worth reading. So they took human mother-infant pairs in which they did PCR and TTGE, and they took samples of the mother's feces, mother's blood, mother's milk, and then they took infant feces. And they showed that there are identical signatures in the mother's feces, mother's blood, mother's milk, and in the infant feces. This absolutely the same organisms down to the genetic level. They also visualized bacteria inside milk monocytes and peripheral blood monocytes and showed that they were the same. And they also did experiments on mice showing that during lactation, there is a huge increase in the number of bacteria in the mammary glands of lactating mice. So then, which are the cells that carry these bacteria? It has been, imagine this is the cartoon of the mother's intestine. So this is the gut lumen. These are the mucosal cells. The dendritic cells are hypothesized or believed to be doing this job. So the dendritic cells have processes. So, the extended processes between the mucosal cells, they actually sample and pick up these beneficial bacteria, get it into the cells, somehow preserve those cells, and they are carried to the mammary gland. So through the bloodstream and lymphatic, and finally the baby, suckles on the mother's breast and is able to get these bacteria. Now, if these bacteria are coming from the mother's gut, and the mother's gut microbiome, as we have heard since morning, is affected by her diet, by ethnicity, by race, by antibiotics, and so on and so forth. So would country of origin affect the composition of the breast milk microbiome? So one of the first people who studied this was Kumar in Finland in 2016. This is published in Frontiers in Microbiology. So what he did was took 10 healthy, exclusively breastfeeding mothers from 4 countries, and they looked at the V4 region of the bacterial 16S RNA gene. So they did a 16S metagenomics using the V4 region. So these are the 4 countries they took: Spain, China, Finland, and South Africa. And here are, your familiar 4 major phyla: the proteobacteria, firmicutes, bacteroides, and actinobacteria. And as you can see, there is a major difference across countries. For example, South Africa, which was, a developing country in the tropics, there is a much higher proportion of proteobacteria in the milk. There is China, for instance, had the largest amount of actinobacteria, and so on. So milk from mothers in different parts of the world has different microbial compositions. This was another study in 2019, the INSPIRE study, where they took 11 sites across the world, including some rural and some urban. So they looked at the V1 to V3 region of the gene. And I would just request you'll have to tilt your heads to look at these sites. So they took Ethiopia, a rural site, Ethiopia, an urban site, Gambia, rural, Gambia, urban, Ghana, Kenya, Peru, Spain, Sweden, and 2 sites in the US. So it had there were representation from across the world, and the number of patients is mentioned here, which is a reasonable number at each site. Now, I would like you to focus on this yellow one, which is proteobacteria. Proteobacteria is often, maligned as that phylum which has, most of the pathogens and gram-negative pathogens. Interestingly, there was a lot of difference across the sites as far as the proportion of proteobacteria was concerned. For example, rural Ethiopia, almost half, 50% was the abundance of proteobacteria. Similarly in Ghana, as opposed to many other sites. When they looked at the actual genera at the genus level, the difference, these are all the colors here. You can see that there are huge differences in proportion of various genera. And I want you to focus on Pseudomonas because I'm going to be talking about Pseudomonas in a little more detail later. Pseudomonas is this gray bar here. You can barely see Pseudomonas in this gray thing in most of these sections. So not much of some Pseudomonas being described from Gambia, but not otherwise. And just keep this in your mind. This is again differences between countries. So Lee et al. In 2017 looked at 6 regions of China and Taiwan. I mean, for a change, China and Taiwan actually cooperated for something. So here is from 6 regions of China and 1 in Taiwan. They looked at again 16S RNA gene. So without going into details, as you can see, the proportion of these various segments is quite different in different places. If you focus again out here, as I'm saying, on Pseudomoniaceae, that is the family that has the Pseudomonas genus, the orange one, you can see that there are certain regions that had very high predominance of this Pseudomoniaceae family. There are others that did not, and there's a lot of variability even within a country. So differences between regions and the Pseudomonas containing family is dominant in certain regions. So the question is that since it is so different across different parts of the world, are Indian mothers any different from the rest of the world? So this is a study that we had done in collaboration with Dr. Bhabatosh, published last year. We took 20 breastfeeding healthy term mothers, full-term normal delivery with no recent antibiotics. So as pristine as possible. And with surgical antisepsis and with great care to avoid contamination, we looked at the V1 to V5 regions. We amplified the 16S RNA gene and did metagenomics. So these lactating mothers, their mean age was 27 years. The mean postnatal age after delivery at which we took samples was about 4 days. Mean gestation about 38 weeks. And we identified 39 core genera with an abundance of more than 1%. Now, this is what we found. When we looked at the overall phyla, we were astonished to see that it was almost completely dominated by proteobacteria. So this was the proportion of proteobacteria and the other phyla. And what was even more astonishing when we looked at the genus level was that the Pseudomonas genus was by far the most important genus, compared to any other. Now, the moment clinicians hear the word Pseudomonas, their blood pressure shoots up because it is associated with all kinds of deadly diseases in all age groups. So the next thing we did was to see what were these Pseudomonas genus species. So we went down to the species level, and if you just glance through this, none of these are the kind associated with diseases. Usually it's aeruginosa and various other species which are associated with. So none of these Pseudomonas genus was known to be associated with diseases, which was of course gratifying. Then we found that when we clustered patients based on the relative abundance of Pseudomonas and alpha diversity as measured by the Shannon index, there were 3 subgroups that seemed to emerge from our small group of patients: those with high Pseudomonas and low diversity, low Pseudomonas, and high Pseudomonas, high diversity. Of course, we did not have a huge sample size, but there were 3 patterns that seemed to be emerging. And this is a heat map again showing the relative abundance, ranked abundance of genera separated according to clusters, which in principle is showing the same thing as the previous slide. Now, were there any other studies from India or developing countries showing similar data? Well, there's only one study from India, Vaidya et al., published in 2017 from Gujarat, where they took 15 urban and 15 rural healthy lactating women and looked at the V2 to V3 region. So this was the Vaidya study on the left-hand side, and they compared it with data from Finland and Switzerland. And look at the marked difference. In this study, again from India, this blue is all proteobacteria. So proteobacteria is by far the commonest phylum in the breast milk of healthy lactating Indian mothers. When they went down to the genus level, same Vaidya et al., the relative abundance of the top 20 genera, this is urban Gujarati women and this is rural Gujarati women. You can see, at least in the urban population, these dark blue vertical bars is all Pseudomonas. So they also reported a very high abundance of the Pseudomonas genus. They did not go down to the species level to tell us what it was. They looked at the difference between India, Finland, and Switzerland. So these are color coded here. They did a weighted unifrac distance where they showed that these were three completely different populations or microbiomes in three different countries, completely different. The distances were huge. There was one other study published in 2017 where they had mothers with mastitis and 18 healthy controls. So I was interested in the 18 healthy controls, and the data of the 18 healthy controls are shown here. Again, the proportion of the major phyla was rather similar to the previous two Indian studies, a lot of proteobacteria in the mother's milk. So the Pseudomonas in human milk, so the question is, could they be at all pathogenic? Are they capable of colonizing the neonatal gut? And is it reported elsewhere also from the rest of the world, or is it something peculiar that we're seeing in India? So firstly, can the Pseudomonas be pathogenic? So as I told you before, no Pseudomonas species detected in our study was a known pathogen. So we did a detailed literature search. So, these are all the organisms. Some had been reported to cause sepsis in highly immunocompromised adults, or only when contaminated IV fluids were given. No reports at all in the pediatric age group. All neonates in our study were clinically normal on 1 month of follow-up, as was expected. And there have been no reports of any Pseudomonas species infections attributed to human milk so far in literature. So it's all safe. So can this colonize the neonatal gut? So there was this study, very elegant study done in which paired milk and neonatal stool samples were taken at weeks 1, 3, 6, and 12. So this is at the phylum level. So out here on the horizontal axis, weeks 1, 3, 6, and 12. So each of them has two vertical bars. The first is in the milk. The second is the neonate's feces. And just concentrate on the purple parts because they correspond to proteobacteria. So what they found in the study that even when milk has a high proportion of proteobacteria, that is not really reflected always in the neonatal fecal sample. So the proteobacteria in the neonatal gut is always less than what it is in the mother's milk. When you go to the genus level and look at the blue here, which is Pseudomonas, so there is Pseudomonas in milk out here reported in this study, but when you come to the stool samples, there's virtually no Pseudomonas genus being reported in the stool sample. So same paired sample. So there's Pseudomonas in the milk, but not really. So the Pseudomonas in the milk, we don't really know what it's doing there, but it is not an efficient, it does not seem, at least from this single study, that it's an efficient gut colonizer of the neonatal gut. So have there been other reports of Pseudomonas? I've already talked to you about the Kumar study from 4 countries where mothers who delivered in South Africa had a relative abundance of, high abundance of Pseudomonas. And these 2 studies from Gujarat, which had reported Pseudomonas. So it is widely reported, though not as a dominant genus from all parts of the world. So is the high relative abundance of Pseudomonas genus more of an Indian or a developing country phenomenon? That's a question that needs to be answered. And the other question that, why is there so much of variation in breast milk flora? One would expect breast milk composition to be kind of stable throughout the world. It's such a basic nutrient for neonates. So how come it's so different? So of course, the possible, reasons for variation are the usual suspects: antibiotic exposure, dietary factors, geographical and ethnic factors. So there is evidence that antibiotic exposure alters the composition of breast milk. Dietary factors are not well studied. In fact, it is the subject of an ongoing PhD thesis right now in PGI. And it seems at the end of the day that each mother almost at an individual level seems to have a unique microbiome based on her own individual microenvironmental factors. Maybe that's what we're finally dealing with. Multi-drug resistant bacteria in breast milk. This is the result of a DBT project which we have finished and yet to be published. So we took 100 lactating mothers delivered at 28 to 34 weeks of gestation, and we looked at the antibiotic administration to the mothers versus multi-drug resistant organisms, MDRO, extended spectrum beta-lactamase, and carbapenem resistant gram-negative bacilli in breast milk and neonatal samples. And to cut a long story short, if you look at the green bars, then the administration of amoxicillin, there was a major increase in the presence of multi-drug resistant organisms, both in the breast milk and the baby's oral swabs. So giving antibiotics to mothers is altering the composition of their bacterial flora, creating multi-drug resistant organisms, and making the neonatal gut get colonized with a resistome, not just a microbiome, a resistome being inherited from the mother through breastfeeding, which is a dangerous thing. So to sum up, there are wide variations in breast milk microbiota between countries and even within countries. The phylum Proteobacteria seems to be predominant in developing countries, including India. The genus Pseudomonas is abundant in Indian lactating mothers, of course, based on limited data, just from 2 or 3 studies, and in South African women. So there seems to be some thing going on in the tropics or developing countries. These species, of course, all non-pathogenic. And there are high levels of multi-drug resistant organisms in mother's milk, especially if they happen to be treated with broad spectrum antibiotics. Thank you very much.
So it's my pleasure to invite Professor. So it's my pleasure to invite Professor Kiyoshi Takeda. He's the Director and Professor of Immunology Frontier Research Center, Osaka University. His research activity is focused on understanding the pathogenesis of inflammatory bowel disease, particularly the analysis on how intestinal homeostasis is maintained by mucosal innate immune cells and epithelial cells. He's also analyzing how microbiota reside in the intestine without evoking inflammatory responses and influences the host health. Professor Takeda has published more than 200 research articles in top journals, including Science, Nature, Cell. His topic is host microbiome interaction in inflammatory bowel disease. Professor Takeda, please.
So thank you very much for the kind introduction. And I'd like to express our sincere appreciation to the Professor Bhavesh Das for inviting me to this nice symposium. So I'd like to talk about the host microbiome interaction in the inflammatory bowel diseases. So actually our laboratory is analyzing on the pathogenesis of the inflammatory bowel diseases represented by the ulcerative colitis and Crohn's diseases. So recent evidence have indicated that the genetic predisposition, particularly intestinal immunity and microbiota, both contribute to the pathogenesis of the inflammatory bowel diseases. Indeed, the complicated interaction between the intestinal immunity and microbiota contributes to the development of the inflammatory bowel diseases. But between this intestinal immunity and microbiota, there exists a single layer of the epithelial cells. So we wondered if these epithelial cells have some barrier function to separate the microbiota from the intestinal immune cells. So in this regard, epithelial cells of the gastrointestinal tract are covered by the mucus layers. And compared with the epithelial cells of the stomach and small intestine, the colonic epithelial cells are covered by the very thick mucus layers composed of the outer mucus layer and inner mucus layers. So when we stain the mouse colon, red stain microbiota are really abundant in the outer mucus layer regions. But when we see these inner mucus layer regions, which is visualized by the back two mutations, this inner mucus layer is essentially be absent. The microbiota is essentially absent in these inner mucus layer regions. Thus, by the presence of this inner mucus layer, microbiota, commensal bacteria, are separated from the host cells. For the mechanisms for this separation of the intestinal microbiota and the host cells, general concept so far accepted was that because these mucus are really slimy, this slimy mucus mechanically prevents the invasion of these bacteria. But we simply wondered if this is only one mechanism. And so we found the unique molecule named LYPD8. This LYPD8 is selectively expressed in the gastrointestinal tract, particularly in the cecum and the large intestine. And in the large intestine, this LYPD8 was expressed exclusively in the epithelial cells, but not in lamina propria lesions. So my former PhD student, Liu, analyzed the function of these molecules and found that this LYPD8 was expressed on the uppermost layer of the colonic epithelia and secreted into the intestinal colonic lumen. And this intestinal lumen, LYPD8, binds to the flagella of the highly motile gram-negative flagellated bacteria. And after binding to the flagella, LYPD8 suppresses the motile activity of this commensal bacteria. And indeed, in the absence of the LYPD8, this highly motile flagellated bacteria invade into the epithelial cells, actually colonic lumen, colonic tissues, and which increase the risk of the intestinal inflammation. So, commensal bacteria, microbiota, are present in the intestinal lumen without directly attaching with the host cells. But intestinal microbiota are well known to influence the host cell functions. In this regard, bacterial metabolites, particularly short-chain fatty acid, are known to act on the host cells, such as the epithelial cells and the intestinal immune cells, through the G protein coupled receptors. So in addition to the short-chain fatty acid, we wondered if other bacterial metabolites also act on the host cells. So in this respect, we focused on the unique intestinal immune cells, myeloid cells. These intestinal myeloid cells are characterized by the high expression of the chemokine receptor CX3CL1. And these CX3CL1 positive myeloid cells are present just beneath the intestinal epithelial cells and extend their dendrite directly into the intestinal lumen and uptake the luminal antigens and subsequently activate the effector T cells. So we wondered if somewhat bacterial metabolites mediate this dendrite protrusion of this CXCL1 positive myeloid cells. So my PhD student Naoki tried to identify these bacterial metabolites. And he successfully identified that bacteria-derived lactate and pyruvate actually induced the dendrite extrusion of the intestinal CX3CL1 myeloid cells through the GPL31. So this photo is derived from the villus of the terminal ileum. So in the case of the wild type animals, as you can see here, dendrite extension can be observed. And after the oral treatment of the lactate or the pyruvate, the number of the dendrite extension was tremendously increased. But in the case of the GPR31 knockout animals, dendrite extension cannot be observed at all. Even after the treatment with the lactate or pyruvate, dendrite extension cannot be induced. Thus, the microbiota-derived lactate and pyruvate induce the dendrite protrusion of these CXCR1 positive myeloid cells through the GPR31. So, microbiota-derived metabolites mediate the host microbiota interaction for the maintenance of the intestinal homeostasis. So our next question is, these microbiota-derived metabolites are involved in the pathogenesis of the inflammatory viral diseases. So my PhD student Yuriko and my staff Hisako addressed this question. So we first performed lipidomic analysis of the plasma of the Crohn's disease patients. And actually we found several lipid metabolites were increased in the plasma of these Crohn's disease patients. Among these metabolites, lysophosphatidyl was most increased in the Crohn's disease patients. This lysophosphatidyl was produced from the phosphatidylserine by the action of the enzyme named phospholipase A or PLA. And this DPLA gene was expressed in the host and several bacteria. So we wonder if this biosis correlates with this increased concentration of lysophosphatidyl in Crohn's disease patients. So we performed shotgun sequencing analysis of the feces of the healthy controls and Crohn's disease patients. So actually, this biosis can be observed in Crohn's disease patients. So because this is a shotgun sequencing, we precisely analyze the existence of the genes encoding the phospholipase A. So actually several bacteria possess this phospholipase A gene. Among these phospholipase A gene, E. coli-derived phospholipase A gene named ECSF3660 was really abundant in the Crohn's disease patients. So, in Crohn's disease patients, we found the increased number of the microbiota possessing the phospholipase A genes and increased concentration of the lysophosphatidyl both in the, actually we do not show the data, but this lysophosphatidyl concentration was increased in both the plasma and the feces. So our next question is whether this lysophosphatidyl is involved in the pathogenesis of the intestinal inflammation. So we performed our mouse colitis models, analysis with the mouse colitis models. So LAG2 immunocompromised LAG2 knockout animals were transferred with naive T cells. In this case, these LAG2 knockout animals developed mild colitis. At the 17 days after the transfer of naive T cells, lysophosphatidyl was administered. In this case, administration of this lyso-PS induced the development of really severe intestinal inflammation, and they showed rapid weight loss. Thus, administration of this lysophosphatidyl induced severe intestinal inflammation. In these models, we then analyzed the number of T cells. So actually, we analyzed the number of the interferon gamma producing or IL-17 producing CD4 T cells in the colonic lamina propria at 21 days after the transfer of the naive T cells. So in this case, we found that after the lyso-PS treatment, the number of the interferon gamma producing Th1 cells or interferon gamma and IL-17 double producing pathogenic Th1 cells were increased in the lyso-PS treated LAG2 knockout animals. Thus, lyso-PS, lysophosphatidyletherine treatment worsened the severity of the intestinal inflammation together with the increased number of the Th1 cells. So we then analyzed whether this lyso-PS directly acts on the CD4 positive T cells or not. For this, we performed the in vitro analysis. Naive T cells were cultured in the Th1 polarizing conditions, and 24 hours later, lyso-PS was administered. In this case, addition of lyso-PS increased the production of the interferon gamma from the Th1 cells, thus indicating that lyso-PS, lysophosphatidyletherine, directly acts on the Th1 cells to increase the production of the interferon gamma. So we next analyzed how this lyso-PS act on these CD4 positive Th1 cells. In this regard, we analyzed the host receptor of the lyso-PS. So actually, several lyso-PS receptor was identified. And these receptors are all G protein coupled receptors, such as GPR34, P2Y10, and GPR174. So we analyzed the expression of these G protein coupled receptors in Th1 cells because the lyso-PS act on the Th1 cells directly, and found that P2Y10 is highly expressed in the Th1 cells. So we analyzed whether this P2Y10 is essential for the lysophosphatidyletherine dependent aggravation of the intestinal inflammation. So we produced P2Y10 knockout animals and introduced in these LAG2 knockout colitis models. So LAG2 knockout animals were transferred with the naive T cells of the wild type animals and P2Y10 knockout animals. So in the case of the transfer of the naive T cells from the wild type animals, they induced mild intestinal inflammation. But in the case of the transfer of the naive T cells from the P2Y10 knockout animals, it did not induce any inflammation. And in this case, at the 20 days after the naive T cell transfer, lyso-PS was administered. In this case, lyso-PS administration induced very severe intestinal inflammation, and they showed rapid weight loss. In the case of the LAG2 knockout animals transfer with the wild type naive T cells. But in the case of the LAG2 knockout animals transferred with the P2Y10 knockout naive T cells, lyso-PS treatment did not induce anything. Thus, lyso-PS mediated exacerbation of the colitis was dependent on the P2Y10. So I'd like to summarize that this presentation. In the Crohn's disease patients, we found that the increased number of the microbiota possessing the phospholipase A genes. And accordingly, lysophosphatidyletherine concentration was increased. In this condition, this lyso-PS directly act on the Th1 cells through the P2Y10 and activate the Th1 cells. This leads to the exacerbation of the intestinal inflammation. So I'd like to acknowledge the contributors of this presentation. Actually, the P2Y10 and the lysophosphatidyletherine analysis was performed with the Hisako and Yuriko and the collaboration of the Osaka University Department of Gastroenterology. So thank you very much for your attention.
Thank you, Professor Takeda, for the excellent talk. As you're joining online, maybe we can have questions for this talk right now. If there are any questions from the audience. Yeah, can you please pass on the mic or?
Dr. Takeda, can you hear me?
Yes.
I was just wondering, probably this is your futuristic work, but is there any molecule, small molecule that you're trying to target the P2Y10?
Oh, actually, at this moment, we have no access this kind of the small molecules. So if you are interested in this, let's collaborate with us.
Thank you very much.
Professor Takeda, that was a fascinating talk. I was intrigued by one of your slides where you said that inner mucus does not have bacteria. But subsequently, we have mucosa-associated bacteria. And thereafter, are you aware about the crypt microbiota? Do you think that the crypts can also have microbiome?
Oh, yes, actually, is in the mucus, several bacteria do exist, but the number of the bacteria is really small. So this kind of the barrier function by the mucosa is, I believe, essential for the inhibition of the intestinal inflammation.
So do you think that it would be counterintuitive in the sense that the bacteria which are more closer to the cells, they would probably play a major role, whereas the bacteria which are there in the fecal stream, they may be more influenced by environmental issues like antibiotics, diet or something. So probably the more important bacteria would be the one which are beneath much.
I see. Yeah, right.
Okay, thank you so much.
Thanks, Kiyoshi. It's a nice talk. So you looked into TH1 cells. Have you tried whether the Tregs or become non-functional? The first thing. The second thing is, what is the role of IL-23 and TH1 like cells, which is now playing a major role in IBD pathogenesis?
Right. Thank you very much for the question. So actually, we also analyzed the number of Treg cells, but the number of Treg cells was not changed in this model. And so we do not analyze the ILC3. We should analyze this. Thank you very much.
Hi, Professor Takeda. It's a very nice talk. I have just one question. The mucus barrier function, do they differ depending on the host genotypes? Do you find that?
Pardon me?
The mucus barrier function, do they differ depending on the host genetics, or it's universal for all?
At this moment, we do not this. So I'm sorry, I do not have any answer at this moment.
Okay, if there are no more questions, we'll end the talk over here. Thank you, Professor Takeda. Thank you.
Thank you very much.
So we'll move on to the next talk. I invite Professor Amit Avasthi. Professor Amit Avasthi is presently working as a senior professor at THSTI. His research focuses on interplay of effector and regulatory T cells in IBD and gut infection, understanding T cell differentiation pathways in immunity, autoimmunity, and cancer immunity. He has several honors to his credit, and to mention a few are prestigious awards like CDRI Award 2022 in Life Science category for drug discovery, G.N. Ramachandran DBT Bioscience Award of 2021 for carrier development, and he was selected as a member of Guha Research Conference recently in 2021. Professor Avasthi, please.
Thank you. Thank you very much. So I would like to thank Dr. Bhavatos for a great, 3 days, 1 day seminar and 2 day symposium the works, hands-on work training. And this is a great opportunity, especially for the students who like to jump into this microbiome field, which is now actually from the morning we are listening that it is affecting or influencing in a way every single disease or normal human physiology, right? So today I'm going to continuously gonna this talk on IVD, and we know it's a kind of there is a direct correlation and association between intestinal inflammation with gut microbiota. So this is the team which I often forget at the end to acknowledge them. So I decided to acknowledge, and the work today I'm going to present a PhD student who submitted his thesis, Rajdeep Dalal. So basically, this is the outline of my talk. We're going to have, my talk is divided into three parts. One is the dietary potassium suppresses intestinal inflammation and experimental colitis. The second one is dietary potassium induce primary bile acid, cholic acid from the liver hepatocyte, which is actually suppresses inflammation. And finally, we identify a novel synthetic cholic acid derivatives, which is targeting effector cells called Th17 cells by inhibiting the function of Th17 cells. So in my talk, you will understand how the basic immunology lead to the translation and then ultimately identify a drug molecule. So this is perfect example of drug discovery. And Dr. Vineet is here. So some of the work what actually Dr. Vineet is independently doing in RCT, in some of the work what I'm actually, so it's kind of two independent work, one at the clinic setting and one is the experimental settings. So this is just want for the audience to remember this. If you have a healthy gut, you have the Th17 and Th1 cells, and these anti-inflammatory cells normally live in the harmony. They are always in the homeostasis, right? And the moment you have a kind of the inflammation, whatever means, it could be environmental, it could be genetic predisposition, or whatever the way it is, there is an inflammation in the gut, and then you have more inflammatory T cells accumulation in the gut, in the tissues, and there is a lesser number or the functionally the regulatory or anti-inflammatory T cells. So what we actually essentially like to do at this point of time, we know this axis is kind of disbalanced, and we would like to bring this axis more into function and this axis more into suppression so that we can bring back the harmony of effector and regulatory T cells during intestinal inflammation. So this is what we wanted to do. So normally if you see inflammatory bowel diseases, it's like Crohn's, it's a patchy disease, it's a small intestine, and you have ulcerative colitis, which is a large intestine, continuous disease, And then if you see this clinician point of view, I mean, when clinical person or, when Dr. Vineet actually sees the patient, he like to see this, like lesions in the intestine and lesions in the larger intestine. But when I see these samples, when I get these biopsies from Dr. Vineet, what I like to do, I really tear this biopsy apart into small pieces, and I go into microscopic level and like to see what kind of inflammatory cells are sitting here and what is the ratio of inflammatory T cells we call disease promoting cells or anti-inflammatory T cells, disease preventing cells, and if the ratio of these cells are changed. So what we would like to bring, the ratio we would like to change more into anti-inflammatory T cells. So this is a very, very important thing. We have seen the genetics, the environment, and the immune response. They crosstalk to each other. And this is very, very nicely actually established in case of inflammatory bowel disease. But I want to introduce another aspect of it, which is kind of environmental factor, but I go more into it, micronutrients. So now the micronutrients, microbiota, and immune system is constantly crosstalk to each other. And there's an interplay between these three factors, and that lead to the outcome of the inflammation in intestine of IBD. So now, when we talk about the micronutrients, you can see there are two kind of diets. There's a diet like Paleolithic diet, like it's a diet in the Stone Age time, when we were kind of emerging the evolution. You see the level of potassium in these diet is much, much higher. You see all these diets we used to take in the past, right? But as we evolved and we become modernized, the diet is actually also changed. And because of the change in the diet, you see a much lesser level of potassium in the modern diet, right? And this is something which my talk is going to be focused about, how potassium is such important micronutrients which actually modulate immune system in inflammatory bowel disease. So normally, if you see the main function of potassium, you can see maintains fluid and electrolytes, right? Required for proper nerve conductions, and then influence blood pressure and all other things. And these are the fruits or the vegetables which is rich in the potassium, and this is the daily requirement for the potassium, right? But the role of potassium in terms of immune system is not really identified yet, particularly in the setting of inflammation. However, there are reports that potassium is important, shapes anti-tumor immunity, increasing T cell stemness. This is published in Science 2019. And then potassium is a key signal host microbe dysbiosis. This is also important, is published in 2017. And there's so many studies actually talking about the potassium is important for regulating the immune system, right? So this is something which is really critical. And we ourselves actually published a couple of papers, the one with the group of MGH, Harvard Medical School, together with our lab, we identify how potassium is actually inversely correlated with the inflammatory bowel disease. And this is what they have done. This is a human study. They have taken around 200,000 individuals, and they have identified some of the factors. Potassium is one of it. And then we have done some in vitro experiment in human settings, and we combine our study together and publish this paper. And subsequently with Dr. Ahuja, we have done a small study in AIMS where we found that if an IBD patient is secreting more potassium in the urine, they are highly likely to have lesser inflammation. So keeping these things in mind, I decided to go into more mechanistic aspect of this study. So therefore, we went into the animal model because there's a limitation of what you can do in human settings. So the disease model, what I actually gonna use in the whole study is a DSS-induced colitis. So what you have to do, you put the DSS in the water, 2% DSS, and these mice keep taking the DSS in the drinking water, and as a result, there is a rupture in the intestine permeability, and the gut microbiota actually started interacting with the immune system, and as a result, you have a chronic inflammation. So this is a kind of time course. It's like 6 to 7 days animal model. And then you see at the microscopic level, it's a normal intestine. There's a gut barrier function is kind of nicely tight. So there is a kind of distinction between the bacterial species living here and the immune system is living separate, right? But when you have the DSS, there is a kind of gut permeability barrier function is breached, and all these microbes start, talking to immune cells and activating these immune cells. And as a result, these immune cells started secreting the cytokines, and that lead to the tissue injury, and we call it inflammation. So now, using this model, what actually we did, we used these various techniques: clinical sign of the science score, histopathological score, deep immunophenotyping, micronutrient measurements, metabolomics, metagenomics, and then microbiota depletion using these animals. So these samples from these diseased animals, we have taken the serum, the feces, and various other samples, and did all these techniques to identify, to address the question. So this is the first part of my talk, what we have actually done, and this is the published data. If you see this in the patient, the first thing you notice that the potassium, if you have the control healthy individuals, there the potassium level in the feces, fecal samples is much higher, and when they have kind of severe colitis, there's a kind of decrease in the potassium in the feces. So this is a similar experiment we wanted to do, and we did an experiment. We generated this colitis mice, and then what we have done, the wild type mice and the mice which fed with the drinking water, the DSS, and we measure the fecal potassium and fecal sodium, because sodium and potassium, they're kind of two micronutrients, they work in tandem. So what we see in the DSS, there's a much less potassium in the fecal material, and the sodium level actually increases. So from here we got a hint that some way this is increased. So Rajdeep Dalal was the PhD student, and he joined the lab, and he wanted to do some experiment. And every time he comes to my room and say, sir, can I do this experiment? I say, we'll think, right? And one day he came with this idea that I want to take the coconut water and want to feed these mice with the coconut water and see what happens to the colitis. So I was kind of fed up. I said, it's nothing gonna work out. I say, you do what you like to do. So at least for 20 days, he will be not gonna come to my cabin, right? So then actually he took this fresh coconut water, and then he started gavaging these mice. First of all, he actually measured what are the micronutrients level in the coconut water, and obviously, and he also found that you have in the coconut water, and started feeding these mice water. And what we, if you actually feed the mice coconut water as compared to the mice which is having disease with the DSS, there is a much lesser sign of disease at the weight loss. You can see that the weight loss is actually reversed by the coconut water. And if you see the intestine, you see this DSS, there's an intestine actually about fibrosis is there. So there's the size of the colon is actually lesser, but when you actually fed this mice with the coconut water, the size of intestine goes back. So this looks kind of very interesting, but then obviously when you start an experiment, it's not this ending, it's just beginning, right? So we asked the question, can we remove the potassium from the coconut water and do the experiment again? If it is through the coconut water, if it's through the potassium, then coconut water depleted with the potassium should not work, right? So he did an experiment again. He depleted the potassium from the coconut water, and you can see that the coconut water with potassium is nice compared to DSS. The intestine is going back, and when you actually remove the potassium from this mice, the coconut water stop working. It's not really making the disease better, right? So this actually gave a clear indication that something is potassium is doing to suppress the inflammation. Now, based on this coconut water potassium feeding, we calculated the dose of potassium, how much potassium is actually taking by each individual mouse. And then we calculated the dose, and then we replaced the coconut water and started giving potassium, dietary potassium to the mice. And you can clearly see that dietary potassium is also suppressing the inflammation, which you can see by weight loss. And you can also see by this imaging, you see the DSS, you have the intestinal inflammation in the gut. And then if you put the potassium, dietary potassium is suppressing this inflammation. And this is again, you can clearly see the supplementation of potassium diet, you increase the colon length again, and the inflammation is scored. Inflammation, ulceration, edema in the histopathological score is actually getting better. You can see DSS, there's a kind of disruption in this, and you can see when you have the potassium, you have nice colon or intestine. And one of the things what we found that when you put the dietary potassium, you see higher number of anti-inflammatory T cells, which is producing interleukin-10. So interleukin-10 is one of the disease preventing cells in case of inflammatory bowel disease, which is increased when you put the mice on the potassium-rich diet. And then IL-17 and IFN-gamma, both are pro-inflammatory, are also suppressed. So somewhere potassium is actually doing this balance in the intestine between the effector cells causing disease and the anti-inflammatory cells preventing the disease. So next, we actually, based on this, there is a science paper actually published in 2011, where they actually identify the microbiome dysbiosis or microbiota changes in the individuals who are having high potassium diet. And one of the certain things you can see, the Firmicutes levels are increased, which is a good bacteria for IBD. And then based on this study, we also did the sequencing, 16S-RN sequencing in the feces of the mice, which is either on the high potassium diet or no potassium diet. And we found is a Firmicutes level is increased with the high potassium diet. And some of these, lactobacillus, enterobacillus, pneumococci, these are the bacteria which is kind of increased. And somewhere these bacteria is linked to the bile acid metabolism. We know the bile acids are produced by the hepatocyte as a cholic acid, and this cholic acid is converted into secondary biles using these gut microbiota. So from here we got an idea that maybe the bile acid is also involved in this axis. And this is the data from the Dr. Beneath's lab, where the humans actually given the IBD patient giving coconut water The clinical trial data, I'm not showing that he's going to publish, but we found the similar result what we found in case of mice. This bacteria is kind of increased in IBD patient when they actually put on the coconut water. So now I wanted to make a point whether the potassium mediated suppression of inflammation is coming through gut microbiota, right? So therefore we wanted to deplete gut microbiota in the mice. And what we have done, we fed these mice with the cocktail of antibiotics for this many days. And then we generated the germ-free mice. It's not exactly the germ-free, but you have the no bacteria in the feces, what we could actually see. And you can clearly see if you culture the feces of these mice, which is fed on the antibiotic, you see no bacterial growth. And you can see there is aerobic as well as anaerobic, there is no bacterial colony found. So that means these mice, what we generate are kind of germ-free. And this is another sign of these germ-free mice, that the colon actually, this cecum actually size increases, what we can also see as compared to control. So now using these mice, we again put these mice with the high potassium diet, right? So we wanted to make sure whether potassium is able to suppress inflammation in the germ-free mice, right? And what we can clearly see in case of potassium in the wild type mice, there is a kind of suppression. You can see the intercolon length is kind of going back. But when we actually use this antibiotic depleted animal, is not able to suppress. Looks the protection you are seeing in these mice is coming from the gut microbiota. So after this, we went into the second second thing in my talk, whether potassium is actually mediating something with the liver. So what we did the serum metabolomics, and we found that there's a multiple, metabolites are up and down. And one of the metabolites we focus here is the cholic acid, which is increased with the potassium as compared to the DSS mice. So this cholic acid is synthesized, and then cholic acid is one of the thing is this enzyme which convert cholic acid to add the hydroxyl group in the cholic acid. So it's called hydroxylation. And after this hydroxylation, this cholic acid can be converted into the small intestine through the gut microbiota into secondary bile acid, right? So what we identify that one of the transcription factor is increased by the potassium is actually increasing this enzyme, which actually make cholic acid to make hydroxylation. So when we actually then we actually inhibit this FOXO1, and we found that potassium-rich diet is not able to suppress if you inhibit FOXO1. So now we identify one more axis that potassium is making cholic acid, and this cholic acid has to be hydroxylated, which is require FOXO1. And if you block FOXO1, the cholic acid will not be hydroxylated. And then we actually replaced cholic acid. So now we said we replaced coconut water with the potassium diet, and now we wanted to replace potassium with the cholic acid, because that's what our axis is, that potassium is making more cholic acid. So if you actually have more cholic, if you feed the mice with the cholic acid, see making this disease better. If you assess and the cholic acid, it is making disease better, and that is actually happening through the induction of anti-inflammatory, you can see in the bottom panel, and suppressing this effector cells, which making disease worse. So now we know the cholic acid. Now we wanted to go in the molecular level, right? How this cholic acid is actually working. So as I mentioned to you that Th17 cells are the one which is disease promoting cells. So Th17 cells having this transcription factor called ROR gamma T, and this ROR gamma T is having a ligand binding domain. So the ligand for ROR gamma T, which is a oxysterol, which is also a product of cholesterol, goes and bind to this cavity. You can see this ROR gamma cavity, and there it makes a conformation change in the ROR gamma T, which makes this IL-17 promoter transactivate IL-17 gene and make more IL-17. So now what we actually try to do, we actually see the cholic acid is also product of cholesterol, and we decided maybe this cholic acid gonna go and fit into the same cavity. Maybe it's an inhibitor, maybe it's antagonist, right? So we find we took digoxin as a control, and then we found cholic acid goes and fits perfectly in the cavity of this ROR gamma T. And as a result, and we identify what are the molecules actually interacting cholic acid with this, and we identify that if you have cholic acid fits into it, it suppresses IL-17 in experimental colitis. So now we identify a molecule, and this we cannot really patent because it's already known, right? So therefore, we collaborated one of the medicinal chemists in Punjab University, Deepak Salunke, and where he actually synthesized a series of compound. So we got this structure, almost 100 structure of cholic acid, novel cholic acid derivatives, and we use these derivatives and went back to computational modeling and fit each of these derivatives into the computational modeling and identify which of these derivatives actually are the one actually potently suppressing Th17 cells, right? So out of this 100 structure, we found 33 derivatives are showing very nice result. And then we took this 33 derivatives and in vitro tested each derivatives, the one for induction of IL-10 and suppression of IL-17. So out of these 33 derivatives, we identify around 3 derivatives, which is giving good IL-10 induction, which is an anti-inflammatory and suppressing Th17. So these 3 derivatives we took from this screen and we went back into the colitis model and tested whether these 3 derivatives can do the job what cholic acid or potassium was doing, right? So we took these 3 derivatives, you can see name here, these all are patented, right? So any companies interested, please talk to us, right? So these are the 3 derivatives which is now given to this colitis mice, and then we found the suppression of colitis using these 3 derivatives based on the histology score. And these 3 derivatives also increasing more IL-10, you can see here, to suppress intestinal inflammation. So with this result, I would like to conclude that dietary potassium supplementation induces anti-inflammatory response while suppressing pro-inflammatory cytokines or response in colitis through cholic acid. And cholic acid and its synthetic derivatives suppresses ROR gamma T mediated Th17 cell generation while induces IL-10 and reduces intestinal inflammation. And again, some of the member was not in that picture who are really very important, like Salen Rasthana has done the work for computational modeling, and Salen with Charu did this work for us, and other people are also here. And obviously, Dr. Vineet Ahuja was not there on the picture. So obviously, we keep getting so precious samples from him, and we have this back and forth collaboration. Every time I see some new result in the experimental colitis, I always call them and say, can we try something in this in the human setting? So I think this is something real translational things we are doing. Thank you very much.
So, thank you, Dr. Avasti, for the great work and the excellent talk. So can we invite Dr. Sarodat also to the stage? So the stage open for questions. Yeah, so the other talk will be with the next session after lunch. So questions, open for questions.
First question for Saurabh. Saurabh, very nice talk, and the data was excellent. I wanted to ask you that, is it possible that since Pseudomonas was out there, and you would expect mainly aerobic bacteria to be there, and that's precisely the reason that you will get more of actinobacteria and proteobacteria out there, and so you're getting Pseudomonas. It is just that you showed clearly that the Pseudomonas were the non-pathogenic varieties which were there. Could that be the simple reason, although in Western data you did not find the same signal?
Yeah, I mean, we haven't really gone. The work will have to look at these kind of things. What initially we thought that maybe ours was some data, other data from Gujarat and also from South Africa, similar sort of conditions as here. We think that may really no, as was said in the first talk, no normal microbiome as such. And microbiome probably adapts to environment of each individual situation or possibly even each individual mother. So that's what I have to say.
But you won't expect bacteroids, firmicutes to be there, means they're being anaerobes. Here, if you're going to find something, it's going to be more of aerobic.
Yeah.
So would that be the reason enough to say that, okay, you're getting more of the proteobacteria and subsequently the pseudomonas?
Yeah, possibly. Yeah.
And Amit, like a talk full of energy as usual. So just to say what Amit was saying in terms of potassium, at the same time, 3 years back, we started a randomized controlled trial giving coconut water to patients with mild to moderate ulcerative colitis. And right now, simultaneously, we have completed both the studies. He has completed his study. In the animal study, we have completed the clinical trial, sending it out for publication. As such, the results show that coconut water was effective in patients with mild to moderate ulcerative colitis. It had a mild effect, as any dietary source would do, as compared to any targeted molecule or something. And it also changed the microbiome beneficially. So there's a very good example of complementary work and translation work going on simultaneously.
So this is to Dr. Datta. Dr. Datta, after you looked at these individuals, you were able to classify individuals into 3 groups based on Pseudomonas diversity, 1 low, 1 high, and I forget what the 3rd group was. My question to you is that, do these translate in terms of some clinical phenotype? Do you find peculiar or distinct phenotypic features based on the Pseudomonas diversity?
No, actually that was not an aim at when we actually started the study. So we didn't even collect data from that viewpoint. It was kind of post hoc. After we got the results, then we tried to see whether there are different groups. And mind you, these are just 20 patients. So I don't think we should read also too much into that kind of a classification. It was just some interesting trend that we found, and that's why we reported it.
Ask? Yeah, so Saurabh, taking that further, so what would be the next step in terms of a clinical application for that? Particularly, you follow the neonates up for 1 month.
Yes.
And you didn't find any, so just trying to understand, because that's very interesting data, and especially for pediatricians and neonatologists around here, that breast milk being sterile, and then you see bugs, and then you follow up the neonate. So what would it be in terms of a next step for clinical application?
So
just a thought there.
I think there's no question about the fact that these bugs have to be non-virulent. There's no doubt about that. I mean, breastfeeding has been going on for millions of years, and that's what babies need. So
Asymptomatic.
Well,
I mean, is it worth? I'm just saying, what are the next questions?
The interesting thing was that the last study that I showed by Murphy et al., where they looked at paired samples between breast milk and the infant feces. So it's not necessary that whatever is there in the breast milk is actually successfully colonizing the infant gut. So that's the first thing we have to establish, that whatever is there in the breast milk, what fraction of it is actually successfully colonizing the infant gut. And now colonization also has so many complexities to it. As was, I think, mentioned by Vineet in the talk by the overseas speaker, is it the bacteria finding in the stool sample? Is it the bacteria which is, the colon, which is near the mucosal cells, which is in the say small intestine, large intestine? Which are these samples which are actually playing a physiological role? We may find that there is no Pseudomonas in the infant stool, but maybe there is Pseudomonas lining the mucosal cells which are not even coming out in the stools. So there are so many aspects to it that it's difficult to really say for sure what is the physiological role, or whatever it is that the Pseudomonas is playing in the breast milk.
So can I just add one more thing? Coming from a pediatric diarrheal angle, none of the probiotics today in probiotic research have actually made a difference to the Indian gut. in terms of diarrhea and therapeutic purposes. So we do need to keep what you're saying. I agree that that has a very big.
Yeah, I agree with you.
Dr. Datta, can I just add to that as well? That was a fascinating talk. This is Neerja. Dr. Datta, I was reading a recent review where they have suggested that the maternal microbiome in the third trimester has more numbers of proteobacteria as compared to what in the first or the second trimester. So like you said, you spoke about the dendritic cells carrying these microbes from the gut to the mammary glands. So is it possible that, I mean, again, just a thought that the dendritic cells in the third trimester actually carry these proteobacteria or pseudomonas, so to say, to the mammary glands, and that could be the mechanism by which you have so many pseudomonas in the lactating mothers in the breast milk?
No, so these are two different things. One is, which are the cells that are doing this transporter job? When are they doing it? And why is it different in Indian mothers and South African mothers compared to the Western world? So possibly the mechanism of carriage is probably uniform across the human race. I don't really think that there are different kinds of cells which are doing this task in, amongst Caucasians versus Indians versus others. So probably that mechanism is the same. The interesting thing is, why is the profile of the organisms different in Indian mothers? I think that's a separate question altogether. And the other thing is that most likely it is not the dendritic cell which is going all the way. The dendritic cells are probably handing over like a relay race, a baton being carried over to some other cell forms, probably of the monocyte macrophage lineage is my guess, which is finally taking it to the mammary gland. In fact, they are trying to look at that in a study Vince is doing as to which are the cell fractions that are actually carrying these bacteria. Macrophages and monocyte lineage cells outnumber dendritic cells by several orders of magnitude in the mammary gland. And it's possible that actually this kind of a handover is happening somewhere, maybe in the mesenteric lymph nodes or something.
So just I have two small questions. So the first question, so do you find any associations of the late onset sepsis versus breast milk microbiota in the neonate? Late onset sepsis versus maternal, this breast milk microbiota, is there any correlations?
No. So whatever case reports of late onset sepsis are there are all with contamination. So either IV fluid contamination with some Pseudomonas species, or mother actually having an infection, an obvious breast infection, and then the baby getting infected. But there are no reports, and I guess there will never ever be reports, because these would be all non-virulent, non-pathogenic bacteria. What is worrisome though is antibiotic resistance. So you may not have pathogenic organisms causing sepsis, but it's very likely that multi-drug resistant bacteria, which are picking up multi-drug resistance, are being transmitted from the mother's milk into the neonatal gut. And maybe a lot of neonates are now being born with a gut resistome, which they have got from their mother. And what implication this is going to have for this kid as the kid grows up is something that's the subject of, I think, research.
Okay, thank you.
With this, I thank all the session chairs and the speakers for the very highly informative and interactive session. Now I request Professor Sharmila Mande to hand over the memento to Professor Saurav Datta. May I now request Professor Shalimar to hand over the memento to Professor Amita Basti. Now I request Professor T. Ramamurthy to kindly come upon to the stage to felicitate the session chairs. So please. May I request Professor Sharmila Mande to receive the token of appreciation, please? Next. May I request Dr. Shalimar sir next? Professor Shalimar. Sir. And Dr. Jiva Shankar. Thank you, sir. We now break for lunch. We request everyone to join us for the lunch and as well for the stall visits just outside the auditorium. We also request the participants who are presenting the poster be present near their respective posters for evaluation. Let us all assemble back by sharp 2:30 p.m. for the start of the next session. Thank you. May I request all the speakers of the upcoming sessions to kindly share their presentations to the registration desk. Thank you.
I request everyone to please come into the auditorium and please be seated. Welcome back, everyone. We hope you all had tinkering discussions over the lunch. We now begin our scientific session 3 of the conference on the theme Microbiome-based therapeutics, new approaches and challenges. For this, I request Professor Shinjini Bhatnagar, Professor Shibnath Majumdar, and Dr. Nitya Vadva to kindly chair the session. Sir, ma'am, I request you to kindly join us onto the stage, please. Professor Shinjini Bhatnagar is a professor of eminence who heads the maternal and child research at THSTI. Her contributions have provided evidence-based recommendations in child health for global and national policy making. In recent years, Professor Bhatnagar has established large multidisciplinary research programs that bridge classical epidemiology with modern science to find solutions for diseases that have major public health significance. Another research focus of Professor Shinjini Bhatnagar is to develop diagnostics and low-cost health products for childhood diseases. Thank you, ma'am, for joining. Professor Shibnath Majumdar is the Professor and Dean of Faculty of Life Sciences and Biotechnology, South Asian University, New Delhi. Professor Majumdar's team worked towards unraveling the various pathogenic mechanisms of zoonotic pathogens and to develop intervention strategies against them. He has established the stellar role of virulence plasmids in Aeromonas hydrophila pathogenesis and successfully used plasmid-cured isolates as vaccine candidates against its spread. Thank you, sir, for accepting the invitation. Dr. Vadva is a trained pediatrician who has transitioned to become a physician scientist. She has been doing clinical research for more than a decade and leads her team at THSTI. She has been associated with several large multicentric clinical trials, and she has been instrumental in establishing a large longitudinal cohort of pregnant women with the primary aim of building risk stratification algorithms for adverse birth outcomes. Thank you, ma'am, for being here with us. In this session, each talk will be of 20 minutes. After the talk of all the speakers, there will be 10 minutes long question and answer session. We request our audience to kindly reserve their questions until the end of the session. I now request the session chairs to introduce the speakers and call them over for their talks. Ma'am, please.
Good afternoon, everyone. We'll begin the session without further delay. My absolute pleasure to invite Professor Vineet Ahuja, a personal friend, a colleague, and one of the leading physician scientists of the country. He's a professor at the Department of Gastroenterology and Associate Dean at the All India Institute of Medical Sciences, New Delhi. His research focuses on IBD, abdominal tuberculosis, gut microbiome, and gut immunology. This is just some of the stuff. He's published more than 260 international and national publications, 24 book chapters. He's the editor of Tropical Gastroenterology and a member of the International Organization for IBD. Over to you, Vineet.
Thank you so much, Sydney. It's a pleasure to be at THSTI. I'm really grateful to Dr. Garg and Dr. Babatosh for putting up a splendid program and inviting all of us. And we have had wonderful talks since morning. And I'll be basically talking about now, as part of microbial manipulation therapies, we'll be talking about fecal microbial transplantation. Now, first of all, I wanted to show you this slide. This is a globe, like this is a rotating globe. And what you see out here is the ozone gap out here, right on the top of Antarctica, right? So this ozone gap is now, in the sense that it was obviously becoming huge, and now gradually it is decreasing, and hopefully another 30, 40 years, it should close down. What's the importance of this slide out here? Do you know that one of the techniques by which they are probably going, which is going to be helpful in closing the ozone gap, is again going to be microbial manipulation, the microbial manipulation of the waste, the way the climatic degradation is going to change. So that shows that microbiome actually is pervading every field. It is no longer restricted to GI diseases, cardiovascular diseases, neurological diseases, or something like that. It has already crossed the boundaries of human and gone into environment totally, and many of our things are going to be dictated by that. Now, at the same time, I would like to put up a slide out here. There's a nature infographic, which makes you, in the sense, it gives you a feeling of enthrallment. This shows that how fast vaccines have been discovered by mankind. If you look at the topmost column, topmost row, you'll find that typhoid fever, it took like maybe around 120 or 140 years for it to get developed after the time the pathogen was discovered. Thereafter, measles, 10, 20 years, hepatitis B, 20 years, and SARS-CoV-2, just 1 year. Within 1 year, the pathogen was identified and the vaccine was made. So doesn't that really enthrall you? Doesn't it tell you that, okay, we are increasing by leaps and bounds? We are really going to orbits which we never thought was so. Yet at the same time, I show you another infographic. There's another infographic from Nature which came up recently, which looked at disruptive science. That is, they decided that they've graded a paper according to how much it can disrupt the science. That is known as the CD index. And the interesting part out here was that most of the disruptive science papers actually came up from 1950s to 80s. If you look at last 10 years or something, the disruptive science papers have been 20. Where we talk about, okay, we think that over the last 20 years, if you ask anybody, a person outside science, he'll also tell you internet has come up and mobile has come up and the whole science has changed. And probably we have had the maximum in terms of advancements in last 20 years. But there is always something which is way beyond it. And that is what I would like to show about fecal microbial transplantation. Fecal microbial transplantation, yes, it's a science meeting. So you would expect me to show, even though I'm a clinician, maybe some kind of mice experiments, mice data to show that what happens when mice are given fecal transplants. Do they benefit or not? But let me show you animal data out here, which is real world experience. This is from a letter which was published in Gut. Gut is one of the top ranked gastroenterology journals, which talked about Cynthia Lynch. Cynthia Lynch was a game rancher and she was in the Northern Territory of Australia. And what she did was, there she found that the kangaroos, they had the babies, which are known as zoes. So these zoes, they often got orphaned because of the killings which would take place, and some of them would develop diarrhea. When they develop diarrhea, as a result, what this lady, the game rancher, would do, she would pick up the poop from the healthy kangaroo babies, and subsequently she would make a poo shake out of it. This is a recent video, but that's what they did, and this became something of a regular thing which is being practiced in the Australian zoo. So they would pick up the poo of the healthy babies and subsequently make the shakes out of it and then feed it to the kangaroo babies. And when they fed it to the kangaroo babies, it was seen that thereafter, these babies, the zoes, they had tremendous improvement. And they have thousands such documented case experiences which are there. This, I say, is the real animal data, the real world experience animal data. So we have 4 generations of gut microbiome manipulation, if you look at it, and that is irrespective of any disease. Antibiotics, yes, they manipulate the gut microbiome. Diet, yes, it does. Probiotics. Then we have the fecal microbial transplantation, capsulized FMT, and subsequently we are going to have microbial ecosystem therapeutics. What is fecal microbial transplantation? It is essentially a pan-restorative approach where you infuse fecal suspension from a healthy individual into the GI tract of an individual with a disease. And as a result, what happens, you hope that you'll refurbish the bacteria, and as a result, things should improve. Is it restricted only to NASH, GI diseases? No. This is a snapshot of studies on FMT, which have been done in various diseases. You talk about GI infections, MDROs, inflammatory bowel disease, irritable bowel syndrome, autism, Parkinsonism disease, which is a neurological disorder, schizophrenia, which is a psychiatric disorder, Alzheimer's disease, multiple sclerosis, which is a neurological disorder, and even things like obesity and weight loss. So that is the expanse of FMT today. And essentially how it is done. You pick up feces, a healthy donor donates the feces, it is mixed, blended, filtrate, and subsequently it can be infused into the GI tract either through an endoscopy, or you are subsequently getting oral capsules which are being formulated. They are already available in US, and I'm sure in India they would soon be available. The next talk is by Kunal, who is already working on that. And so this is the delivery. Like you can deliver it through the nose, or you can deliver it through a colonoscope. You have liquids and pills are being formed. So a very simple procedure. Unlike bone marrow transplantation, renal transplantation, hepatic transplantation, where there seems to be, there are lots and lots more of intricacies. Here there is only one intricacy. That intricacy is that you should select the best of the donor, the first amongst equals. All people who apparently look healthy may not be healthy. Why? Because here it is very important when we look at gut microbiome and its association with autism, its association with obesity, association with schizophrenia. For all you know, you may be transmitting a mood disorder. A person may develop a mood disorder years later from a donor who looks healthy. So the key point out here is that the donor should be very well screened, should not be, broadly speaking, not be having an infection, not be on antibiotics, not be on proton pump inhibitors, should have been thoroughly investigated to see that there is no infection out there. In fact, this checking is so stringent that if you screen 10 donors, maybe you will find out one donor who is fit for donating feces and for the transplant. At our center at AIIMS, we have been doing fecal FMTs from 2014. We would have done around 300 procedures till now or something. And generally, since we have been doing FMT for patients with GI diseases, essentially, we have been doing it by colonoscopic route. Shalimar has started doing FMT for non-alcoholic steatohepatitis, and I think he uses the duodenal route. So now coming to the disease of which I'm going to talk about, inflammatory bowel disease. You had a talk in the morning by Professor Takeda, then by Dr. Avasthi. So you would know about the disease, but I still thought that I would put up one slide out here what exactly this disease means. It is different from what people think of as irritable bowel syndrome. Inflammatory bowel diseases means there's a definite inflammation in the small intestine or the colon. It can be ulcerative colitis or Crohn's disease. It has a definite morbidity and some amount of mortality also. People are afflicted in this disease right in the youth, very young age, and this disease stays with them all through their life. Unfortunately, it is not curable as yet. It is only controllable. You can only control the inflammation. And how do you control the inflammation? You have to give a lot of agents which decrease the immune responses, which dampens the immune responses, and that's how it works. And so a lot of research has been done on, and we have a lot of targeted molecules which are available. We have a lot of monoclonal antibodies which are available, which may target either the TNF antagonist, which is what I would say the cornerstone, lodestone of therapy for IBD, infliximab and all. You have lymphocyte trafficking inhibitors, and you have IL-12, 23 inhibitors. So they are all effective, but there's a therapeutic ceiling. They're effective in only 60 to 70% of cases. Number two, they are again infinite therapy. They do not cure the disease. They only control the disease till the time you're giving them. Number three, the cost. The cost of this therapy in India would be roughly around 2.5 lakh rupees to 6 lakh rupees a year for the patient. And again, as I told you, it's not finite therapy. You do have patients with IBD who may not require this kind of therapy. Roughly, I would say 60-70% of such patients are there. But there also, they require treatment year after year, month after month. And obviously, this therapy also has its share of a lot of adverse effects. So is there any way else we can treat it? Is there a more physiological way by which we can treat these patients where it is not so costly, not so expensive, at the same time not associated with adverse events? So coming to the pathogenesis of ulcerative colitis or IBD, 4 circles: genetic susceptibility. Second is immune response, aberrant immune responses and all. Third is intestinal microbiota, and obviously the environmental triggers which can affect the intestinal microbiota. So the question is that, can you change the microbiome? Can you somehow modify the intestinal responses, the environmental triggers which may be affected by diet, and thereby modulate the disease? What about fecal microbial transplantation, which can change the microbiome? Is there data available? Yes, there is data available. There are 4 trials which are available, which have shown the benefit of FMT in patients with ulcerative colitis. But then these studies were done only for 8 weeks. And they saw the response only for 8 weeks, showed that yes, there is a good response, but subsequently, obviously, then what to do? Why this is important? Because ulcerative colitis or IBD or Crohn's disease is something which does not just settle down. It is something which has a fluctuating course. There may be the increase in disease activity, which is known as relapse. The patient may settle down for a few weeks or months, that is known as remission. And suddenly after months or years, the patient would also have a relapse. In other words, it is a fluctuating course. And secondly, those patients require therapy all along. These studies, which are there for the FMT studies, they're just short 8-week studies. And obviously you need something more than that. So the research hypothesis for us was that in patients, okay, fecal microbial transplantation probably works by changing the gut microbiome. Can you also change the environmental triggers in sense of changing the diet out there? So if you give a kind of diet which probably reduces inflammation, if you combine both of them, is it going to be effective? And then you induce the response with that, decrease the inflammation. And as I told you, these are diseases which have relapses and remissions. So subsequently, you want to maintain the response, let the patient only be on anti-inflammatory diet and see what happens. So with this hypothesis, we conducted a randomized controlled trial. This trial was published in Gut and it was considered as editor's choice, and they put Qutub Minar on the cover page, which was very interesting for the Delhi people, at least if not for India. And this is the whole team which did the study. There out here is Saurabh and me who led the study. We had the diet team, we had the FMT team, we had the microbiome team. There were at least 15 people who played their role. Each one of them played their role in conducting the study. The objective was to evaluate the effect of a combination of multi-donor intensive FMT and anti-inflammatory diet in inducing and subsequently, if you maintain only with anti-inflammatory diet, whether it helps in patients with mild to moderate ulcerative colitis. What we did is that we picked up donors, which I told you need to be intensively screened. Where were the donors picked up? They were rural donors. And subsequently, they were screened and FMT was given by colonoscopy. You would ask me why we picked up rural donors. We picked up rural donors because in a study earlier done with Babatosh, where we had picked up fecal samples from patients who were living, healthy people living in an urban area, healthy people living in a rural area, and subsequently we also picked up samples from people living in Ladakh. Every year we go for a health camp in Ladakh and we have never seen a case of inflammatory bowel disease out there. We thought that Ladakh communities seem to be insular from this kind of disease, probably because of the gut microbiome, probably because of environmental factors. So we picked up the fecal samples from out there, and Baba Toshtash and subsequently Tarani also joined the effort. And the results are very interesting, that these are 3 healthy populations which we picked up. The rural healthy population, they had a much better gut microbiome as compared to the healthy urban population. And Ladakh lay population also was quite healthy, almost as good as the rural people. Suggesting that if rural, what you are looking for, you're looking for the best donors. So obviously, if rural people have possibly a healthier microbiome, why not use them as donors? We don't know whether they'll be good for FMT, but definitely we knew that they had a healthier microbiome. Second is diet. What kind of diet are you? What do you mean by anti-inflammatory diet? For this, you have a lot of experimental data, the mice data and all, which shows, okay, maybe gluten promotes inflammation. Dairy products, they promote inflammation. Whereas AHR-rich ligand food like cauliflower, cabbage, broccoli and all, they would decrease the inflammation. So based on this, if you can somehow form a diet whereby you're avoiding gluten-based greens, but recommending rice, maize and all, avoiding dairy, but giving fresh curd and buttermilk, etc., avoiding red meat, but you can give fresh chicken or egg or something like that, and avoiding the tough texture seed vegetables and giving, as I told you, vegetables which have a good AHR ligand-rich diet. And we randomize patients into 2 arms. In 1 arm, we give 7 sessions of fecal microbial transplantation every week after week, along with anti-inflammatory diet. The other arm, we just gave them the standard medical therapy as they would have for ulcerative colitis. Patients were followed up for initially 8 weeks and thereafter for till the end of the year. And some of the outcomes which I'll show you, the clinical response was definitely better in patients who were given FMT plus diet. Deep remission, that is clinical plus endoscopic remission, was better in patients who were given FMT plus anti-inflammatory diet. And this group also maintained its response right till the end of year, till 48 weeks. So this was obviously very interesting. The other secondary outcomes like endoscopic response remission were also, there were some good signals we were getting. So this was obviously very interesting. They showed that if you had a physiological approach, you're just changing the gut microbiome, the adverse events were almost very few. There was no serious adverse event which was reported in the trial. So if you're just changing the gut microbiome, it does not lead to any major adverse events. Along with that, if you change the diet, you are possibly able to modulate the course of the disease, which requires a constant immunosuppression. And again, on the basis of one trial, one cannot say that, okay, it needs to be immediately put into clinical practice. These are kind of studies which need to be validated again, but at the same time, this is very interesting data which tells you that there's a strategy which potentially should work. We also wanted to see that, okay, now FMT, the problem with FMT, it's an orphan. It's like a Charles Dickens novel, like David Copperfield. You have the targeted therapies, monoclonal antibodies. There's a lot of industry, pharmaceutical companies running after it. They would be promoting it. They would be having seminars, symposium. FMT does not have any godfather, obviously, in the sense. So nobody is much willing to conduct a head-to-head trial whereby you may compare FMT, let us say, with a monoclonal antibody or something like that. So we did a network meta-analysis whereby we looked at the effect of FMT in patients with mild to moderate ulcerative colitis and compared it with other well-established monoclonal antibodies and oral small molecules. And it was found that it was efficacious and safe as compared to other targeted pharmacotherapies in patients with active ulcerative colitis. So the next thing was, what is happening backstage behind the scenes, like how is it being effective? Should we look at only the microbiome or should we look at the metabolite? If you're going to look at the microbiome, which site microbiome is more important? So this work was done by PhD students, Aditya Manaswani, who came up with some very good work where they showed that FMT refurbishes the mucosal microbiota and metabolic, the fecal as well as the mucosal microbiota and metabolic profiles in patients with ulcerative colitis. Essentially, the second part of the study, where the samples were taken for patients who underwent FMT, and this is before they underwent FMT, and 8 weeks later, once FMT has been completed, and it was compared with healthy controls. And we had samples which are taken fecal samples. In the same patients, you also took up the, not only the stool samples, you also took up the biopsy samples. I think I'll use this pointer, this would be better. So biopsy samples as well as stool samples, you had the clinical data and the dietary data, and thereafter looked at not only the microbiome, but also looked at the metabolomics, and you compared it with healthy controls who were undergoing sigmoidoscopy. So you had access to the rectal biopsy and looked at the mucosal microbiome out there as well as the fecal microbiome. And it was seen that FMT refurbishes the diversity of both luminal and mucosal microbiota. In both the sites, it was seen that diversity improves after fecal microbial transplantation is given. And this shows that we looked at core of both the microbiota, the mucosal microbiota as well as the fecal microbiota. These are patients, these are controls. These are UC patients. This is post-FMT. Look how sparse the microbiome is, both in the fecal microbiome as well as the mucosal microbiome before they were given FMT, when they had the active disease. The moment you give them FMT, the microbiome gets refurbished out. It is like a forest fire which is raging. You give something which put water on it. So FMT is like that. It just quenches the forest fire out there. And subsequently, the grass can grow back. And this is how you can see that the bacteria are coming back, both at the mucosal level as well as the fecal microbiome level. What is happening to metabolomes? Metabolomic profile at mucosal level as well as fecal level showed that obviously there was movement. These are controls. These are patients who are pre-FMT, and you find that post-FMT, the metabolite levels or the metabolomics show a shift towards the control, which was more marked in mucosa as compared to feces. And again, the Venn diagram. So these are colonic tissue metabolites. This is post FMT, this is control, and this is pre-FMT, and you find that how there's a change and you get reformation of the typical signatures. You show Shalimar showing the signatures today in terms of liver tissue and subsequently also. They're the similar signatures. It would be secondary bile acids would be increased. You heard Amit talking about cholic acid, and that's a debate we have. It should be lithocholic acid or deoxycholic acid rather than cholic acid. But Amit has explained to me that the cholic acid subsequently is getting converted into deoxycholic acid, and that is where probably the effect is coming. So secondary bile acids increase, the tryptophan metabolites, they increase, the short chain fatty acids, they increase. But this change may occur for some in mucosa, for some of the metabolites in only feces, and that is very important. So this data shows that FMT and AIH efficiently restores beneficial bacterial populations and related metabolic pathways, not only in the fecal stream, but also in the relatively reserved mucosal microbial niche. So subsequently, Atha and Manaswani are working on organoids in terms of checking out the metabolites on organoids and seeing what it can work. And suppose something works, then I'll give a shout out the way Amit gave a shout out in his talk that is there any company out there who wants to pick up the cholic acid? So maybe we can also give a shout out like that. Now, the last piece I find it most interesting, right? This is some very interesting data. Gut microbiota, we know, we talk about fecal microbiome because it is easy to get that sample. Obviously, it has caused the least discomfort to the patient, easy to collect, so easy to do it. Or oral microbiome, you want to do the salivary microbiome. Mucosal is a little tough because there has to be an indication to go ahead and take a biopsy out there, and you do that. But microbiota is ubiquitous. You saw microbiome in breast milk. You have a poster outside which shows microbiome in pancreatic tissue. It is ubiquitous. So it's not possible that the microbiome would not be existing in the epithelial cell or below that. So the third part was to look at crypt-specific microbiota, bacteria, crypt-specific bacteria, which are near the stem cells. Stem cell is obviously the master regulator. If there is a prime minister out there who's dictating everything, that is the stem cell. We all know that. So obviously the cautery which is around the stem cell is going to be the most important cautery. The cautery around the PM is the most important. So which are the bacteria out there? Can we elucidate those bacteria? Can we find out what they are going to do? Or are they there or not? If they are there, are they doing something or not? That's the question we ask. And again, Aditya and Manaswani looked into it. These are obviously supposed to be very resilient bacteria. They are probably aerobic bacteria. Why? Because their hypoxia is not there. They are near to the blood vessels. So they would be aerobic bacteria. They would be resilient bacteria. And they did turn out to be very resilient bacteria because to get to these bacteria, you have to do laser capture microdissection of the crypts from the mucosal biopsies. And our resilient students, Aditya and Manaswani, they did take 2-3 months in terms of getting the LCM machine. It would work in one place, not work in one place. They moved from AIMS for a few days. Then they came to THSTI, the common facility out there, worked out here. Then they went to NIPGR, worked out there. Finally, they went to NBRC and were able to complete the work. They completed the work and they showed very beautifully that there are bacteria which are there in the crypts. See that? There are bacteria which are there in the crypts. Professor Takeda said that yes, they may be there, but why? Because there's the second such study which is there. In fact, for ulcerative colitis, the first such study which shows that there are bacteria in the guts, but they are very sparse. Second, these bacteria, they are different in the sense that you have more in the controls. What you see, you have more of actin bacteria and protein because hypoxic conditions are not there. On the mucus and above, there would be hypoxia. Here, there is more of oxygen near the epithelial cells. FMT, pre-FMT ulcerative colitis, the bacterial population changes and subsequently it refurbishes back. The actinobacteria, proteobacteria come up. And not only those bacteria are there, these bacteria correlate with the disease activity. This is an index of endoscopic index of severity. This is a clinical index of severity. This is fecal calprotectin, which is an index of biochemical inflammation. And you find that with all these three markers, these bacteria had a very good negative. That is, once FMT was given, these are the bacteria which came up and they had a negative correlation with the inflammatory markers. More the inflammation, less the bacteria, more the inflammation. So what I've shown you today is, one is that microbiota, essentially, whatever ubiquitous are there, FMT or microbiome manipulation therapies are also becoming ubiquitous. One. Number two, if you talk about inflammatory bowel disease, FMT and anti-inflammatory diet is beneficial in active ulcerative colitis. That is what our randomized controlled trial data showed. But as I told you, such kind of things need a little bit more validation, and then they can immediately go into clinical practice. Changes in microbiome following intervention may be at a fecal level as well as a mucosal level. And those changes are different. It is not the same change at fecal level as it is at mucosal level. There are not only changes in microbiome, there are changes in metabolites also. And we all know today metabolites are much, much more important than microbiome. We are not looking at microbiome pills 2 years down the line. We are looking at metabolite pills 2 years down the line. Third is some interesting new data. There's change in crypt-specific microbiome also following the intervention. And unanswered questions for the people in the audience out there, the young people out there, if you want to really answer some questions. The unanswered questions are okay, whether you would like to give anaerobic bacteria, FMT preparation bacteria, disease duration, early, would you like to intervene or not? Disease has to be mild, or just 30 seconds more. But the most important question out here, if you want to answer, is that donor has a very significant impact. There's something called a super donor. And if you can define that with the help of your microbiome, metabolite, microbiome, virome profiles, that would be the most interesting question to answer. And with this, I thank all the team members out here. Each one of them is sterling, bright, and I'm blessed to have such people with whom I work. And not only that, we have terrific collaborators who are more than collaborators, they're friends like. And you can say that this is the collaboration only from the NCR, and all of them are good friends. THS Dr. Bhabatosh Das, Pallavi, I want to thank for the metabolite work which has been done. The second part which I showed you, the metabolite profile from in the fecal and mucosal, it could not have been possible without Pallavi and our students, and I really thank her. Amit Avasthi is not only a very good friend, but we collaborate and we on immunological work more. RCB Shrikant, I want to thank you for the last work, the crypt specific work. The crypt specific, it was possible because Shrikant really helped us when it came to LCM and everything. Avinash out here and ICGB, Dheeraj and Dinesh. Thank you so much for your attention.
Thank you so much, Vineet. Thank you. That was really excellent. We'll request you to come here.
So I have the pleasure of inviting Dr. Kunal Rai for the next talk. Dr. Kunal Rai is an associate professor at MD Anderson Cancer Center. He is also the scientific director of the MD Anderson Epigenomics Initiative, which is geared towards rapid translation of novel advances in the epigenetics field. Over the years, Kunal has made seminal contributions in understanding of epigenetic processes during normal organ development and abnormal cell growth during tumorigenesis, and published papers in top journals, including Cell, Cancer Discovery, etc. Presently, his lab focuses on understanding the contribution of epigenome to cancer progression and identifying new venues for therapy and diagnostic tools. Welcome, Dr. Kunal.
Well, thank you for the introduction. My name is Kunal Rai, of course. My lab, as the introduction said, focuses on epigenetics. I don't consider myself an expert in microbiome here, so we'll see how much justice do I do to the work. And this is the first time I'm presenting this work, so please pardon any mistakes that might be. Right. So I'll start with acknowledgements because obviously this is critical for this piece of work. This work is done in close collaboration with Dr. Helen Pivnikavam's lab, whose lab actually established the role of fasting in chemo prevention and radiation damage prevention. I'll talk about that. And a world-leading expert, Dr. Robert Jenk in microbiome. The work was done by a postdoc who's a joint postdoc between my lab and Helen's lab, Praveen Barodia. So this is not working. So Praveen is actually a PhD from ILS here. well led by computational biologist Amri Arsalan, who's the principal research scientist in METI, and a previous instructor in my lab, Christopher Terranova, who's an epigeneticist. So it's a great team effort that we pulled together. I also want to thank the funding. So my lab's funded through various resources, NIH to CPRIT to Department of Defense, etc. And finally, the patients and the mice that we actually, through which we provide useful information. So all right, so my talk, this is focused on understanding how can we prevent the toxicity that emerges from the chemotherapy or radiation therapy, which, as you know, is a major mode of therapy, say, across variety of cancer types. And these are just listed some of the side effects that you have, and many of these side effects are due to the toxicity in the GI tract, right? So that's what we wanted to understand and see if there is a way we can sort of limit the toxicity or improve upon it so that we can improve patient care. And this work started with work published from across, I think, several groups, but this is just one of the papers that suggests that when you starve starve the patient, so starvation-dependent stress resistance protects normal but not the cancer cells against the high-dose chemotherapy. And that really was the basis, was the work which was done then in Helen's lab, where they showed that the fasting protects from the chemotherapy by preserving a small intestinal crypt architecture. So here is a schematic scheme. This is done in mice. So you take the mice. These are regular black 6 mice, and you from day 0, you feed or fast them. You give high dose etoposide. That is one of the chemo agents. They've done this experiments with multiple different kinds, and you follow up. And what you see is a very drastic survival curve, as you would see. When you have fasting, you actually protect all the mice compared to all the fed mice that die, right? So that's very strong when you think about this. And what you see is in the intestine is, if you look at this is really bad looking, intestine, which is what the toxicity arises from. But in fasted, it looks pretty normal, right? So that's really what we are trying to recapitulate with the whatever we do. And this is also the case with radiation-induced damage. And it's again, same thing. Here we make use of total abdominal radiation therapy, where you only give radiation to the half of the mice, the bottom half of the mice, the abdominal portion. And again, what you can see is that fasting completely protects these mice from radiation damage. This is a lethal radiation dose. And again, same question, same point. You can see in the fed one, you have really bad looking intestines, high toxicity, and the fasted mice look very nice. So our question that we started to address 3 or 4 years ago now was to how does fasting improve these intestinal stem cell architecture. So we obviously, being epigeneticists, we were interested in understanding how does it modulate the epigenome, how does fasting modulate epigenome, and how might that in turn change the transcriptomic structure for this. So just as a background, I mean, as you guys may know, epigenetics, one of the key components of epigenetics is histone modification, something that we have expertise in. There are more than 100 such histone modifications. And these histone modifications essentially are modifications on these unstructured tails of nucleosomes. And these nucleosome tails are very important because they essentially serve as binding places for various regulatory proteins. And these are of different kinds: DNA methylation, lysine methylation, acetylation, etc., etc. And as you can see, again, there are various kinds of modifications. They happen at different positions in the histone tails, and they have different regulatory potential, right? And this has been well established over the last 20 years. One of the first being from David Alice, who just died actually a month ago or so in the Rockefeller. And why these are important is because they can stay at different regulatory elements in the genome. and they can have different kinds of effect on the gene expression. Okay, essentially, so K27 acetylation, I'll talk a little bit about this active enhancer mark that is present on enhancers and has a positive impact on the gene expression. That's the active enhancers. There are others such as K9, K27 trimethylation that are repressive marks. So for those who don't know how to read this, it's a histone H3 lysine 9 trimethylation, histone H3 lysine 27 acetylation, etc. So it's a short form for that. Now, why that's and then the second layer of this is that you have in any given cell type, you have a set of transcription factors. They work together to make the cell identity or the cell unit, right? So this is called master transcription factors. They work together to set up the whole cell identity. And the prime example of that would be OCT4, OCT2, and OCT in the IPS setting, for example, just to give you a flavor of this, right? And this happens in every single cell type in our body. And this can happen in a normal condition or an abnormal condition. The importance of this will become later. Just wanted to give you a background. So we published a paper a couple of years ago now, where we showed that the reprogramming of a specific modifications, which is called histone H3 lysine 9 BHB, so beta hydroxybutyrate, at regulatory elements is a key feature of fasting in small intestine. And for those who obviously look at metabolites from microbiome, it will become clear why this is important, because 4-BHB is one of the things, or beta hydroxybutyrate is one of the things that some of the microbes produce, for example. So what we showed in this paper was, and I won't go through detail, but just give you a summary, that fasting induced this particular metabolite, beta hydroxybutyrate, through production of through an enzyme which is called HMGCS2. So when fasting happens, this enzyme goes skyrockets, it's like very high fold. So top gene you will ever see. And this actually then marks histone H3K9 BHB, that marks the regulatory program for metabolic gene programs, and that includes many. So this is what we showed in this paper. And this is produced specifically in the small intestine. Liver also produces it, but the small intestinal production of this enzyme is critical for fasting mediated protection. Now, that was important, right? So beta hydroxybutyrate is one of the metabolites. So we also we took the fed and fasted mice, and we did LCMS analysis from the metabolites and just looked for what might be different. So beta hydroxybutyrate is one of them, but then we found a bunch of very novel ones that I'm not listing, just mostly because we're filing the IP right now completely. But anyways, the bottom line is we can see now various metabolites that actually impact histones in a different way. Okay, so when we started to think about these, and some of these will eventually go on to make propion, propionate, and acetate, etc. Which again, because of as I'm sure you heard today a lot, that these gut microbes, along with the diet plus the primary bile acid, etc. interactions will make some of these metabolites. So that's what got us interested, thinking about, hey, there may be a role for gut microbiome here, right? And and there is obviously enough literature in the field that suggests that diet induces gut microbiome induces, and gut microbiome is critical for GI tract homeostasis, as you've been hearing today. So that was my foray into understanding how gut microbiome field might be, impacting. So that's what we wanted to then study. Does gut microbiome induce help with the fasting-induced radioprotection? So first thing we did was we obviously fasted the mice, took the fecal material and profiled that. And what you would see is this green. Well, we didn't see any drastic change in the load as such, the bacterial load. But obviously, if you looked at the green, the Akkermansia muciniphila kept coming back and back, right? So we repeated the experiment like 5 times because, as many of you know, I mean, there's quite a bit of heterogeneity in these kind of mice or experiments when you do these. So we had to really play with it a lot, like get the mice from the same rooms, and many different conditions. So. But when we did all of that, then we started seeing this pattern emerge all the time. There are other changes as well, as you can see, Clostridium, and there's some loss of others, etc. So we asked, okay, great, AKK is changed. This is also being shown, at least in part, by people. But we wanted to then study, because AKK at the same time in the lab next door, Jennifer Vargo's lab, who you might have heard multiple papers from as well. She studies ICB resistance and looking at gut microbiome. So her lab, as well as Tom Gajewski's lab in University of Chicago, they were all discovered that AKK is probably impacts the immune checkpoint therapy resistance in cancer. So that also was of interest. So basically, yeah, here then we asked, does AKK protect, right? So we did a broad spectrum. We then took the mice, gave them the broad spectrum antibiotic, did the AKK gavage, and then followed up with the radiation, like the lethal radiation, and see what happens, right? And you can see that we built up the AKK again by just 16S profiling. And the results was interesting, of course. So as you would see that, the fed mice died, the fasted mice survived. But when we did this, broad spectrum antibiotic plus AKK plus fasted mice, now we got a lot more survival. Although results were heterogeneous and were, not like as clear cut, but bottom line was AKK did, along with fasting, protected the mice. And again, when you look at the gut architecture, now it looks very nice. And then number of crypts increased significantly, as well as the length of the crypts, or height of the crypts also improved. This effect was through small intestinal stem cells, because you can look at intestinal cells. So OLFM4 is a marker for these intestinal stem cells, and you can see that seems to increase when you have AKK. And this is fed mice, this is fasted mice. So only in the AKK condition, you can start to see a lot more staining of OLFM4. And we know that this occurs because of our previous experiments with the metabolites, etc. When we look at the staining, they actually stain quite heavily on the intestinal stem cells. So that was one. Then we thought we'd do a specific experiment. So the knowledge from Rob and others suggests that tetracycline treatment specifically depletes AKK. And this is shown here. So we did a tetracycline experiment. You have untreated mice or treated mice. You can see you can completely eliminate AKK, but you do have other bacteria remaining in the gut when you do this, and also shown here by qPCR, the depletion. So when you do this and you do the tetracycline treatment, and now you reintroduce AKK, and then again giving the total body, total abdominal radiation again, you can see that we'll build back the AKK. So this is just a confirmation that when we give AKK back, you have AKK coming back up. Obviously, fasting increases some in AKK in some mice as well. So that's why you're seeing that. Yeah, so here the results are very clean. As you can see, you have the protection was mostly through tetracycline plus AKK along with the fasting, and the fed mice and all died. So beautiful experiment clearly shows that you need fasting plus AKK to actually protect the mice. And that then sets you up for what is it doing, right? So now we wanted to ask, what is AKK doing, and what is intestine doing without AKK? So you want to understand what is the contribution of AKK and what's the contribution of fasting as such. So we focused on obviously short chain fatty acids because they have direct role in histone modification, which I showed you earlier, because acetate, obviously propionate, hydroxybutyrate, then et cetera, et cetera. There can be many layers of histone modification. Even serotonin modifies histones, and the roles of those are not well known. So you need to understand all of that. So here first we set up an organoid system, and this is just a way, just a slide showing how we set up the organoid system. Oh, sorry. Okay, I'll try to wrap this up quickly. So yeah, we set up organoid system through a specific way. It has been established earlier, and now we start to give them different different short chain fatty acids, or the ones we find in our metabolite analysis, with or without AKK or with or without beta hydroxybutyrate. You can see they have specific roles in modulating specific histone modification patterns. Okay, and we then profile them through that epigenomic analyses. And I'm not going to show you any of the data because this is complicated for today's audience. But what you see is there is a combinatorial effect of AKK plus some of these metabolites specifically that drives that mimics the fasting condition to protect the mice. And again, this is just one of the slides to show that AKK does this in vivo as well. So this is H3K27 acetylation. You can see beautiful staining with AKK in the crypt, whereas in minus AKK, you don't see that. And H3K27 acetylation is a key marker for all enhancers in the cell, which actually sets up most of these positively setting up state. So basically, our marking model is that in the fasting, you have effects on different compartments. You have I didn't talk about this. You have higher goblet cells that produces mucins, and that leads to actual enrichment of AKK in fact that produces specific short chain fatty acids. Some of these are your usual culprits and others are not. And this modifies histone states in a specific way to create gene activated states on certain genes as well as gene priming on others. And this is also contributed through just small intestinal stem cells through production of HMGCS2, which is independent of the AKK, so microbiome. So it is a combinatorial effect. And finally, we've done more single cell experiments to understand how this core transcription factor circuitry plays a role. So what we did here was we took the stud and fasted mice, did a single cell multiome, which actually gets you the data from same cells. So we look at single cell RNA and single cell ATAC. This is something we have expertise in my lab. We do a lot of this, and we generate this data plus the histone modification data, use specific algorithms to try to understand what's going on. And we use something like this. This is a CRC mapper algorithm. Happy to talk about this later. But now we can identify specific regulatory circuitry that participates only in the fasting condition, and that creates that cell states that actually protect the mice. So that was one. Then we've gone on more recently and done experiments taking the normal mice, the SPF controlled mice, germ-free mice, AKK monoclonal mice, so only AKK in that condition. So germ-free plus only AKK, and then tetracycline treated, which is only AKK, which is AKK-free, as I told you earlier. So then you can do a single cell experiment. We've done this well now. Same single cell RNA and multiome, and you can identify different clusters of information, which is shown here. And then you can find specific observations that occur due to AKK specifically. Now, so we are now learning specifically what AKK modulates it. One of the key things we see, and I won't talk about this at all much, that there are some specific effects on the stem cells and the goblet cells, but importantly, they also start to show now effects on B cells, the T cells, and the macrophages. So we're learning also in how tumor microenvironment might be modulated by fasting as well as AKK. So yeah, this is my last slide. So the take-home message for everyone is, if you want to take one statement, the fasting associated microbiota contributes to protection of SI in small intestines through damage induced by chemo and radiation by modulating the epigenome in the small intestinal cells. Okay, thank you.
Good afternoon. Our next speaker is Dr. Shobhik Mukherjee. Dr. Mukherjee is an assistant professor in National Institute of Biomedical Genomics, where he studies the host metagenome interactions in chronic human diseases. His lab is the first to profile the healthy skin microbiome in any Asian Indian population and identify the differences with Western skin microbiome. Currently, Dr. Mukherjee's team is characterizing the host microbiome dysbiosis in dry skin disorders like atopic dermatitis, psoriasis, and development of antibiotic resistance in chronic non-healing wounds. In collaboration with THSTI and CHRD-SAS, his research team is involved in investigating the role of maternal microbiome in adverse pregnancy outcomes and infant gut microbiome in child growth retardation, respectively. He has many publications to his credit, which includes papers published in PNAS, Nature Communications, etc. With these few words, I would welcome Dr. Mukherjee to deliver.
Thank you, THSTI, and I will thank Bhavatosda for inviting me for this talk. It's good to come back to THSTI every time because we have multiple collaborations here. So the topic of my talk is host microbiome interactions in chronic lifestyle disorders in Indian population. And standing in this M.K. Bhan auditorium, I fondly remember Professor M.K. Bhan. He was a great, I don't want to say was, he is a great visionary. And Professor Bhan, we used to have magical conversations with him, and this is how he used to create magic in youngsters like us. And, this is his picture in the first NIBMG Foundation Day. So the foundation of several institutes like HSTI, NIBMG, all of these focused institutes on multi-omics have been done by him. Coming back to my talk, the major aim of scientists is to find the missing heritability of complex diseases. So when I say lifestyle diseases, it's like the complex type kind of diseases. And when we started doing our research, during our PhD, we were sold to this particular thing, common disease, common variant hypothesis. That if you have a common kind of disease in different populations, the variation that is associated or causing the disease will be common. And that was the era of candidate gene and GWAS studies. This could not identify most of the missing heritability. Then came the era of common disease rare variants. When we became, we are doing postdocs or we became young faculties at that time, this was there that although diseases may be common, there are some pathways. Genes don't interact, genes don't function in unison. So there are some pathways, and in the pathways, there can be private mutations in different populations. Then came the era of next-gen sequencing. But still now, not that more than 40% or 30% of missing heritability has been identified. So where is the problem? The problem lies in the etiology of complex diseases. It's not the Mendelian kind of genotype-phenotype correlation. It's like genotype, there are many subphenotypes, there are the environment, and then that contributes to the disease. So epistatic interactions and different kinds of interactions are there. So now there is this era is of MWAS, metagenome-wide association studies, and there are studies that are saying that the human microbiome is the answer to the missing heritability problem. So microbiome, many of the speakers have told that it is our second genome, and when the Human Microbiome Project started, it was like they said that 90% of our cells are microbial. Well, that's not true. It has been revisited, and it has been said that 50% of our cells are microbial in origin. That also makes it very interesting. If 50% of myself is not me, then that's also interesting to study when we are studying some connection to a disease. Microbiome, most of the speakers have harped on a particular organ, the gut. I would like to mention that microbiome is everywhere. And, I'll skip this slide. So microbiome is everywhere. It is in skin, it is in ocular surfaces, it is in nose and many other places. And they all contribute to some disease or the other through some axis. Now, when I talk about the NIH microbiome project, quickly, they did 250 normal volunteers. As you can see, normal is mentioned there. I don't know what is normal, but they tried to mean healthy. They tried to create a baseline. Like you see, the Human Genome Project also tried to create a baseline. So microbiome project also tried to create a baseline based on which people in different parts of the world will try and understand the perturbations in diseases. And there were some seminal publications that were very enticing. Like the GWAS gold rush, when we were doing our PhDs, now is the time of the microbiome gold rush. And any kind of diseases are associated with microbiome, and Nature and Science are flooded with publications of the kind of microbiome-based association studies. But the problem is my slides have gone some epigenetic modifications. So please bear with it, the headings and all. So the problem with the microbiome is that although the human genome has been characterized to get a baseline, but the healthy human microbiome is not same. This is one of the kind of papers that opened our eyes that we cannot rely on the Human Microbiome Project for studying the Indian or any other populations outside the Western countries. The gut microbiome of the Hadza hunter-gatherers were completely different. They were containing something like Treponema, Prevotella that are considered dangerous to the Western gut. but they are all very healthy. It depends on the ethnicity, it depends on the food, it depends on the climate. I'm not going to go into the details of it. So coming back to our work, we primarily work on skin. Skin is the largest organ of the human body, and it also has got these innate immune system harbored in it, the cell surface receptors, and how does they interact with the commensal microbiome of the skin. And you can see the oily, dry, moist, food, different parts of the skin also has a distinct biogeography of microbiome. Yeah. So why study skin microbiome? Because the skin encounters continuous assault by the potential pathogenic organisms, and it also exhibits significant topological, temporal, and inter-individual variations. It resists colonization of opportunistic pathogens, and sometimes it also regulates the complement system, which in turn also regulates the composition and diversity of the skin microbiome. It has been seen in C-section babies and in the vaginally born babies. There is a difference in skin microbiome that can predispose them to allergy and asthma in later parts of their life. These biases in the skin microbiome has been associated with atopic eczema, psoriasis, different kinds of skin inflammation. So it's important to study the skin. So we ask this question: is that what are the factors that are contributing to this inter-individual and temporal variations in the human facial skin? We have been funded by Unilever, and what we found, Professor Majumdar also shared this particular publication, and that was the first study that we did, and we found a distinct skin microbiome in the Indian samples compared to the Western. And also that the sebum and hydration levels in different parts of the facial skin, they actually contribute to the increase or decrease of specific bacteria in the skin, so that we can devise a prediction model which is more than 70% correct of the oiliness, dryness of the skin. And this one was one of the first of the healthy Asian or Indian skin microbiome, and is being widely cited for. Currently, I'm going to share with you, after we made the skin baseline, we tried to study the skin microbiome in one inflammatory skin disease, or autoimmune kind, called atopic dermatitis, or commonly called as eczema. Here, our major aim was to understand the host microbiome associations, not only the microbiome, 50% microbiome, 50% host. So let us study both of them. It has been known that there are skin barrier permeability genes which contribute to the atopic eczema in different populations. Like filaggrin is a protein that binds with collagen and makes the skin tight. Now, filaggrin is coded by a gene called FLG, and this gene has got different stop codon polymorphisms distributed in different parts of the world. We thought that what is the Indian prevalence thing of this loss of function mutations, we will study and stratify the patients and then find out what is the difference in their microbiome. So our question was, do the presence of these host risk genotypes modulate the composition of the lesional skin microbiome? And we will slowly study their pathways and gene families too. So filaggrin is a part of a epidermal differentiation complex. It is present in chromosome 1. It's a big battery of skin genes. 55 genes are there, and they are all of skin, and it is 16 MB long. So filaggrin is only one of them. And as you can see, profilaggrin has to be cleaved to filaggrin, and this filaggrin monomers goes and binds to the collagen. Well, I told about our research objectives. So let's get into the work. Now, the study design was that we collected biospecimens from AD cases and controls. As you can see, the cases and controls vary in the ratio 1:3. We did it purposefully because we wanted to adjust for the inter-individual variations in the healthy skin. So this was age, gender, and site-matched controls and cases. So we studied the lesional and non-lesional skin swabs for the microbiome study. We collected the human blood for the host genetic study and human serum for the cytokine study. And the areas that were taken is antecubital fossa and also the neck or the cervical region. The criteria for choosing the atopic eczema is based on Williams criteria. And we collected blood and did Sanger sequencing of the entire filaggrin gene at that time by using 16 overlapping primer sets. I'm not going to the gory details of what is Hanifin and Rajka or Williams criteria. They were experienced clinicians who did this. Our inclusion criteria only added not on any antibiotics for 15 days and not on any emollients or something for the last 7 days, because that also influences the skin microbiome. So filagreen, as you can see, these are the loss of function mutations globally present. And the ones that are highlighted are already found to be associated with the European, African, and Asian skin, meaning Chinese and Japanese populations. We actually tried to see that, is there any such LOF distribution in the Indian population? We found that in our cases and controls, we don't have these particular mutations. Only in the cases we have 3 such sporadic one individuals each, some of these mutations. Now, having said that, our sample size was low. So what we did, we tried to find out what is the prevalence of these filagreen loss of function mutations in the general Indian population. We had these more than 500 whole genome done from the Genome Asia project in NIBMG from more than 20 different ethnicities in India. We tried to find out the prevalence of all these LOFs. As you can see, it's only 0.6 out of 533 individuals, compared to in the Europe or in the 1,000 genomes, they are more. The prevalence of these loss of function mutations in Europe are significantly more than in Indian population. So our question was, what is happening? Why? So this is a distribution in all the ethnicities, and I'll not go into the gory details of it. So this shows that the Indian AD pathogenesis is not driven by filagreen mutations. There are studies that have shown only 14% of heritability would be explained by host genomic factors in AD. So what other factors can be responsible? So we did this swab sample collection, and then we did DNA isolation, and we did this shotgun sequencing. We didn't do 16S, we did shotgun sequencing for this study. So the data analysis, I'll not go into the details of the data analysis, but here, as you can see in Metaflan, we have got more than 17,000 microbial genomes integrated. And the taxonomic classification can be done up to the species or strain levels, unlike the 16S, where you can do up to the genus level. Then we have this human 2, where we have gene families and pathways identified and curated. This is exactly following the similar protocol done by the Human Microbiome Project. We found some phyla, genera, and species. Among them, what we found is S. aureus is actually significantly higher, 22%. This percent is a relative abundance of the DNA sequences. So 22% present in the cases and 0% in the controls. Whereas S. hominis is just similarly present in the control, 16% in the controls, and 0.25% in the cases. So we thought that what is happening, are we doing some wrong in analysis? So we tried to analyze again and again with different kinds of pipelines, but we found the results are coming same. Then we found that, by some co-culture assay, that S. aureus and S. hominis cannot stay together. And some publication came concomitantly that says that S. hominis has some proteases that can cleave the S. aureus. Now, coming back to filagreen, although we didn't get any loss of function mutations, we got some amino acid changing mutations that in silico came to be damaging. We got more than 15 or 16 such amino acid changing mutations. Among them, 5 were them that are by in silico we are finding to be damaging mutations. And these are the 5 RSIDs, the reported SNPs and the changes in their alleles. And you can see there they are changing the amino acids. This is their minor allele frequency in the cases, and this in the controls. So what we did, by the damaging allele, presence or absence of the damaging or the mutant allele, we tried to make a score. And this is called damaging allele dosage score, by which if you are capital A, it is 0 because it's the reference allele, no risk allele present. If it is capital A, small a, it is 1. And if it's small a, it is 2. So we just went on adding this thing for each individual of all these 5 SNPs, a haplotype based approach. So it's like, these are all the 5 SNPs. And in these 5 SNPs, we tried to add up their risk allele scores. And this is exactly following a particular publication in Genome Medicine, where they have tried to integrate them. Then we regressed them with the the core microbiome and tried to find out what is happening. So we found that there is a host microbiome interaction, that is S. aureus, that is the bacteria which is associated with AD, that is present more in those individuals where these damaging alleles are absent. But where the host damaging alleles are more present, S. aureus is very less and proteobacteria is increasing there. We then did the functional microbial assessment by shotgun, and there we found 3 particular pathways: histidine metabolism, Staph aureus, and arginine to proline metabolism pathway to be significantly enriched in the cases than the controls. So these are the gene families that were found to be significantly higher in the cases that inhibits the innate immune system. Now, in the histidine metabolism pathway, there is a particular enzyme, histidase, that actually cleaves the L-histidine to a compound called urocanate. And this urocanate is cleaved by another enzyme called urocanase. And these are actually, urocanate and histidine are called natural moisturizing factors for the skin. And these pathways in the S. aureus can have those enzymes that can cleave these things and make them dry. And urocanase is not present in the humans. Histidase is present, but not in the skin so much, in our other organs. So we actually devised a model, a predictive model, that all the filaggrin loss of function mutations are not truncating the filaggrin protein in India, in Indian samples, but these S. aureus, histidase and urocanase are actually chopping them down, because filaggrin is a histidine-rich protein. Now we have published this recently, and we are now doing this ongoing work is on the skin microbiome and virome profiles, along with their gene families and pathways, and also validating this result in the in vitro cell culture assays. We have devised this as a pilot study, and we have calculated the power and found that we need 50 AD cases and 70 controls to reach 80% power. In the phage, we have got some interesting results. I'm not going into the details of the analysis, but we have found some lytic phages of these S. aureus in the controls, and some lytic phages of the S. hominis in the cases. We are doing more analysis now. Last few slides is that this is our lab, and we have a central theme to unravel these host genomic and metagenomic crosstalk associated with sustenance of human health and characterize their perturbations. I have few students, and Ankita is here, and she is actually with a poster here. And I have 4 PhD students and 2 data analysts. And these are some of the other researches that we are doing. We are doing with psoriasis. We are funded by SARB on psoriasis, by Department of Biotechnology on maternal and child health. Some of us, we are doing with THSTI, with SAS, with IPGM here in Kolkata, we are doing diabetic foot ulcers. These are 3 publications from our lab. Our lab is 6 years old, and we have done these 3 publications in 3 different areas. And I'll not go into the details, but please have a look into my student's poster. And she has come with the child growth failure and their role in, with the gut microbiome. And this is the study design. I'm not going to the details of it because she will present. We have done an India EMBO symposium on human microbiome in 2019, where we had invited some of the speakers from India and abroad. And these are some of the glimpses. Bhavatosda, Dr. Shinjini were all there. And after that, COVID started, so we had to stop the workshops. But now that we have all been back to normal, we hope that we will be also able to start our workshops. We have a genomics gallery. We know NIBMD is known for its genomic facility. We have also made a microbiome facility. Beyond this, sequencing and analysis, we are now doing anaerobic culture. We are now doing co-culture assays. And we have now all these functional armaments in NIBMG. And soon we will meet, and I will be able to show some functional results also. This is my HMR team, and thank you.
So, thank you, Dr. Mukherjee. Yeah. I request you to come and sit over here. So the last speaker for today's this session is Dr. Krishna Mohan Atmaguri. Dr. Atmaguri is currently an associate professor at THSTI. Atmaguri's research focus is in evaluating the transmission dynamics of bacterial infections causing culture-positive sepsis among Indian neonates in hospital settings to understand host pathogen interactions and exploit the findings towards the development of novel vaccine candidates, therapeutic agents, and diagnostic tools. He has received the prestigious Ramalinga Swamy Fellowship from the Department of Biotechnology. With these few words, I request you to give your presentation. Thank you, sir.
Thank you for the kind introduction. So today I'm going to talk about little work that we have initiated on neonatal sepsis, trying to understand some of the factors, the environmental factors, as well as the host factors, which basically trigger the neonatal sepsis. First, I should thank Dr. Garg as well as Baptosh for the opportunity. And I also thank all my lab members who worked very hard to put the data which I'm going to be presenting. So in general, sepsis is defined as a life-threatening syndrome that leads to multi-organ failure due to the dysregulated the host response to an underlying infection. So for a long time, it was slightly confusing as to whether the pathogens actually trigger the sepsis or it is simply an dysregulated immune response to some non-infectious agents too, which was shortly called as SIRS. But later on in 2016, the definition was altered to saying that sepsis is always because of an underlying infection. So the infection basically can lead to possible different organs failure, and different organs could get infected and lead to finally the sepsis and the septic shock, which mostly ends with death. That's the true and the ugly face of the sepsis. You can see that there's a lot of challenges one has to undergo if the sepsis was to be severe. So just to give a flavor of the burden, approximately 50 million cases are reported, of which 11 million deaths. It's sort of one of the major killers, which is basically not recognized clearly because of the underlying differences with respect to different pathogens causing sepsis. So primarily it accounts to approximately 20% of all deaths. Then so if you look at children, it accounts to 50% of, oops, sorry. Can you change this? So in children, it accounts to approximately 50% of all cases and 25% of the sepsis-related deaths. And if you take the disparity, approximately 85% of the sepsis deaths is primarily in the low-income and the middle-income countries as compared to the high-income countries. So if left uncontrolled, sepsis is thought to be the #1 killer by 2050, which can lead to annually 50 million deaths. This is complicated by the further problem of the AMR, given that most of the pathogens which cause sepsis are either MDR or XDR. So the India alone accounts for approximately 40% of the global neonatal deaths because of the sepsis. And it's such a huge problem, which to a large extent has been difficult to address, primarily because of the AMR problem. So there are a lot of pathogens which basically cause the sepsis. Any opportunistic pathogen for that matter can cause sepsis, but the primary ones are the escape pathogens. In terms of the bacteria, there are a lot of viruses which can also lead to the sepsis, as well as the fungi. In general, mostly the bacteria, because they're able to multiply so fast, and gram-negatives especially, they have taken the primary focus. But in general, any of these pathogens can cause sepsis because the culture positivity of sepsis is only approximately 20%. The rest of the 80% is culture negative in the sense that they're non-culturable because either the media is not favorable or the pathogen cannot be cultured under the conditions. So there was a huge study which was done. So the data or the sort of project work that I'm going to be discussing is a huge collaborative project between AIMS, and then Baptosh is also involved in these particular projects. So the Denny study, which was done between 2010 and 2015, basically identified a few things. One is in terms of saying that the neonatal sepsis is rampant with basically the gram-negative pathogens, Acinetobacter, Klebsiella, Pseudomonas, and E. coli. And what is interesting is that almost 60% of the neonatal sepsis happens within the first 3 days of being born, which is called as early onset sepsis. And approximately 90% happens in the first 7 days. So indicating that this is a huge challenge, it needs to be addressed as quickly as possible, because the current diagnostic tools which are available basically takes anywhere between 3 to 7 days before one can even identify what the kind of pathogen is. And you can see, irrespective of what the day of the birth is, from the day of the birth, basically you can see that the pathogens are sort of identical, indicating that the early onset sepsis and the late onset sepsis is possibly caused by the same kind of pathogens. And to a large extent, as I said, they're basically MDR and XDR for different antibiotic resistance. So primarily raising a few questions, and to some extent we are able to try to address these questions by by establishing a multi-institutional, multidisciplinary, multi-hospital sepsis program. This basically came into inception in the March 2021, and it took almost a year before the cohort started. Basically, the enrollment started in March 2022. So we have some data. It's a currently ongoing study, so that it basically raises a lot of interesting findings and concerns, which basically could give us some highlights of why the sepsis is so rampant. So the primary goals of this particular program was to see how best we can reduce the AMR as well as the sepsis burden, and in order to see how best we can understand the underlying mechanistic role of these particular pathogens with respect to driving the sepsis. So there are a lot of projects in this particular sepsis program, and I'm not going to discuss into that, but THSA is involved in the transmission dynamics, which I'll be talking about in a brief while. And there is also a JIVA study, which basically we're trying to look at what are the kind of host SNPs, which basically could trigger the sepsis. I'm not going to talk on that particular one. So there is also Baptosh, to a large extent, is looking at the evolution of the gut microbiota. So I'll be talking about that also a little bit, trying to understand what is the kind of the gut microbiota which is present in the neonates to begin with, and what kind of a gut microbiota which possibly could lead to sepsis. And if you were to recover, what could be the possible consequence of that microbiota in terms of the profile. And the other project that THSA is involved in is also the host genome response. I'm not going to talk about that again. So one of the questions which basically is very important is to understand how do these neonates basically get these pathogens? Is it coming from the hospital? Is it coming from the mother? And these are classically defined as this being vertical transmission, and this is called as the horizontal transmission. And so because of that, we try to understand what is the kind of the transmission dynamics, at least in the Indian settings, with respect to these neonates. And the primary objective of this was to look at least 4 different pathogens. So the transmission dynamics certainly will vary depending upon what pathogen you're looking at. But we looked at, given the burden and given that the gram negatives are so intense in terms of how they cause the sepsis. So we looked at Acinetobacter, Klebsiella, Pseudomonas, and E. coli to see what kind of a genetic relatedness they have with respect to the isolates that we would get from the mother and from the environment. So the hypothesis is that basically, possibly these pathogens have a genetic relatedness very close to the environment rather than to the mother. So the work plan was basically first, we got 4 tertiary hospitals, and basically the swabbing is done of these hospitals, the immediate neonatal environment. We are looking at NICUs, the labor room, and the OTs. And so then you have approximately around 4,000 mothers from where you get these neonates, and approximately 1/10 of them will get into the NICUs. And so the environment is continuously swabbed. the mother's rectal and the vaginal tracts are swabbed, and the blood cultures from the pathogens, from the neonates pathogens, blood cultures are basically taken up. And so they are basically going through VITEC, which is able to identify the pathogen, also able to give us the AMR profiles. So once we have the Acinetobacter, either Klebsiella or these pathogens of interest, they get stocked, and basically then they go into the sequencer, and then we do the comparative whole genome metagenome or the whole genome analysis. So there's a sample size depending upon the number of mothers. You end up looking at approximately 40 neonates with Acinetobacter, 40 neonates with Klebsiella and aeruginosa, 15 with Pseudomonas and 40 with E. coli. So far, quite a few samples have been processed. I'm not going into the details, but we do have a lot of culture positives and culture negatives. In general, this is what is the trend which is seen and reported earlier as well. And so we also processed quite a few of these environmental swabs in order to look at whether we can see in terms of the metagenomic analysis, what kind of the profiling we can see with respect to bacteria. And also we have processed all the swabs of the mothers as well as the environmental swabs for microbiology culturing to see if they can grow some particular pathogens of our interest. What came as surprising is that you can see that these are the top 10 reservoirs of the NICU. So where basically you would want a sterile environment. So by a repeated culturing, we can constantly see these reservoirs replete with bacteria. By PCR also, we can identify that the 16S is amplifiable. Then you can see different pathogens basically being isolated from these samples. So indicating that the NICUs from these tertiary sites are basically replete with the pathogen when they're supposed to be sterile. So quite possibly this is one major source from which the neonates could be getting infected. Of course, we don't have the data at this point of time in terms of the genetic relatedness, but we'll be looking at that soon. The same thing with the labor room as well as the OTs. We do see all the pathogens of our interest. So indicating that basically the NICUs, the labor rooms, as well as the OTs are actually replete with the pathogens which should not be there. And these are some of the reservoirs that basically show those enriched organisms. And of course, when we do the metagenomic analysis, they fall into quite a few taxa. And if you go into the details to go all the way to the genera, you can also see the Acinetobacter, Enterobacter, E. coli, Pseudomonas, all basically showing up. So indicating again that quite a few of these hospital reservoirs are primarily maybe the major sources of the pathogens. So indicating that at least 1/5 to 1/3 of the immediate hospital environment are replete with these pathogenic bacteria. And they also come up with a flavor of different antimicrobial resistant genes. You can see that they're highly resistant to aminoglycosides, beta-lactams, and other antibiotics. So we also looked at the rectal swabs of the mothers with respect to some of these pathogens. And you can see almost these pathogens. You start wondering where exactly are these pathogens coming from. Of course, we will be able to say that once we have the comparative genomics done. But what came as a surprising, whether you look at the rectal swabs or the vaginal swabs, is that so far, all the neonates who basically are blood culture positive or sepsis positive, none of them basically came from mothers who basically carried any of these 4 pathogens. So that came as a surprise to us, indicating that the pathogens possibly are not coming from the mothers. I repeat this, all the mothers who basically had Acinetobacter, Klebsiella, none of their neonates were sepsis positive. Or those neonates who were sepsis positive, none of the mothers were carrying that. So quite possibly indicating that maybe this is the environment which is driving this. So I'm going to skip the summary in the interest of the time, but just to say that possibly the horizontal transmission is more as compared to the vertical. The other objective which Baptosh is largely looking at is to see whether there is a diversity in terms of the composition and the dynamics of the microbiota of the gut. Just take 5 minutes to finish this. So he's not only looking at the healthy, but he's also looking at different time points in the healthy, also looking at when the suspicion happens and during sepsis, as well as at the recovery. This is very important to basically see whether there is a consequence of the changes of the microbiota that is leading to the trigger of the sepsis. And so he's looking at not just the bacteria, but also looking at the virium and as well as the fungi. And so in the morning, of course, there was some small discussion on in terms of the transport media. So this transport media was also sort of kind of analyzed, basically made sure that this is able to, retain the composition and the integrity of all this, the DNA, which is basically coming with them for a long time, so that this could be used for the metagenomic analysis, past storage. And when he looked at the suspected as well as the sepsis positive, sorry for this, but also the recovery samples. So there's not much of a difference at this point of time, given the sample number, but at least it is showing that Klebsiella, E. coli, Enterococcus are pretty much enriched within these particular gut microbiota of the neonates. So the same thing was happening with respect to the fungi. There's not much difference at this point of time, but you can see Candida alternaria basically coming up. And these are known to be pretty much involved in pathogenesis to a large extent. So also interestingly came out is that if you look for neonates who basically are blood culture positive, for example, Klebsiella here, they not only are showing Klebsiella in their gut microbiota, they're also showing Acinetobacter and E. coli. So indicating again, you have a heterogeneous population and a gut microbiota, which basically one of them could be triggering the sepsis. So the question is, where is this particular pathogens coming from? And again, we see that they carry a lot of antibiotic resistant genes in general, indicating that whether you look at the hospital environment, or you look at the gut of the neonate, or you look at the vaginal or the rectal swabs of the mothers, they're all replete with these pathogenic bacteria and which are highly resistant to a lot of antibiotics. So I stop there and thank you for the patience, and basically I can answer any questions, if any.
So with this, we come to the end of today's afternoon session. I request you to come and sit over here. I'm sure you all would agree that we had a wonderful time, and the speakers would be more than happy to take questions and give you answers.
Any questions?
Yeah, my question to Krish is about the transmission. You said you are looking into one of the objectives. So this transmission is maybe from the environment. The other is from the gut to the bloodstream. So there are a lot of literature available. Say, for example, zonulin is one of the markers that that indicates the leaky gut. Means the tight junction of the cells are leaky, so that the so-called commensal or the pathogen goes into the bloodstream. Are you looking this point into in
So we're not looking at the gut of the neonate to the bloodstream. We're not looking at that part. What we are looking at is the source in terms of the reservoir. And if it is from the mother, if the mother basically, so how does it come to the neonate? So it has to be either from the mother or it has to be from the environment. Of course, there is a challenge that it would have gone from the environment first to the mother and then basically come in. So that's the reason we are doing a whole genome analysis to basically see how best there is genetic relatedness. Also, it would give us an indication of if there are some 53 reservoirs, do we have the isolates from specific reservoirs, which could be the major source? So I think in terms of the transmission dynamics rules, one of the rules is to basically see if at least 2 neonates get infected by the same organism. Of course, one of the reasons why this sepsis could trigger in neonates could be that it is basically from the gut microbiota. But normally what is known is most of these sepsis cases, at least based on what the clinicians say, is monomicrobial. Quite possibly gut being the polymicrobial, it's tough to say how one pathogen would dominate when you have almost all of these pathogens in the gut.
So my suggestion is to look into the zonaline.
Okay.
So that is an indicator of a leaky gut.
Okay.
Yeah.
I will talk to you later. Yeah. Yeah.
I have a question to Vineet. So Vineet, when you said about the combination of this anti-inflammatory diet with the FMT, would you want to try, I don't know, maybe anti-TNF, combination of anti-TNF or anti-inflammatory drugs together with FMT, because that is more direct, because the diet, what you're giving, anti-inflammatory diet, is just, there's no kind of proven thing, right? It's there that is anti-inflammatory.
No, Amit, I know you always have the bright ideas. There's no doubt about that. So we have already started this study whereby we are combining one oral small molecule with FMT and seeing whether it potentiates the effect of oral small molecules. As I said, it's a pleasure always interacting with you.
Thank you. So just adding to the Kunal's question, the diet. So basically, one of the major challenge, what you said, is screening the right individuals based on the different analysis, not only including the psychology and other things. And then you put them onto the fasting things, right? Then you take the isolate do the FMT from those fasting individuals.
So Amit, that study has been done in Scandinavia, where they didn't do fasting, but they enriched the diet of the donor. It is something like bakre ko mota taza banate ho na, something like that.
So my question to
Baptosh, can I just ask you a question related to that? Sorry, I'm going to be a little bit of a bully. So Vineet, the same question of a donor. You're taking a cross-sectional screening of disease, is it? So would that make a difference?
When we say cross-sectional history, we also obviously look into the past history. When we take cross-sectional history, then we take things like recent events, any recent history of GI infection, antibiotics, PPI, and previous history of hypertension, diabetes.
Documentation, or is just history based? I mean, would there be some documentation?
No, the test in terms of this is history which is taken. And if there is no history, rest we are doing all the blood tests, the viral markers, and the stool tests.
Thanks. Yeah, Baptosh.
Yes, my question to Professor Vineet Ahuja. So particularly for the IBD, so when you look to the fungal signature, so in the OS, it was quite evident that if you have the increased abundance of the candida, then they're quite susceptible to the IBD. But when you look to the healthy Indian, almost 90% of they already have the candida. Particularly the Indian IBD patients.
I'm not able to understand, please. I could not follow.
What I'm talking that if you look to the fungal signature in the IBD,
fungal signature,
particularly for the Europe and US, they reported that if you have the increased number of candida in the gut, those patients are quite susceptible to the IBD. But when we look to the normal healthy Indian, we do find that almost 80 to 90% of the Indian, they already have candida in their gut. Do you find any Indian IBD specific fungal signatures like bacteria?
So Babatosh, so that is a good thought. And right now, I think Saurabh may show the data. So we have analyzed along with Tarini, the microbiome of patients with Crohn's disease, not only the bacterium, but also the microbiome. Similarly, in this study, which I showed you, we have also looked at the microbiome of these patients of ulcerative colitis. I did not show the data today. So microbiome obviously has a very good, not only it has an effect in the sense, but it also has an interaction with the bacteria. And that interaction is very, very important. Not only there's an interaction between the microbiome, the virome, and the bacteria, there's also an interaction between the mucosa bacteria versus the crypt bacteria and the fecal bacteria. So there is a set of networking which is going on, and this networking needs to be deciphered. And today, I think this networking can only be decided with the help of a very good bioinformatician. And I think that's why I say we are blessed to have a team, and Tarini is doing that job for us.
So the second question also I have exactly for you. So particularly for the FMT. So when you select a donor, do you also screen the fungal component in the donor?
Can you please repeat,
So,
Baba?
for the FMT, when you select a donor, so do you also look to the fungal component in the fecal sample of the donor?
No, right now we are not checking. We are checking for research purpose for the microbiome of the donor, but in terms of enrollment at that time we are not checking. Subsequently, when you enroll a donor, you enroll a healthy donor, and I fully agree with you. That should be the next step. In terms of when you have 10 donors, check their microbiome also. And if you find somebody is very rich in Faecalibacter prostate and all, they may be very much better donors, and you can enroll them.
Thank you, sir.
One more. You have a question? Okay. Can I ask? Okay, that's to Kunal. Kunal, both Nitya and I have looked after a lot of pediatric diarrheal diseases, and we are bringing up the same question that when you talk about fasting being beneficial, that's not the policy we follow for our children during diarrhea. And so this is a very practical, clinical based question. What are your thoughts on that? Because we feel, and there are studies and evidence to show that children who are given ORS with feeding do better than children with just ORS or those with just fasting. So, and this is old data. And that's why for decades now, fasting is not allowed for treatment in diarrhea. So any thoughts on that?
Mice versus.
Exactly. I was going to say, I mean, our studies mostly focus from, damage perspective, right? The small intestinal architecture perspective. I think, sure. I mean, it could be a mice versus men question. It could be, are you capturing the same biology with ORS diarrhea plus this? I don't know. I mean, so I don't have enough knowledge about the with the infants and also no idea. Sorry. It's interesting. So obviously we should look into that as to why that happens. But in our case, we clearly see, In fact, along these lines and using similar like diets that enrich in the same sort of metabolites, that various trials are being done at MD Anderson actually, and they're seeing very positive results with it. So I think, and one of the key aspects of this, and this may not imply to what you're talking about, the ketogenic diets actually start to show similar. They can't capture completely what fasting does, but at least partially they can see the same diet and protect the intestinal stem cells.
You would be happy to know, interested to know that Ayurveda literature also talks about absolutely the same effect. And that is why in all religions, you'll find there's a period of fasting that is supposed to decrease the aging, increase the longevity. And
thank you.
Yeah, Dr. Ahuja, I have a couple of questions to you. You said that the bacteria have a distinct tissue trophism. So then when you go for this FMT, how would the fecal bacteria repopulate the entire gut?
how would bacteria?
The fecal bacteria populate or repopulate the entire gut. When you say that there is a distinct tissue trophism.
So when we say that there's a trophism, that is like in the sense that the fecal, when you let us say it is putting in all the contents inside. So when we do it first time, let us say, we go right up to the junction of the small and the large intestine known as the ileocecal junction. And the first infusion is done out there. Subsequently, the infusion can be done in an enema form, or you can just leave it in the left colon. Now, it depends upon the engraftment, depends upon many factors, obviously. The first factor is, if you look at, we can start from host genetics. Some of the hosts, the engraftment would be better. Second would be, what is the bacterial content composition that would decide it. Third is the disease activity, which will decide how much engraftment is going to be there. So this has what we showed also, that when this thing was done, the engraftment changed the microbiome at the fecal level, as well as at the mucosal level, as well as at the crypt level. At all three levels it changed, and possibly that is why the effect comes. If you change only the fecal microbiome, this I'm not very sure, but it's a thought from our side that possibly the effect may be very transient. Whereas when you change the mucosal microbiome, the effect may be probably more long lasting.
And the other question is, how similar or dissimilar is the microbiome in Indian patients when you compare it with patients from different other countries who are suffering from IBD?
So logic says it has to be different. Like in India, you have more of vegetarianism, you have more of carbohydrate diet, so you should have more of Prevetola. And there is data which shows that Prevetola is more compared to Western population. But when it comes to disease, this is my observation, just which is obviously, I'm not very sure about the thing of, is that when I look at patients talking about liver disease, then I hear patients who are talking about neurological diseases, then I hear patients who are talking about ulcerative colitis, or if I hear patients talking about cardiovascular disease, everywhere the gut microbiome and the metabolite signature is almost similar. The only outcast out there is Kunal's bacteria, Akkermansia mucinophilia. That seems to be a little paradoxical. Sometimes when it is high, it is good, sometimes it is bad, right? Whereas rest all the bacteria, they are just moving through the same direction, the universal signature. Secondary metabolites, secondary bile acids, tryptophan metabolites, short chain fatty acids. When it comes to the bacteria, majority of the bacteria in terms of Faecalibacterium group and the bacteroidetes group, it would move the similar way for majority of the diseases. Although you expect a very strong geographical variation depending upon the ethnicity, host genetics, and the type.
Thank you.
I think we need to wind up, right?
Yes, ma'am. Thank you to all the session chairs and the speakers for the wonderful session. I now request the session chairs to kindly felicitate the speakers. First, I invite Professor Shinjini Bhatnagar to hand over the memento to Professor Vineet Ahuja. Ma'am, please. Next, I request Professor Shibnath Majumdar to hand over the memento to Professor Kunal Rai. Sir, please. Now, I invite Dr. Nitya Vadva to hand over the memento to Dr. Shauvik Mukherjee. Ma'am, please. I once again call upon Shinjini ma'am to felicitate Dr. Krishna Mohan sir. Now, I request Professor Sharmila S. Mande to kindly come upon to the stage to felicitate the session chairs. Ma'am, please. To Professor Shinjini Bhatnagar. Next, to Professor Shibnath Majumdar, sir. And now, to Dr. Nitya Vadva.
We will now move on to our next session. So the theme for today's fourth session is microbiome and methods in microbiome research. For this, I request Professor Jayanta Bhattacharya, Professor Prasenjit Guchet, and Dr. Ranjan Nanda to chair the session. Sir, I request you to kindly join us on the stage, please. Professor Jayanta Bhattacharya, it's a pleasure to have with us, sir, who has been the head of the Division of Molecular Virology and a member of the senior management team at the National AIDS Research Institute, ICEMA, Pune, and successfully steered various national and international projects, primarily through extramural funding as a principal investigator. He has contributed a significant understanding of the mode of the pathogenesis of HIV-1 clade C circulating in India through basic and fundamental research, especially in the area of virus entry. Currently, he is a principal investigator at THSTI, working extensively on HIV prevention research and development. Thank you, sir, for joining us today. Professor Prasenjit Guchet is a well-known Indian virologist at the Regional Center for Biotechnology, who is popularly known for his research on molecular signaling of thrombosis, inflammation, and innate and adaptive immune responses in human health and diseases, and identifying biomarkers and molecular targets to develop potential therapeutics. He has majorly contributed to SARS-CoV-2 research by focusing on molecular signaling of inflammation, thrombosis, and thrombophilia, promoting lung injury and cell death, leading to clinical conditions like ARDS and hypoxemia, and developing pulmonary fibrosis in SARS-CoV-2 infected animals and patients. It is a pleasure to have you here today, sir. Dr. Ranjan Nanda is a group leader at the Translational Health Group at International Center for Genetic Engineering and Biotechnology, New Delhi. The current focus of his research group is to elucidate the molecular and cellular details in anemia of inflammation and chronic inflammation in diabetic conditions, and especially the influence on the T cell and dendritic cell metabolic function, to explain tuberculosis susceptibility in vivo model systems before correlating with patient data. He is also keenly observing the gut microbiome dysbiosis and altered fecal metabolome in tuberculosis patients with respect to controls and in vivo model systems to identify probiotics and postbiotics for nutritional interventions. It is an honor to have you here, sir. In this session, each talk will be of 15 minutes. I request the audience to kindly reserve the questions until the end of the session. I now request the session chairs to introduce the speakers and call them over for the talks. Sir, please.
So without further wasting any more time, let me first thank the organizers, especially Dr. Bhavatos Das, for providing this opportunity to chair the session. So the first speaker in this session is going to be Dr. Sachet Shukla from MD Anderson Cancer Center, US. Dr. Sachin Shukla is an Indian immunologist working at the Department of Hematopoietic Biology, Malignancy, University of Texas, MD Anderson Cancer Center. His research area focuses on antigen discovery and development of personalized immunotherapies. He is known for creating the next generation of personalized cancer vaccines designed to target antigens identified within each patient. The title of his talk is going to be Long and Short Read Analysis. So may I invite Dr. Shukla, please?
Okay. Good afternoon, everyone. And I can see only the most sincere and the most dedicated people are still here. So that's, thank you first. Thank you. So this is a short talk, and upon Bhavatos's recommendation, this is really geared towards the trainees. And what I'll try to do in this session is just give you my perspective as a computational biologist and as someone who has been working for a long time with next generation based sequencing methods, bulk as well as long read. What I think where this space is, and I'm a relative newcomer to as my introduction suggests, I'm a relative newcomer to this field. So let me just jump right into it. I won't belabor this point. We've already seen this, speaker after speaker has talked about the role of the human microbiome is in maintaining homeostasis, and also how dysbiosis can underlie many different diseases. So from my perspective, there's really two key questions that you are trying to answer anytime you're doing a microbiome study, right? First is, in any given sample, what is the bacterial composition? What are the bacteria, which is a question of diversity? And second, what are they doing, right? What is the actual functional role, and how are they affecting the phenotype of interest? So the ideal goal that we would all like to get is, from any given sample, we want to get completely assembled genomes down to the strain level resolution and with adequate representation of low abundance species, right? So this is the ideal that we would like to get to with the technologies. But of course, the feasibility of getting to this ideal is constrained by the technology at our disposal today, right? Both experimental and computational. So you can think about the technology in this space in like 3 different generations, right? The first generation, which I won't spend too much time on, is based on culture-based methods. So these were, take a sample, try to culture individual bacterial species, do some biochemical assays, maybe do some low throughput sequencing, try and identify them and look at their roles. And this still has a role when we are trying to really do a deep functional annotation or characterization of the bacteria. But by and large, I would say that these methods are getting less and less relevant by the day. Which brings us to the second generation. These are largely based on short read sequencing. And I would say these are the basis of most current investigations in the field. And more often than not, the technology of choice is Illumina. And there's 2 distinct applications, as you've heard since the morning. There's the targeted amplicon sequencing. The central idea here is to do deep sequencing of a shared but very diverse gene region. And for the purposes of this talk, I'll be simply talking about bacteria, but the idea is generalizable to different kinds of microbes. The main goal here is to try and characterize the diversity in the sample. The alternate approach, which takes a very different view into things, is to try and do shotgun sequencing, where the idea is not just to focus on a single genetic region, but instead try to do as much sequencing and as broad of a sequencing of as many genomes or transcriptomes in the sample. So this is shotgun sequencing, and what we can achieve here is diversity, but also some understanding of what is the functional role of the different bacteria that are present in that sample. So here again, 16S rRNA sequencing, or the 16 rRNA gene, is what is typically targeted in the amplicon sequencing based methods. And this is a ubiquitous gene found in all bacterial species. It has 9 hypervariable regions, and these hypervariable regions, as the name suggests, are variable across different bacterial species. So this is what we exploit to infer the phylogeny. And the conserved regions between these are conserved across species, and these can be used for the primer designs. Typically, you focus on, with the short read sequencing, you really can't get to the whole amplicon or to the whole gene, which is about 1.6 or 1.5 kilobases long. You instead just focus on maybe the V3 and V4 hypervariable regions, or the V1 and V2 that are commonly targeted. With shotgun metagenomic or metatranscriptomic profiling, you create these short read fragments, you do sequencing, then there's 2 broad ways of trying to get to representative context. Either you go the OTU or the ASV route, which I won't get into now. But once you have these representative contexts, you can either take an assembly-based approach or a reference alignment-based approach, which is feasible in cases like the human gut microbiome, which is very well characterized, and you actually do have a good reference that you can use, or some hybrid approach followed by QC, alignment, normalization, etc., and then downstream analysis to understand taxonomic composition or the functional profiling of these samples. Okay, so now here are some of the limitations, some of the critical limitations of short read based methods. The first is resolution, right? So the average operon length in bacteria is about 3.5 kilobases. And when you do these Illumina based short read sequencing, you get really short reads, which are, just a few hundred base pairs long. So you clearly are not going to get very high resolution. With amplicon 6 sequencing, as this table shows, even at the genus level, sometimes the resolution is kind of limited and you really don't do too well on the species or the strain level. With metagenomics or metatranscriptomics, you have decent resolution on the genus level more often than not. On the species and strain, it's limited or downright poor. The second big limitation here is ambiguity, right? So in any such study that you do, let's say a metagenomic study, 30 to 40, sometimes up to 50% of the features that you identify are unclassifiable, simply because these are things that we've never seen before, right? Which brings us to the third big limitation, that is, in most cases, you are limited to discovery of what has already been characterized before and deposited in databases, right? So things like just completely undetected features, there's hidden taxa, definitely strain level diversity, all of those things you will not be able to characterize using short read based sequencing. Which brings us to the third generation, which is the long read based sequencing. So we've spent like a year working with these technologies, and there are two main vendors or two main platforms that are available. One is with Pacific Biosciences, which I'll be calling PacBio, and the third is the nanopore based technologies. ONT, or the Oxford Nanopore Technology, is the leading platform in that space. And I'll talk about the central ideas there, but there is really a trade-off between these two platforms, between accuracy and throughput. So let's talk about the PacBio platform, and their technique is called single molecule real-time, or SMRT, smart sequencing. So imagine starting with a double-stranded DNA fragment, right? So the first thing you do is adapt these smart bell adapters. And if you can see here, what that essentially does is circularize that whole DNA fragment, right? So now you have a construct which is essentially a full loop, okay? The next thing you do is, and by the way, the DNA ligases are at the bottom of very tiny nano wells. These are called ZMWs, or zero mode waveguides, which are less than the amplitude of light, right? So they're actually immobilized at the bottom of these wells. And once you add the primers, these individual circular constructs are essentially pulled through the immobilized DNA ligase. And as you can see here, what that effectively does is repeated sampling or repeated sequencing of the original DNA fragment. And at the end of this, you get a long read, which is essentially multiple samples of the original DNA construct, right? So these individual samples or these individual instances are called subreads. And now you can use these subreads to create a very high quality consensus, which they call a hi-fi read. Typically with about 4 of these, you get to about 99% accuracy. The Oxford Nanopore technology is completely different, right? So what this consists of is an electrically resistant membrane on which you have arrayed a number of pores, which consist of these motor proteins, right? So the whole idea here is to create an electrical gradient through which the DNA or the RNA molecule will now be pulled through by the motor protein, right? And as this nucleic acid is getting pulled through, the string of nucleic acid is getting pulled through, the different bases cause a very characteristic disturbance in the electrical flow through this instrument, which is recordable electronically. And because of each of these bases have a characteristic disturbance, that forms the basis of the sequencing that happens, right? So in my view, the PacBio is actually something of a hack, right? All you're doing is they came up with this clever way of generating this circular sequencing, and that's great. The ONT, on the other hand, is a breakthrough kind of technology, which I think is very scalable. It's infinitely scalable. It is very cheap to set up as compared to PacBio. Their latest instrument is like, almost $750,000 of a million. And this one is, you're talking about tens of thousands, and you get these really small chips. They can be deployed on the field. So it's really real time. The problem is it is plagued by very high error rates. And I'll show you that in a minute, right? So like I said, we spent almost the entirety of last year working very closely with both of these platforms, and we tried a number of their early access flow cells and library preparation kits and whatnot. And I'll just give you the take home message here, right? So the first table here, the ONT table, is using their LSK kit with their latest R10.41 flow cell in what they call the super high accuracy base calling mode, right? And I'd like to draw your attention, and the second table obviously is the PacBio result. And I'd like to call your attention to two metrics in particular. The first is the mean read quality, right? So this is the read quality of a typical read generated by that platform. With the ONT, that quality is 17. So that translates to 98% accuracy, right? So if that sounds like decently high accuracy to you, you haven't been working in genomics for a very long time, right? So that's actually very poor accuracy. That means for every 100 bases, you'll get 2 wrong. And we cannot really, live with that for many applications. On the other hand, with PacBio, the mean read quality is 51, which translates to 99.999% accuracy, right? That is fantastic. The second metric I'd like to draw your attention to is the number of reads, the throughput, right? So with ONT, you get 78 million reads. With PacBio, you only get 4.5 or less than 4.5 million reads, right? The third metric, which I haven't really highlighted, but look at the mean read length. With PacBio, you actually more than double the read length itself, okay? So as it stands today, I think PacBio for most applications, and at least certainly for the applications that I am interested in, which is antigen discovery, right? Where we design personalized and we actually treat patients with personalized cancer vaccines based on individual mutations that we find in the genome or in the transcriptome. So we are very sensitive to errors, sequencing or technical errors, and we do our best to choose the best platforms and computationally correct for those particular errors. This is another view into the data. So the Q20 line is 99% accuracy, Q30 is 99.9, and Q40 is 99.99, right? So with ONT, you can look at both the read lengths and the read quality, right? So you can see there's barely any read that is more than 99.9% accuracy. And look at the distribution that you see even above Q40 for PacBio, right? So about half of these 4 million reads were greater than Q40. So how do we use this long read, right? So let's revisit everything we did with the short reads. With the 16S amplicon sequencing, the expectation would be if you could have a read that spans the entire gene, that would give you the best resolution or the best possible resolution with just 16S amplicon sequencing, and you would be right to expect that. So what PacBio reports is that they can get up 3 to 5 million hi-fi reads that span the entire gene with Q at least 99%, and this is very consistent with the data that we have generated. And when you apply it, you actually do see exactly that, right? So you see the Shannon entropy index is much higher when you do the full length 16S versus V3, V4, and the number of species that you detect is also much higher, not quite double, but significantly higher as compared to doing just V3, V4. The second improvement that you see with long reads is that you see improved taxonomic resolution. So in the interest of time, I won't really go into the details here, but if you were to do a sequencing run and then create these representative contigs and create a phylogenetic tree, then the percent of unclassified members within a clade, and if you see, there's the red tells you the percent of unclassifiable members within a clade, and look at with V1, V2, V1, V3, V4 alone is like mostly red. Look at what happens when you do V1 to V9. There's V9, right? There's hardly any reds. This is a small point, and I think you guys, this was already spoken of. So amplicon approaches, whether you're doing short or long read, are much better than shotgun metagenomics in terms of taxa identification. So the second application here is for the metagenomics, not the amplicon based. So with long reads, not surprisingly, you see many more long contigs that are generated. And I think this is a recent paper that was generated by my collaborator at PacBio. What they see is that with long read metagenomics, you can actually get full length assembled genomes. So they are called MAGs, metagenome assembled genomes. And what is not being mentioned here is that this wouldn't have been possible without some computational improvements. So they actually had to come up with new algorithms for assembly and for haplotype resolution, etc. But now we have a pipeline that, based on PacBio metagenomic profiling, you can now get a pretty high percentage of fully assembled genomes, right? So this is getting closer to the ideal that we set for ourselves at the beginning. So with that, I won't go through the conclusions. You can read these yourselves, and I'm out of time anyway. I'd like to acknowledge my new lab. So I'm a new PI at MD Anderson. It's been a little over a year, and I'd like to acknowledge my funding sources, and happy to take the questions at the end. Thank you.
I think probably we'll take the questions at the end, right?
So quickly, so the next speaker is Rajkumar Haldar. And Rajkumar was a scientist earlier in THSTI. So currently he has a company. I think this morning that our secretary DVT launched a product, which is, I think it is from his company. So he's doing very good. And so actually he did his postdoctoral fellowship with Professor Benjamin List, who is a Nobel laureate. And Rajkumar is a very good chemist and biologist. So let's, Rajkumar, stage for you.
Can you hear me? Oh, that's good. Okay. Change from here. Okay, all right. Thank you. Thank you, Dr. Pajin, for introducing me. And it is my privilege. It is a different talk than rest of the talks, whatever you have seen it. But first talk of these 2 days, it was like how to encourage you, how to get your potential best for the societal development. So far, the people who are doing PhD or postdoc, we are all using taxpayer money. So to be honest with you, we are not using that much knowledge for application. So to make you sure that how many of you want to be entrepreneur? Anyone? Oh, I got 1, 2, 3, 4, 5. Not bad, all right? So how many of you want to be scientist? So it's equal, all right? Oh no, a little bit more, few of them. But try to find out that if you want to be scientist, that you want to develop product that should be in the market, or you want to be scientist, that your work should be published in the just journal. Think about it, okay? Now today, whatever we are going to do it, you see the biome labs, all those microbiota, this, skin disease, all of this. So far, very recently we are passing through, or we are still passing through COVID-19. Have you got freedom to operate? No, am I right? And every time what you did it, you kill microbes. Have you ever thought how to preserve microbes? It's one of the toughest things. Have you ever thought that how we can live together? It is even much tougher. You can only decide how we can live together if we know about them. If we don't know about them, then it is the obvious thing that either we'll always think they're our enemy, and we'll make sure that the benefit we'll get it is highly compromised. That is what is going on, am I right? So this is one kind of media. As you saw that in the morning, it was already launched by Deputy Secretary. It's one kind of media you can use. Any specimen of your choice. It can be neonatal. It can be the old person that you cannot take care. And it is also your own being, because most of the people, they are getting non-communicable diseases. And that might be possible that there are certain kind of bacteria which are upregulated, or even more colonies than others, and that's why we are getting problem. Let me tell you something that might be like my personal, but some of you who wanted to be entrepreneur, try to find out that if I can do it, you can also do it. So there's not that much difficulties, but you may have couple of sleepless nights. That's all. So if you look at this one, 1.42 billion people in India, and we have very, very skilled people in India. And we are all intelligent. This is our own thought. But the people who are telling that we are intelligent, sometimes those good bacteria, which is promoting our self-esteem, with the age and time, they goes down. That's why before 39, before 29 or 30, most of the people are dead. Most of the, it is not my word. I think somebody said, am I right? That most of the men on this earth are dead before 30s. And then we just keep trying until we are bullied off. If 1.42 billion people, and look at this number, death, we think we are very smart. We are not hygienic, am I right? If we are smart, we should be hygienic. And if you look at this one, number of diarrhea, 500,000 people, sorry, 5 lakhs people are dying. And out of that, if you look, that is very much shameful for India. 20% kids, they die. At the time we started this lecture, almost 60 kids died. And we don't know why they are dying. If we have all the protocols, if we know really we can help each other, if we think that science we are helping, we are taking because of taxpayer money, we can promote to the others, that we are not doing it. And I was also one of them who was almost going to die. Not one, twice, thrice. So this is one of the picture. I am not that person. That's why I like this one. I am who I am. It is not you. It is the bacteria who is deciding how you will be. As we are ignored, that's why we always believe, okay, it is good or it is bad, all those kind of things. But let us try to know each other, and if possible, try to live together. That is very much important. Now, if this is my life, until 13 years old, I did not know what is car. The people who want to be entrepreneur in this room, please listen very carefully. I did not know what was car. I was living in between cobra and tiger from that remote village of Sundarban, West Bengal. Okay, so from there, how I change it? I did believe education can change. And the education, the institute I traveled, I think all of you can also travel. I passed out from IIT Kharagpur. Okay, and then I had a chance to do PhD in Spain. That's what we all wish, am I right? And then from there, I went to Max Planck. In 2007 to 2009, when I was working, that metabolites, that is what you have seen in the whole talk, whole day, the short chain fatty acid, that is where it recognizes what metabolites you should have it and which kind of disease you will have it if there is a up down of that. And the person with whom I worked 2007 to 2009, everybody thought that we are very crazy. And until 2021, he got the Nobel Prize, we became hero. So sometimes the entrepreneurship is like that. And the people who wants to be scientists, you have to be very much crazy. And to do so, you have to understand that there is some data, some valuable input that we have to carry on it. And one of them is biome life. That is what believed it in scientific domain. And that's why we are translating it. But after that, this is the Peter Schultz. He's the CEO. I think the people who are working in this biology, am I right? Everybody knows who is this guy. He has 8 companies. Sometimes you have a wish, but you need guide. He was my guide. Okay, so he is the person where we came up that Microbiome tuberculosis you can kill when it is latent as well as active. First time in history. The compound is in clinical trial. Think about it. One person who did not know what is car, 13 years old, and coming to the village, from village to other places, traveling the whole world. If he can do it, you can do it, am I right? Yes, you can do it. Now from there, this is the same person who took me and made me scientist at GM, Novartis. I did not pay anything. It is the sincerity what brought me from Novartis to, from Scripps to GNF. At the same time, I work in both sides. What I did it until Spain or with Nobel laureate, I was a full organic chemist. I did not know what is microbiology. I did not have any idea what was microbiology. Then we are trying to find out what is that. And you look at this. 90%, trillion, billion, nobody knows it because we did not spend that much attention. But we think if you take the yogurt, your stomach will be very good. It is because of lactic acid after it is getting fermented, or pyruvic acid, or short chain fatty acid, or some kind of microbes that is helping our whole GI tract to be 3.5. And that's why we are getting healthier. Okay. Now, if you look at this, 90% diseases can be recovered if we use microbes properly. The problem is we only know 0.9%, even less than that. That is the biggest problem because we don't know how to preserve them. If we don't know how to preserve them, the aerobic as well as anaerobic, we cannot work on them. And what is the best example? The things you cannot see it, they control you most. And that is exactly happened very recently with COVID-19. Now you saw your all reserves all gone, and next one is also coming that will even drain out much more. And one is already going, that is somebody said our sepsis, right? That is even going silently. Sepsis, AMR, all those kind of things. So how to handle all those things? If you understand that matters and dark matters, here you have to also understand aerobic and as well as anaerobic bacteria. You cannot just take the aerobic bacteria and say, okay, this is what I have it, this is what I can control it, this is what I am for. No, it is not the case. So you have to have the anaerobic with aerobic, but culturing them together is extremely difficult. Even you take the sewage water, or if you take any water or any soil, keeping them anaerobic microbes in microbes is extremely tough. Will we appreciate somebody who is discovering it? Will we? Am I right? And if it is first time in the world, will you do it? Of course. So that was done at THSTR. That is what is Biome Lab about, that if we can preserve it, microbes as well as aerobic as well as anaerobic microbes. If you look at this whole world, you think that they are selling good product, it is coming from another country. Let me tell you something. This is the compound you buy 3,200 rupees per ml. 3,200 rupees per ml. First of all, you will not buy it because it is very expensive. Second thing, it is suboptimal. Only one particular specimen it works, and that is only for 4 hours. What happened? That 32 rupees, I give you another 80% discount and say, this is the product, I have it, and you can take any kind of specimen and keep it over there. Not only 1 day, 2 days. You can keep it 1.5 years minimum, and then you can take any microbes of your choice and you can culture it and find out what was going on. Yes, this is what we are doing it. Look at this. Any kind of water body, when you can take any tissue, you can keep it over there, and you can go to the proteomics, metabolomics, functional genomics, all these things you can do it. More than that, the compound you buy from us, you can keep it for 24 hours at any temperature. You just without keeping your specimen. That it does not mean that, okay, I bought it and within 5 minutes or 5 months it is gone. No, it is 24 months that you can preserve on the table. Look at this. Here, any product, the biologists who are sitting over here are going to be scientists. Any product you have to ship from here to some other places and you need cold chain is extremely expensive. What happens? You take any specimen, within 48 hours, you can go any part, not only India, any part of the world. And if you do so, you can save a lot of money. So that is the call, go green. And because of global warming, I think that is very much needed, am I right? Now, if you have only 20 milligram samples, sometimes you said, okay, somebody don't want to give me my pancreas, all those kind of things. 20 milligram sample you need it, and you check it out which kind of microbes you have it, 20 milligram. You put in that solution, you make it pellet, and you can do the metabolic studies. And you can go also microbe studies. So it is a powerful invention, and this is a powerful solution. I think anybody who is trying to do it, that clarification I can show you in this PPT. Look at this. If you want to go for the aerobic microbes, you have particular media. You have go for aerobic microbes, you have particular media. This is one microbes biome life that you can do all of together. So what is happening? Innovation is around you, which we are willing to provide you. Whatever you want to do it. The reason it, how many times you listen that people or scientists from Europe and US are doing much better than us. They have more resources. How many times you listen that Indian scientists are not capable of doing it? Because we don't have solution. Because we don't know how to do it. Got my point? So this is what we are coming, we are saying it. The research can be done at institution level. We can take that product and we can give it, and you can be the global leader. You can be the global leader to make your research work profound. You say that, okay, should I buy it? Where did you try it? Look at this. If you go for the fecal collection, sample analysis, reproduction, reproduction organs, environmental sample, rectal swab, tissue, gallbladder, sepsis, all have been done. Microbe studies, whatever studies we said, all have been done. And who is our first customer? You may not believe me. You may not believe me. But there is one institute which is controlling the whole epidemiology, nothing less than Johns Hopkins. They are the first customer who took the products, not only one tube, two tubes, thousands of tubes they are taking from us, because they believe that this product has a value. Okay? Obviously, this is done over here, National Institute of Health. You go to this one, Manipal University, Naipar, Guwahati, then you got AIMS, the people who are sitting there already taking our product, and then BJMG Pune, then IT3 Kolkata, Max Health. They are going for lakhs of samples. And then you are also going for already Johns Hopkins, they already took the product for it. What you can study? Somebody says we have PD, Parkinson's disease. Somebody says I have Alzheimer. Somebody says I am getting this inflammation. All things can be done. And if you look at this cardiovascular disease, what Professor Garg said in the morning, that if you have the trimethylamine oxide, that it's upregulated, and then you get all those kind of cardiovascular diseases. This is where you can be also check that which kind of bacteria they are responsible for it. Then diabetes, cancer. India call 1.4 billion people, 1.42 billion people, but try to find out how many people are healthy. Then you have huge market, all right? You have a huge market. So if you go to this is the what European microbiome, $227 billion market. So innovation, look at this. Innovation, if you don't promote it, it will be sitting over here. It has developed at THSI itself. Somebody has to understand that it has potential. If this is the case, same case, you have to also tell yourself, or somebody should listen that you have a potential, then that person may promote you. That is very much important. Okay? In India, only $4 billion market. $4 billion. The market, whatever we said, only $4 billion. And in the whole world, during COVID-19, it was $147 billion market was going on. It's so small, am I right? But still you are thinking, okay, what is this discovery? This discovery has a huge potential. And I think THST is also coming with other discoveries that can conquer not only India, in the whole world. Now, what we did it, this is what we are trying to cover it. And to do so, we are sitting in Delhi. And this is where the distribution network is going on. Now, about juvenile, this is our building, startup company. Startup company, this is our whole building. We have also another big park. So we have 22,000 square feet R&D center. Okay, the vision is very clear. Innovate, deliver, better public health. Nothing less than that. But we got it. So far we got 12 patent, I think 12 patent, 10 trademarks, and DIPB certificate, QCI approved. The quality we are maintaining, it is not for you. Otherwise, international scientists or Johns Hopkins, they will also not. And HDFC Bank, even more than that, we have DSI recognized. I'm coming, and very recently, not this year, last year also, because of our innovation, we have been recognized as a deep tech pioneer from Paris through global competition. Who is funding us? You. The tax you gave to government, they gave to us. So I give thanks to you. The problem is, it will not be, I want to give one more thanks if you can use our product. The money came from you, but sometimes you also don't know that how to utilize it. Whereas other foreign people, they are using the same product. Yes. Now, CNRS, not only you, the talent of the integrity of the science we are doing it, CNRS from France, they are also giving us fund. Apart from government, BIRAC big grant, BIRAC GG grant, Nidhi for COVID to all those grant, you have to go through so many competition. And you have to earn that one. Until you establish yourself, nobody will get it. And during COVID-19, somebody came from Europe and they signed a LOI of 5 million euros. If you don't have technology, whatever you put on the web, nobody will be interested. But if you have the technology and it has a power, they can come and they can sign it. So they are interested to invest not only 50 million euros. Who are collaborating with us? We are a startup. Who are collaborating with us? Look at this, from South Korea, IIT Tirupati, JNU, Amrita University, ICGEB. This is from California. This is California. This is in the Dante. So they are visiting tomorrow. This is Talishan from Budapest, who signed the LOI of 5 million euros. Yeah, this is one of the biggest cosmetic company who believed in us, who is funding us heavily, and something we solved that has been solved for 120 years. Something we solved that I cannot tell you which company it is. One of the biggest company you can consider in cosmetics. Everybody knows it. That's 120 years they waited to resolve that problem, and we are working on drug delivery, and they found out that is good for them. Small, but it can be powerful. We don't have any wish to be too big, but we want to be the key of the car. It is as simple as it is. We want to be key of the car. If you don't have it, you cannot run it. So that is the whole idea. You see that these are the people, couple of them they're already outside. It's around total 37 people on off. And that is how we are growing on daily basis. Apart from that product, whatever we did it, have you seen this product? Contact killing solution. I told you now, this is also in the market. That is how we are surviving. This is already in the market. And you know who is buying this product today? One MNC. They are all from Europe. They need 200 liters in one shot. This is the product you can spray on any surface, and it's based on human metabolites. No toxicity. Because we believe, when I was in Germany, the person who got the Nobel Prize, we believe that human metabolites has a power. If they come together, you don't need antibiotics. They work as antibiotics. Okay, so that is what we did it. This is already in the market in Amazon also. If willing, you can have it. And this is antimicrobial coating. This is also available. And you know who are using this product? One more interesting thing, this product, you can apply on any surface, not only in ICU, any cold, hot surface, and you can write and you can erase. That's a huge application. Every school can use, every house can use. And more than that, this product, variants of this, you can apply outside of your house. It reduces 12 degrees temperature, 0 power AC. Think about it. 0 power AC, that's what you cannot save in Delhi. This is a huge power. So with that, this is what we have journey. Last year, we won National Bioentrepreneurship Competition, apart from being deep tech pioneer from the global. And very recently, Harvard Business School, as well as Chicago Booth Business School, selected Rubenile to study that how our innovation impact the social innovation and improving the public health. Thank you very much.
So the next talk is by Dr. Saurabh Kedia from All India Institute of Medical Science. Dr. Saurabh is an assistant professor at Department of Gastroenterology, All India Institute of Medical Science. His top areas of expertise are viral gastroenteritis, colitis, hemorrhagic proctocolitis, colonoscopy, and endoscopy. He has published several research articles in reputed national and international journals with several thousand citations. So, Dr. Saurabh, the floor is yours.
So is there any problem?
Yes, sir. First one. Pointer.
Good evening, everyone. Thank you, sir. So after an extremely inspiring talk from Dr. Rajkumar, we again move back to the previous session on the role of gut microbiome in various inflammatory disorders. And at the onset, I would like to thank Dr. Garg and Dr. Babatosh for this opportunity. And in the next 15 to 20 minutes, we would be discussing about the gut microbiome signature of inflammatory bowel disease or IBD. So as Dr. Vineet has already told about that, what is IBD? So basically it comprises of, it's a chronic relapsing inflammatory disorder of the GI tract, comprising of ulcerative colitis and Crohn's disease. So ulcerative colitis involves only the colon in a contiguous fashion, and the inflammation is limited to the mucosa, while Crohn's disease can involve any part of the GI tract, can be multisegmental, and and it can involve all the layers of the GI tract and can cause transmural inflammation. We have seen that recently the IBD, which was previously considered to be a Western disease, and its burden has been increasing in the developing countries. And on the basis of this modeling study, if we consider the overall Indian population, then the overall burden of IBD in India was second highest in the globe, only second to USA. So what could be the possible rise for this disease burden of IBD in the developing world? Well, it's probably the changing environment, the changing diet, which in turn probably changes the gut microbiome, which could consequently lead to IBD in the patient, in the genetically predisposed individuals. So in the further outline, I would like to discuss about what is dysbiosis, what is the dysbiotic signature of IBD, is dysbiosis cause or an effect, and what are the implications of gut microbiome signature in IBD. So broadly, dysbiosis can be defined as the decrease in gut microbial diversity because of shift in the balance between the commensals and the potentially pathogenic or the pathobionts in comparison to a healthy control. However, not only taxonomy, but dysbiosis can be defined in various layers. As to we start with that who are there, which can be defined either with the 16S amplicon sequencing or the sequencing of the entire bacterial genome with the shotgun sequencing. When we further deepen into this, then with the metagenomics, when we sequence the entire DNA, not only we can have a deeper information about the taxonomy up to the species level, but also we can have the information on the genetic content and we can know what can they do. Further, if we look at the RNA, that is the RNA which is transcribed from these bacteria, we can get the answer to the question as what is being done. And if we go further deep, then if we look at the metabolites or the proteins through the LC-MS-based techniques, we can see the end result. So the dysbiosis can be at various levels in this ladder. Now, whenever we look at any gut microbiome study, be it IBD or any other disorder, we need to look at the confounders while we interpret the studies or while we compare the studies. So these could be the technical confounders, which would include the method of analysis, 16S versus metagenomics, whether the mucosal flora, which is closer to the disease and which can look at the gut segment specific alterations, or it's the fecal flora, the process of stool handling, the method of DNA extraction, the sequencing platform which was utilized, the bioinformatics pipeline, and whether the sample was analyzed at one time point or it was analyzed in a longitudinal manner. We can also look at the biological, we also need to consider the biological confounders, which include the demographics such as age, gender, and BMI, the disease type and the disease phenotype, Crohn's versus UC, or whether the different phenotypes of IBD, the disease duration, the disease activity at the time of sample collection, diet, which is one of the major determinants of the gut microbiome, the geography, ethnicity, and the genetics, which again influence the microbiome, and the medications which the patient are on at the time of sample collection. So with this basic introduction, we'll be discussing about the various perturbations which have been reported in the gut microbial signature of IBD. We start off with the cross-sectional changes. So this was a landmark paper by Dr. Xavier's group in 2014, which characterized the mucosal as well as the fecal samples of IBD treatment-naive Crohn's disease patients through the 16S based analysis. And they showed that certain health-associated bacteria, such as the Bacteroides, Firmicutes, Bifidobacteria, were depleted, while the potential pathovoyants such as the Enterobacteriaceae, Fusobacteriaceae were elevated in patients with Crohn's disease as compared to the controls. They also showed that the differences were higher in the mucosal than the fecal samples. And they also looked at the predicted functional analysis through the pie crust algorithm and showed that there was reduction in the biosynthesis pathway. The analysis was further deepened by the same group when they published the metagenome as well as the metabolome of patients with inflammatory bowel disease in discovery as well as in the validation cohort. So if we look at the broad changes, then on the PCO analysis at both the metabolite as well as the microbial abundance level, the CD patients clustered separate from the controls. The CD patient had more dysbiotic microbiome than the controls, and the UC lied in between CD and the controls. Further, when they looked at the species changes, there were 50 species which were differently abundant, of which certain health-associated genera such as Roseburia and Doria were elevated in the controls. Those which were perturbed in IBD included the Roseburia, which again are health-associated taxa, which were depleted in both Crohn's and UC, the Bifidobacterium and Clostridium, which were enriched in UC, and certain genera such as Ruminococcus and Escherichia, which were elevated in patients with Crohn's disease. They looked at the metabolite classes as well, of which there were certain metabolites which were elevated in Crohn's disease, including the primary bile acids and the sphingolipids, and the depleted were the long chain fatty acids, cholesterols, and the triglycerols. And primary bile acids are converted by the gut bacteria into the secondary bile acids. And it is known that secondary bile acids are good for the gut health and primary bile acids are bad. So in the state of dysbiosis, the primary bile acids were elevated while the secondary bile acids were depleted. Certain other metabolites such as lactate, vitamin B5, and short chain fatty acids were again depleted in patients with IBD. So short chain fatty acids, as told by the earlier speakers also, and the secondary bile acids are important for the gut health and immunotolerance. The same group also look at the transcriptomic signature and compared it with the metagenomic signature in patients with IBD on cross-sectional as well as longitudinal sampling. And if we look at the salient features of this study, then when they compared both the metagenome and the metatranscriptome in patients with IBD, they found that most of the features correlated with each other. So there were 3 patterns when both DNA and RNA abundance were proportional to each other. And with certain taxa, they were only abundant at the DNA level, minimal at the RNA abundance, suggesting that they were probably not of any physiological significance. And there were some which had irregular correlation, suggesting that if only looking at the taxonomy through either metagenomics or 16S analysis, we could be missing many important contributors to the disease. And further, when they looked at the dysbiosis, they again looked at the RNA level, DNA level, and at both the levels. And again, there were certain discrepant signatures, while at DNA level, there were certain bacteria which were only dysbiotic at the DNA level, indicate again a minimal functional consequence. There were certain such as Ruminococcus nevus, which again is an important pathobiont established across multiple cohort studies, showing that it had more significance at the RNA level, again highlighting its functional potential. And there were certain such as Bacteroides fragilis, which were both dysbiotic at both the levels, but again more at the RNA level. Sorry. And finally, this group also look at the multi-omic interaction of the gut microbial ecosystem, looking at the fecal samples, the biopsy samples, and the blood samples. And they again looked at a cross-sectional as well as a longitudinal analysis of the gut flora. And broadly, they showed that the inter-individual variation accounted for the most variance across all the measurement types, be it the genomics, transcriptomics, or the metabolomics. And at the taxonomy level, overall, there was no difference between the IBD and the control group overall, probably because of both active and inactive disease patients being there in the same cohort. And they further calculated a dysbiosis score, wherein they looked at those samples which were highly unlikely as that of the controls. And so both UC and CD had dysbiotic as well as non-dysbiotic patients or cohorts. And at the metabolite level, again, there was difference between the control and the active disease group, showing overall reduced diversity at the metabolite levels in IBD and perturbations of certain metabolites. Again, if they analyzed on the basis of dysbiotic score, then similar changes were observed, including upregulation of Ruminococcus gravis and Escherichia and downregulation of the Faecalibacterium. And again, at the metabolite analysis, The short chain fatty acids were depleted, primary bile acids were elevated, and there were again perturbations of certain other fatty acids. So if we look at the Indian data on the role on the gut microbiome signature in IBD, then one of the very first studies was done by Dr. Vineet in collaboration with Dr. Jayashree Praul from GNU, wherein 84 patients of IBD were compared with the 64 controls on basis of 16S RNA-based analysis. And similar to the other studies, the healthy bacteria such as Bifidobacterium, Bacteroides, Ruminococcus, and Lactobacillus bacillus were depleted in IBD, while the pathobions such as sulfate-reducing bacteria were upregulated in IBD. Further, the butyrate producing bacteria in fecal samples were also looked at with the fish and flow cytometry, and the butyrate was also quantified with the gas chromatography. And again, a dysbiotic signature was observed with the depletion of these bacteria in the most severe cases of IBD. And similar changes were also observed with the butyrate concentrations being most depleted in patients with severe IBD. Similar signatures were also observed in patients from South India by Dr. Ramakrishna's group. And again, the butyrate producing bacteria were highly depleted in patients with Crohn's and UC. Further, going on with the study which Dr. Vineet had already talked about the role of FMT in patients with IBD. So if we in this study, if we only look at the baseline samples, that is comparison of controls with the pre-FMT ulcerative colitis patients, then the ulcerative colitis patients clustered away from the controls, and there was a gross depletion of the core in patients with UC as compared to the controls, again indicating lower diversity and depletion of the commensals in patients with ulcerative colitis. Similarly, on the metabolic profile level, again, the pre-FMT UC or the ulcerative colitis as baseline clustered away from the controls, and the similar changes in the metabolites were observed, such as depletion of the short chain fatty acids, phospholipids, and upregulation of the primary bile acids. The mucosal associated macrobiota also showed similar changes, but at the lesser differences as compared to the fecal samples in this study. And finally, the gut microbiome signature in Crohn's disease was also looked by Aditya and Manaswani, and we compared the signature of patient with Crohn's disease, intestinal tuberculosis, which is a close mimicker of Crohn's, and the healthy controls. And it was shown that both states of inflammation, be it Crohn's disease or be it tuberculosis, had similar reduction in the alpha diversity. They clustered together and separate from the controls, indicating that both had a state of inflammation and both had a dysbiosis, but not much difference between each other. And when compared to controls, again, the healthy bacteria such as Storia, Coprococcus, and Acinetobacter were depleted in patients with Crohn's disease, indicating similar perturbations of the gut microbiome in the Indian patients with IBD. So if we look at the longitudinal changes in the gut microbiome signature, then this study again by Dr. Xavier's group, which looked at the 16S RNA sequencing at 3 monthly intervals over 1 year in a cohort of patients with IBD. It again, in this study, they defined what is known as a healthy plane, across which there was some minimal fluctuations of the health of the bacteria of the controls, while the diseased bacteria or the microbiota of patients with IBD had much more fluctuations from this healthy plane as compared to the controls. And when they subclassified the IBD, when patients with ileal Crohn's disease, which is a subtype of Crohn's disease involving the small intestine, who underwent surgery. So up to 50% of patients with Crohn's disease ultimately require surgery, and a requirement for surgery indicates a very aggressive phenotype of Crohn's disease. So the Crohn's disease, which has the most aggressive phenotype, the microbiota in this population deviated most away from the healthy plane, indicating the longitudinal stability of the gut microbiome was reduced in patients with IBD, and there was a gradient with respect to the disease severity. Another study again also looked at this microbiome stability and showed that there was similar instability across all levels of dysbiosis, including the metagenomics, metatranscriptomics, and metabolomics, finally concluding that the gut microbiome in IBD is longitudinally unstable as compared to the healthy controls. A word about the microbial dark matter, that is the fungus and the virus, which has been less explored in IBD. So if we look at the fungal dysbiosis, then it's overall higher fungal diversity, depletion of Saccharomyces, and elevated Candida, Malassezia, and Demeromyces in patients with IBD. And if we look at the bacteriophage signature, then the cordovirials, macrophages have been shown to be elevated in patients with IBD. We also looked at the gut microbiome signature of patients with Crohn's disease. And unlike the bacterial signature, these are the initial results. Further analysis is going on. Unlike the bacterial signature, the microbiome abundance was not different with respect to the controls. And on beta diversity also, they clustered similarly. However, on differential abundance analysis, as reported earlier, we saw that the Candida albicans and the Malassezia, which have been reported to be elevated in Crohn's disease, was also seen to be elevated in the Indian patients of Crohn's disease as compared to the healthy controls. So finally, coming on to the next question, dysbiosis in IBD, is it a cause or is it a consequence? It would be a cause if it occurs before the onset of IBD, and there are certain indirect associations which we will be discussing, and it would be a consequences if it occurs after IBD due to the altered oxidative stress and inflammation of the gut microenvironment. So epidemiological studies have shown that early life factors such as the mode of delivery, breastfeeding, antibiotics have a negative influence on the microbiome, and these factors have been associated with the risk of inflammatory bowel disease, indicating that gut microbial dysbiosis could be preceding the onset of IBD. Similarly, the mice models of IBD, be it genetic or be it chemical, don't develop IBD in the germ-free environment, again indicating towards the role of bacterial dysbiosis. Also, if we look at the phenotypes of IBD, then the IBD most commonly occurs at the sites of microbial stasis, the ileocecal area and the rectum. And these are the sites where the IBD most commonly occurs. Also, patients with Crohn's disease who require surgery, if they are having a stoma proximal to the site of resection, then this fecal diversion prevents the recurrence in these group of patients as almost, and once this continuity is restored, almost all patients with Crohn's disease will have an endoscopic or a histological recurrence. And certain IBD susceptibility genes such as NOT2, ATG16L1 have been related to microbial handling. There are two interesting mechanistic studies also, which also highlight this concept. So in this study, infants who were born to mothers with IBD, their microbiome was characterized. So both the pregnant mothers as well as the kids' microbiome were characterized and compared with the controls. So in pregnant women with ID and the offspring, there was lower bacterial diversity and altered composition with compared to the controls. And when the germ-free mice were inoculated with the third trimester IBD mother and the 90-day infant stools, there was significantly reduced alpha microbial diversity, and this regulated adaptive immunity in these mice. Finally, IBD has a familial predisposition, and siblings or relatives of patients with IBD have a higher risk. So this study looked at these high-risk individuals and followed them up till they developed IBD. So there was a cohort of high-risk individuals who developed IBD, and there was a cohort who did not develop IBD. And when they compared their pre-IBD or the baseline microbial signature, then they saw that the pre-UC microbiome was had proteolytic activity and was dysbiotic as compared to the matched healthy control. And when inoculated into the humanized mice, this led to chronic inflammation and proteolytic activity. And if you look at the single bacterial associations, then there have been associations of bacteria such as Microbiome avium paracellular and AIEC with IBD. And in these two studies, it was shown that there was higher prevalence of pathobionts such as AIEC, Listeria, Campylobacter, and Yersinia and Crohn's disease. And in the other study, higher prevalence of MAP was shown in patients with CD than controls. But again, there are inconsistent reports on this. These are only associations and cannot imply causation. So if we summarize the evidence and we look at this diagram, then as we move from a healthy state to a pre-disease state, there could be fluctuation both at the mucosal as well as the luminal level in the immune environment, the epithelial barrier, and the microbial dysbiosis, which could then further progress, and with the mutual interaction could cause the initial disease with inflammation and further dysbiosis, and which could then lead to chronic disease. So microbiota can be a perpetuator of inflammation rather than being a cause or effect. So if we conclude, then dysbiosis in IBD, whether it is a cause, if the cause is not known, but it certainly would be a perpetuator of inflammation. And finally, the transitional implications of gut microbial signature in IBD, I'll take only 2 minutes. So it has been explored as a biomarker for disease, as a predictor of disease course, as a predictor of treatment response, and microbial manipulation as therapies are already been talked by Dr. Vineet. So in this study, in collaboration with Dr. Babadosh from THSTI, we compared the gut microbial signature of 3 cohorts: healthy population, mild to moderate ulcerative colitis, and acute severe ulcerative colitis, which is an extreme form or is the most severe form of ulcerative colitis. And what we saw was that in terms of alpha diversity, beta diversity, the ASC cohort clustered away and had the least microbial diversity as compared to the UC and the healthy controls. And at the phyla as well as at the general level, there were certain healthy bacteria which were most depleted in ulcerative acute severe colitis, and there were certain pathobionts which is highly upregulated in acute severe ulcerative colitis, indicating that there was a gradient of the microbial dysbiosis with respect to the gradient of disease severity of ulcerative colitis. As a marker of treatment response, we had recently conducted a trial on intravenous steroids in combination with exclusive enteral nutrition in patients with acute severe colitis. So IV steroids are the mainstay of treatment, and they caused improvement in about 60% of patients. And we explored whether the addition of exclusive enteral nutrition augments this treatment response. So, and we found that addition of EN led to improvement in the IV steroid response in these patients. So we compared their baseline gut microbial signature, and we saw that in the entire cohort, as well as in the cohort which received EN, the baseline signature could differentiate the responders versus non-responders with a reasonable diagnostic accuracy, highlighting the importance of gut microbiome as a predictor of response. And in a recent systematic review also, both the microbiota diversity and the taxonomic features were associated with the efficacy for a range of IBD therapies. And also as a predictor of disease course. Okay, sir, I'll move on to the last slide. Okay. So to summarize, we have moved gradually progressed from the 16 RNA based taxonomic profile to the metagenomic analysis of the taxonomy as well as the functional potential, to the realization of functional, to the metatranscriptomics, metabolomics, and the metagenome and the metaproteomics. And finally, we have also seen that there is longitudinal instability of the IBD microbiome as compared to the controls. We are also moving from the cause versus effect conundrum to the microbiome being a perpetuator of inflammation. And finally, we have realizing the transitional potential with the use of microbiome as a biomarker, as well as also as a mode of microbial manipulation. Thank you. And the last, so this is our AIMS IBD team headed by Dr. Vineet, and thanks to our collaborators.
So thanks to all the speakers of this session. It's Saket, Rajkumar, and Saurabh. Saurabh, can you please share? So since we are running at least more than half an hour delay, the expected session, so we need just one question for each or 3 questions for one. So only we'll take 3 questions quickly. So if not, yeah, quick. So there.
Thank you, sirs. I would like to ask a question to Dr. Sachit, sir. Since your lab is working long read as PacBio and Nanopore, so how do you think Nanopore plus Illumina is to analyze any genome or transcriptome? Nanopore plus Illumina is better than PacBio as per cost effective, or it can be done itself in PacBio?
Sorry, Nanopore plus
Illumina. Nowadays, there's a hybrid approaches scientists are applying to get more closer to complete genome sequence. Nanopore plus Illumina.
I'm not getting the last part.
Nanopore sequences plus Illumina, they are
plus Illumina. Oh, yeah. I see. Short read.
Yes, yes.
I see.
But you are in yours, you claiming that PacBio is more accurate than Nanopore, and it is more greater long sequences. But which portion is cost effective? PacBio as well as Nanopore plus Illumina.
Okay. So, I mean, first of all, I'd like to say that, PacBio doesn't pay me anything. So, I'm not I'm not trying to promote PacBio. I think this idea of using long read in conjunction with short read sequencing is a powerful one. People have trying to, so like I said, right, I think you have to think about both the technical innovation and also the computational innovation, right? Because that is very important. And in the last slide that I had up there, right, where they were getting fully assembled genomes, it would not have been possible without the invention of new algorithms, right? So having said that, there are some things which are unfixable, right? So because short reads have their own constraints, and people have tried these kind of hybrid sort of approaches to make contigs with long reads and contigs with short reads and try to put them together. The end result was still not as good as the PAC-BIO, right? Which is not to say that the short reads are not good. So one way, this is maybe less relevant for ONT, but if you were to do short read sequencing and do long read sequencing, which is something that we are trying to do with PAC-BIO, because the throughput is low, the dynamic range of transcriptional expression that you can detect is kind of limited, right? So it is great at isoform characterization. You'll find all manners of, splicing variants and, structural aberrations and all that, but you will not get too much in terms of, the resolution of the dynamic range. So that's where you can use short read to amend it. And the last point I want to make is that ONT is continually improving, right? So in the last, 5-6 years, right now they're single, the last kit that they had just a year ago, the single molecule accuracy was only 90%, right? Now it's 98.3%, right? So I think the future is ONT, but the thing is, at least for our purposes today, PAC-BIO would be the way to go. But, we are fully, trying to keep up with both platforms. Yeah.
So any more question quickly? Yes, Walters.
Yes, I have one question for Saurabh. So Saurabh, particularly for the IBD, so how crucial these non-antibiotic drugs to lead the dysbiosis into the gut microbiota?
Sir, please can you repeat the
Those
question?
are non-antibiotic drugs,
Yes.
how crucial they are to induce the gut dysbiosis?
Sir, we have not exactly looked at the role effect of these non-antibiotic drugs on dysbiosis, but studies from the recent studies have shown that non-antibiotic drugs such as antidepressants and all have a role in causing dysbiosis. But in patients with IBD specifically, we have not looked at.
And the second question, particularly for the antibiotic also, so do you find that after antibiotic treatment, is there any increase in the fungal abundance or fungal diversity in your patient populations?
So like the initial, we haven't looked at it, but the initial study by Dr. Xavier in 2014, when they compared the CD, treatment naive CD, then the patients who were exposed to antibiotics had a greater dysbiosis as compared to patients who were not exposed.
Okay, thank you.
So if there is no more question, I hand over to
I thank all the session chairs and the speakers for sharing great knowledge with us. I now request Professor Jayanta Bhattacharya to hand over the memento to Dr. Sachet Shukla. So please. Next, I request Professor Prasanjit Guche to hand over the memento to Dr. Rajkumar Halder. Next, I request Dr. Ranjan Nanda to hand over the memento to Dr. Saurabh Kedia. May I now invite Professor Amit Avasthi, sir, to kindly come upon to the stage to felicitate the session chairs. First, to Professor Jayanta Bhattacharya. Next, Professor Prasanjit Gulachit. And Dr. Ranjan Nanda. Thank you, sir.
So you may kindly join the audience. We now have come to the final session of the conference. Last but not the least, we have our young investigators presenting on an interesting theme, microbiome and AMR. I request Professor T. Ramamurthy, Professor Ramandeep Singh, and Professor C.V. Srikanth to kindly come up onto the stage to chair the session. Sir, please. Professor T. Ramamurthy is an eminent molecular epidemiologist currently working as the INSA senior scientist at the National Institute of Cholera and Enteric Diseases, Kolkata. He has also served as the former director of NICED. Professor Ramamurthy's research interest spans from basic microbiology to advanced molecular epidemiology studies using next-generation sequencing technologies. It is our honor to have you here, sir. Professor Ramandeep Singh is a senior professor at THSTI. He is well known for his studies on the pathogenesis of Mycobacterium tuberculosis and understanding the metabolic pathways that enable Mycobacterium tuberculosis to survive in stressful conditions. He has also undertaken many vaccine development projects as the principal investigator. Welcome, sir. Professor C.V. Srikanth is a globally renowned biomedical scientist, a great visionary who mentored several of the students on infectious and autoimmune disorders of the gut. He focuses on the key events that cause inflammation during infections and autoimmune disorders of the gastrointestinal tract. Welcome you, sir. As we are running behind the schedule, I request the speakers to restrict their talks to 10 minutes. There will be 2 questions that can be asked from the audience at the end of the session. I request the audience to kindly reserve their questions until the session is completed. I now request the session chairs to introduce the speakers and call them over for their talks. Sir, please.
Thank you so much for inviting us for this last session. So I would like to thank the organizers, the director of this institute, Dr. Garg, and my friend Baptosh Das for inviting us to chair this last session. In this session, the first speaker, I would like to invite Dr. Tarani Shankar Ghosh. Dr. Ghosh is an assistant professor at IIT Delhi, who is a computational biologist who specializes in studying the human microbiome. More than 50 of his research papers have been published in prestigious journals like Nature Medicine, Nature Ecology and Evolution, Gut, etc. He also has numerous US, European-based patents, primarily in the areas of effective metagenome informatics and algorithms, and the discovery of novel microbial consortium that may be therapeutically effective in treating fertility and colorectal cancer. So, Dr. Ghosh, the podium is yours.
Thank you, sir. So good afternoon, everyone. And I really thank THSTI and Dr. Das for giving me this invitation to present, for coming here and presenting. One thing that this session is on AMR and gut microbiome. So I will not be talking anything about AMR as such, but I'll be touching into one particular concept, which is very briefly in one particular concept, which is pertaining to the recovery of gut microbiome post antibiotic treatment. That is the determinants of recovery post antibiotic treatment, which are essentially, which we like to call as the markers of resilience in the gut microbiome. Now, the gut microbiome, and you have been listening to this from the very beginning today. Now, the gut microbiome has 3 primary roles. Okay, it's like an organ, it has 3 primary roles. The first role that the gut microbiome has is it's a sensor. So something goes wrong in the host and the microbiome senses it and it responds accordingly. Like for example, in IBD, you have a microenvironment change that is happening because of the inflammation, and you have certain members of the gut from the oral microbiome, say Veillonella species and things like that, which come to the gut and colonize it. The second function, which we are all interested about, is that of a modulator. But changes in the gut microbiome directly impacts host physiology, and it actually causes a certain disease or a certain disorder. And another, and the gut microbiome, besides that, is actually modified not only by the host, but by a range of environmental factors like our genetics, our birth mode, our lifestyle, gender, our age, the diet that we take, the medications that we take, and also from transmission from other sources like our mothers, like our parents, which is the vertical transmission, or horizontal transmission through our social contacts. Now, interestingly, another major role of the gut microbiome is to actually as a transducer, where it actually transduces the signal that comes from the environment on the host physiology and function. And one of these major transducer, or one of the major modifier of the gut microbiome as well is medications, right? And among medications, since this session is about AMR, we'll talk about antibiotic treatment. Now, antibiotic treatment, every treatment, antibiotic treatment landscape, if you look into the time points of, so say this is a gut microbiome trajectory, it's the trajectory of the diversity of 3 different individuals of the diversity gut microbiome. And you have various time points like before treatment, during treatment, and after treatment. So as you can see over here, there are 2 kinds of individuals that we can notice. These 2 guys who don't have any, this guy who doesn't have any effect. There's a second guy whose diversity actually goes up. That means the antibiotic has an effect, but then it actually comes back. So it has basically recovered. And there's a third group of, and there's a third person for which the antibiotic treatment actually caused a decrease in diversity. But the thing is, it didn't come back. It went into a different configuration, or it went into a low diversity mode. Now, how a person will behave depends on different factors like our age, our environment, the diet that we take, the medications that we are taking along with that, our comorbidities. It also depends a lot on our baseline gut microbiome, that is the microbiome we have when we are giving the treatment, which we'll be talking about. The effect also depends on the dosage of the antibiotic, the spectrum of the antibiotic that we use, and the duration of the treatment. And finally, what effect that this will cause depends on these factors, like the entry of resistant strains, the transfer of resistance from the resistant strains to the susceptible ones, and the loss of the beneficial taxa. By beneficial taxa is again what we'll be covering in our next few slides. Now, there are some elements from what we see over here. It seems that there are some elements that is attributable to our baseline gut microbiome that actually could influence our trajectory post antibiotic treatment. So knowing these elements is important first, because if we know these elements, then we can actually stratify individuals into their probable recovery trajectories, like recoverers and non-recoverers. If you know these elements, then probably on the long run, we can come up with consortia, artificial consortia, or a group of microbiome taxa, whom if we supplement at this stage, we can actually enrich these elements of recovery prior to the treatment, so our gut microbiome remains healthy throughout the treatment. Or we can also, for those guys who still suffer from the loss of diversity, we can actually give this consortia post-treatment, and that can help in enriching the members of recovery post-treatment, and could drive the recoverers into a faster recovery trajectory. So these are the different benefits of actually studying of identifying a baseline gut microbiome. Now this particular, so the question is, are there such elements? Can we identify these recovery associated elements? If so, what are these elements, or what are these members that we are talking about? What are their ecological role? Like they all are part of a community. So what role do they play? And how do they influence host physiology? And to what do they respond? Like Dr. Vinita mentioned, like you give FMT, and along with that, you give an anti-inflammatory diet. So the diet actually feeds the gut microbiome, and you have a longer resilience of the microbiota, or a longer effect of the microbiota, which responds to the post FMT treatment. So what do they respond to? Okay, so we'll come to the first 2 questions now. So this is a story that came up when I was working in Singapore way back in 2017. as 2016. And I was basically working on some microbiome samples from the Tang Tok Seng Hospital in Singapore, where these were like individuals were given antibiotic treatment, and after antibiotic treatment, they were looking into the trajectory that the gut microbiome, how the gut microbiome changes for these individuals. So essentially, there were like 180 samples for 45 individuals. No, apparently, like there were no healthy controls. So all of them were diseased, and they were all given treatment. There were no pre-treatment samples. So it was a very not so good data. But what we generally observe here is, so what I generally observed here is, even though it was a very weird kind of a cohort, I observed something like this. Like there are two groups of individuals. The first individuals followed this particular trajectory, like I'm seeing over here. So they had a higher diversity to begin with, the diversity fell, and then all they recovered post-treatment. And there was the second group of individuals, they had a reasonably similar diversity, it came down and it just stayed down. Same thing happened when you look into the samples, post-controls, like post-treatment samples. So these were the green guys were the controls that we took from another cohort. They were like a healthy Chinese cohort from a different study, we took them, which ideally shouldn't do, but in this case, it worked because they were from the same geography and things like that. And we analyzed them using the same bioinformatic pipelines. And so when we looked into the recoverers, we found that they were clustering close to the controls, but the non-recoverers, they were completely far off. The same thing that we see over here, this is the distance. This distance is just represented at the box over there. So what we see over here is there are two kinds of people. One group of people, for one group of people, the diversity goes down and then it comes back up again. And for the other group of people, diversity goes down and it just simply goes down. So this actually excited me a bit. And then what we did was we went into the literature and I started gathering other data sets, so that if we can see patterns like that in other data sets. And after doing a bit of literature search based on whatever data was available, we came up with four different discovery cohorts, these four. And at the same time, we also got actual control data from Singapore, from the NUH HS hospital. And then what we did was for each data set, I did the same thing, that identified patterns like this, and I classified individuals into recoverers and non-recoverers. Then what I did was we compared the pre-time point diversities of the recoverers, the pre-time point species abundance profiles of the recoverers and of the non-recoverers, and identified species that which over here were higher for the recoverers than for the non-recoverers. And then using kind of a meta-analytic approach, we try to identify if we can identify a set of species that are more or less showing consistent patterns. What we came up with was a close list of 21 species that were showing significant association in either the recoverers or the enrichment in the recoverers in at least 2 of the 4 cohorts. So these are the 4s, and they are ranked based on the strength with which they showed the association with. So as you can see, there are lots of species like Faecalibacterium. So some of these names you have already heard in the past few talks, right? Dr. Kadia's talk, Dr. Auzia's talk, and there were a few other talks before that. So we keep seeing all these names. So these names are all here, like Faecalibacterium prausnius, Roseburia univorans, Ruminococcus bromai, Bifidobacteroides uniformis, Bifidobacterium adolescentis, Bifidobacterium longum. So these are the names that we came up. Okay, they have something in there. There are something in there, and because of that, probably these species are actually coming up in this particular list. So nevertheless, that is the reason what we thought was, let's probe this functionally what these guys could do. So one of the things that we at that point we thought could happen is, because these particular group of species could somehow, because they are facilitating recovery, they could somehow be responsible for actually promoting the growth of the community. Like post diversity, there could be these species could actually promote the growth of the community overall. They could be growing high, and they could also promote the growth of other members. So that's the reason what we did for each, and at that particular time, there was a particular algorithm from Corem, it was a Weizmann Institute of Israel group, which came up in science, which is the PTR algorithm, the peak trough ratio algorithm. So what they do is something is very simple. So they have all these reference genomes for each of the species, and they just simply start mapping the reads onto these genomes. So ideally, what you expect is the number of reads, because there is a replication fork that you see, the cells divide as a replication fork. So you have a replication fork, and it opens up, and then you have the replication and 2 DNA emerging. So what you have is, ideally what you expect is the number of reads that map to the ORI sequence, that is the replication origin, should be higher than the number of reads that is mapping to the TAR sequence. And the more replicative a particular cell is, the more is the ratio that of the reeds that will go to the trough divided by the ratio of reeds that will go to the ratio of reeds that will go to the ori and the ratio of the reeds that will go to the turf, right? So that peak to trough ratio is somehow indicative of how fast a particular species is growing. And once you have the species growth rate for the individual species, you can compute that for the overall community. Now, once you did that, what we started to observe was these particular species, if you look into the recoverers had a consistently higher set of growth rate so this is just for 2 cohorts. For shotgun cohorts, for 16s, we could not. For the different cohorts, we could find that these recovery associated species were associated with higher community growth rate. And that when we looked into the diversity of the overall cazymes of this RAB species, we found that that was actually driven by the diversity of the cazymes in the particular, or the carbohydrate active enzymes in the gut microbiome. Now, what are these carbohydrate active enzymes? They basically are the enzymes that degrade different kinds of carbohydrate moiety. So the more diversity that we have, the more is our energy harvest capability of our gut microbiome. So this was somewhere associated to this. So what we observed is that there was a certain group of recovery associated bacteria. These associated bacteria, they are consistently observed across multiple populations, and they facilitate the growth of the community because of their diversity of the way in which they can digest other carbohydrate nutrients. And because of that, probably they are facilitating the community growth. Now, what are their ecological roles and how do they influence host physiology? Now, this is again, so this was for that model, and then we did a range of other analysis. But what I'm coming with, their role, the ecological role is something that I'm looking to now. So this is from a completely different data set. So this was the work that I was doing like when in my last in my postdoc. So basically what I was looking into is I was looking into elderly health, like older people gut microbiome. And in these gut microbiomes, I will try. So each of these data sets has different indices for elderly health. Like some have frailty, some have depression scores, some have like mental MMSE scores. So each of these, there are like 12 different data sets, and each of them has its own degree of measure for computing these biases. And each of them, they have their own metagenomic abundance profiles. And what I was trying to create is I was trying to create type of a meta network using which I could identify the overall gut microbiome structure that is common to all these 13 studies. So I will not go into the method details over here. So what I did was I used a meta-analysis, random effect-based meta-analysis approach to compute individual species pair associations and then build a meta network. So this meta network is built from 5,000 gut microbiomes from older people, cumulating 12 different studies. Now what you can see over here is there are some modules that you can identify. So this particular module, which is a Firmicutes module, we have all our good guys over here. Over here, if you look into the species carefully, so so pivotal is over here, and over here, if you look into her, this is another good module, which is again dominated by, you see a lot of Bacteroides over here. So there is a Firmicutes associated module, there's a Bacteroides associated modules. There is this module of bad guys, Clostridium form, Ruminococcus nevus, Clostridium hatri, Egerthella lenta, again a drug medication responder. So you have again one bunch of bad guys over here, so-called bad guys. Then you have these oral guys over here, Veillonella, Rothia, Streptococcus, which are coming from the oral microbiome. So there are 4 distinct modules which came up. In the same study, and these are our RAB members. So they are forming the core of this modular architecture. In the same study, I then moved on to the younger people. Sorry for the typo. Right? These are the younger people, and we have again 13 different studies, 8,000 gut microbiomes, all individuals less than 60 years of age. You look at the architecture. The Clostridium cluster, completely different data set. The Clostridium clusters, Flavinifracta, Egerthella, all going together. This is again the Firmicutes cluster over here. Okay, so this is the Firmicutes cluster of Coprococcus, Ruminococcus, and then you will have Faecalibacterium somewhere over here. This is the Bacteroides enris cluster, the same one that we saw earlier, but in a completely different data set. And this is the oral cluster. So you have again these 4 different clusters. The third one, this is again a completely different data set. So this paper is currently in the revision Nature Communication. So here we looked into the gut microbiome from COVID patients and controls from again like at least 14 odd studies, and I try to build the same thing. Again, you see the same thing. There is a Clostridium cluster, there is a Firmicutes cluster, there is a Bacteroides cluster, and then we have all these pathobiont and oral clusters. So the 4 distinct types of modules are getting reproduced across completely different data sets. And among them, the recovery associated bacteria are actually over here. I'll just take another 2 things, are actually over here. So what we see over here is that there are specific bacteria that are associated with faster recovery post antibiotic treatment. These bacteria, they seem to occupy the key or central positions in the overall gut microbiome. They are generally associated with health. They have higher abundance of specific carbohydrate active enzymes, and then they drive the growth rate of the gut microbial community. Now, how to enrich or what do they respond? So to answer that, some of the answer is through diet, and I will also show you the results of a med diet intervention studies that I was a part of, which focused on like elderly health, elderly individuals, around 623 elderly individuals from 5 different institutions across 10 different institutions across 5 countries. And we looked into the effect of Mediterranean diet on the gut microbiome, and from there on the elderly health aspect. So what we observe is like, so I took dietary adherence as a factor or as a measure of the adherence to diet, and then I try using machine learning approach. I tried to identify bacterial species in the gut microbiome which were actually positively responding to diet. And as you can see over here, the central taxa that I reported over there, they are the ones that are positively influenced by it. And later on, when I did the validation studies, we found that it was these positive diet responders, the enrichment of these positive diet responders were actually associated with a improved health outcome post intervention. So what we see now is that retention of the core is sometimes one of the most efficient measures of retaining gut microbiome health. There are certain members which form the core. These are the members that are they account for the resilience of the gut microbiome, and retaining these particular members using diet or using any other specific strategy or using postbiotics is key to retaining the gut health rather than supplementing with specific probiotic members. So, however, this retention will also depend on the kind of network architecture that we have. How bad is just 1 minute?
You have one more slide.
Yeah. So what particular are the state, the baseline gut microbiome, and our demographics. So with that, I would like to end. We are doing some studies again to probe the core across multiple cohorts. But before that, I would like to thank all my predecessors, like my postdoc advisor, Professor Paulo Toole, Professor Ferguson Han, Professor Sharmila Mande, my PhD advisor, who is already here, Dr. Niranjan Nagarajan, who was my postdoc advisor in Singapore. And so I just started my lab this July, and these are some of my lab members. Saurav is my PhD student, so he's here. And these are so Lavanya, Bindya, Abhishek, Shivangi, they are doing their masters over here. Amit will be graduating this March, so he'll be the first alumni of my lab this year. So I'd like to thank my colleagues at the IIIT Delhi and Professor GPS Raghavat, the head of the Department of Computational Biology, IIIT Delhi. And I would like to take questions. So there are some other things that we present over here that I was trying to present that I could not. So sorry for the exceeding the time, and I will take questions later.
Thank you so much. Thank you.
Yeah. Thank you. Thanks to Dr. Bhavatosh and the organizer for giving me this opportunity. The next speaker is Dr. Jyoti Verma. Jyoti Verma is a research scientist in the THSTI. She has worked in the field of antimicrobial resistance and commensals, as well as enteric pathogens during her doctoral research. She has published her work in reputed journals like PNAS, and she is also a recipient of the Yakult Young Investigator Award. She received this award in 2020. Her current research is focused on development of microbiome-based interventions in inflammatory bowel disease, as well as in preterm birth. She's going to talk on antibiotic resistance in commensal and enteric bacterial pathogens. Dr. Jyoti, the floor is yours.
Thank you, sir. Good evening, everyone.
It's a great honor for me to be a part of this conference and talk about antibiotic resistance in commensal and enteric bacterial pathogens. So as we all know that the intestinal commensal microbiota provides a myriad of benefits to the healthy host. However, any perturbations may lead to dysbiosis, and with the administration of antibiotics, it may lead to dysbiosis. With the administration of antibiotics may lead to dysbiosis, which may contribute to the loss of colonization resistance provided by the commensals, which leads to the increment of the resistome of the intestinal microbiota. So AMR, as we all know, that AMR stands when a bacteria changes in the bacteria causes the drugs used to treat infections to become less effective. AMR is a significant global public health threat, and WHO has declared AMR to be one of the leading cause of death by 2050, leading to 10 million deaths by 2050. So it has spread globally and emerged. So talking about AMR burden, so a comprehensive analysis of the burden of AMR till 2019 with estimates for 204 countries and 23 bacterial pathogens and 88 pathogen drug combinations, it predicted an estimate of 4.95 million deaths associated with bacterial AMR and 1.27 million deaths attributed to bacterial AMR. And the highest rates of AMR burden were in sub-Saharan Africa. So as we know, the emergence of resistance being a natural evolutionary process, but many factors such as imprudent usage of antibiotics, high disease burden, poor public health infrastructure have aggravated this crisis. And there has been emergence of resistance shortly after the introduction into the clinical settings. Since 2010, we have only 3 new antibiotics introduced into the clinical settings. So this depicts a huge gap. So we know the mode of action of antibiotics. They mostly they impede the DNA replication, transcription, translation, cellular biosynthesis, or integrity of the nucleic acids. So to combat the action of antibiotics, the bacteria evolves and comes up with the various resistance mechanisms. So to understand the prevalence, genetic nature, and dynamics of antibiotic resistance traits in the commensal and pathogenic enteric bacteria, several commensals from healthy subjects and clinical isolates from patients across various sites from India were analyzed. So one of our lab study explored the gut microbiome of healthy subjects from rural and urban areas. So the key finding was that we could see the enrichment of xenobiotic metabolism in the rural and urban population. So with this insight, we further moved to study the AMR traits of the commensal human microbiota. So at this study, in which we explored the antibiotic resistance traits of commensal human gut microbiota, so we observed that the genes associated with tetracycline resistance were mostly abundant in all the metagenomes and followed by the beta-lactamases. And there were other resistance genes which encoded resistance to streptogrammin, lincosamide, aminoglycosides, vancomycin, and fluoroquinones. And they were further, the genome of gut commensals, they were observed to be enriched with various integrases, transposases, and other mobile genetic element related protein encoded genes physically related to linked to the AMR genes. So these AMR genes, they could be disseminated through several natural processes, including transformation, conjugation, transduction or fusion of OMDs in the gut microbiota. Further studies were done to have molecular insights into antibiotic resistance traits of enteric pathogens. So we analyzed 654 gram-negative enteric pathogens belonging to different genera and different species, isolated from 2009 and 2015. So we, all those isolates, they were screened against 22 antibiotics, and the resistance pattern was seen to increase over the years. And further analysis of the genomes of the XDR isolates, it revealed the presence of various AMR genes. The XDR isolates, they harbored more number of AMR genes. So there's the size of bubble, it indicates the abundance of resistance genes. And further, the subclasses have been mentioned. And all the And all these AMR genes, they were linked with various mobile genetic elements. Further, we also studied the evolution of XDR Vibrio cholerae. So we had around 443 Vibrio cholerae isolates. So out of them, these IDH06781, which was resistant to all the antibiotics, was sequenced. And from after the sequencing result, what we found that this isolate, it had around 40 AMR genes. And we could see the linkage of various AMR genes with the various mobile genetic elements. So with this, I would like to conclude that the ARGs are widely disseminated, both in the commensal and pathogenic bacteria. And more than 90% of current clinical isolates there are MDR. Resistance in the commensals and enteric pathogens is due to the horizontal acquisition of insertion sequence elements, transposomes, plasmids, and ICs. So to curb the spread of AMR, the possible steps that can be utilized includes increased public awareness, improving sanitation and hygiene, regulating the usage of antibiotics in the agriculture and environment, increasing the surveillance, and looking for vaccines and other alternatives, and working on the rapid diagnostics and making new drugs and drug potentiators, and further international coalition for action. So in the end, I would like to acknowledge my mentor, my collaborators and my lab members, DBT and THSTI for the support. And I thank you all for your kind attention.
Our last speaker for the day is Dr. Deep Jyoti Paul, who is the recipient of the prestigious M.K. Band Young Research Fellowship. He is also the recipient of Young Scientist Award from the Microbiology Society of India and Young Investigator Award from the Bill and Melinda Gates Foundation. He has published more than 40 research articles in international and national journals. The current focus of his research is to create a One Health microbiological study to tackle human, aquatic, and environmental antimicrobial resistance. And today he would be talking about rapid detection of AMR genes in gut microbiota. Deep Jyoti, it is all yours.
Thank you, sir, for the warm introduction. And good evening, everyone. Today I will be discussing about the rapid detection of AMR genes in gut microbiota. So why this AMR is very important? One of the major reason is that it poses severe human health threat. And if we look at the very recent data, the majority of the infections like respiratory infections, bloodstream infection, or urinary tract infection, those infections which are mainly cause for the death of the patient are either associated or attributed to the antimicrobial resistance. And it is not only in Indian context. If we see the data from all over the world, it is a very common phenomenon, and the death rate was also very significant. And at present, the AMR cause approximately 7 lakhs death per annum. And if the same trend continues, it has been projected to be 10 million death, like my previous speaker has already told, 10 million death per year, and it also creates a loss of 3.5% in global GDP. And this AMR is not only a problem of human health. If we look at any other sector like animal, environment, and food, so this AMR is a major concern, and which leading to an economic problem and food security issue. And a few days back, I was going through some online stores and I was just looking. I was surprised, and it was very surprising to see that their advertising strategy, they're selling chicken and eggs with the name of that it is antibiotic residue free. So from that, we can understand the intensity of the high uses of antibiotics in farms and agriculture, which is really a major concern. And if we take any community, like it is gut or environment, there used to be a high diversity of bacteria, like many of them are sensitive and resistant. But under the pressure of antibiotics, the community structure changed, as the susceptible bacteria got died and only the resistant ones remain there. And this resistant organism got expanded by their vertical transmission through cell division, or by the resistant genes in this resistant genes in the organisms got transferred by different mobile genetic elements like plasmid, integrons, or transposons. So to understand the prevalence of these resistant determinants, many previous studies has already been done from our lab, and it has been observed that beta-lactamase resistant determinants, aminoglycoside, on quinolone resistance mechanisms, and efflux pump are the major resistant determinants that are associated with the gut microbiota. And if we look the other studies also that from around the world, the similar kind of resistant genes has been found that with having a strong association with the gut microbiota. And if we see the very recent data in Nature Microbiology, they have found that 3 different resistant genes, beta-lactamase resistant genes, CTXM, TEM, and OXA48 are the major concern in this gut microbiota. So keeping these things in mind, and we so many sentinel studies have reported the association of these resistant genes with gut microbiota, we thought of design or develop a rapid detection method that could help to detect the different resistant genes. So from the prevalence data, we have selected 16 resistant genes and had tried to identify the specific nucleic acid signature to design the probe. As a single resistant genes may have very different number of resistant variants, like this NDM, which is known to be the New Delhi metallo-beta-lactamase resistance, and it was named after our capital New Delhi, from where it was first discovered. So this NDM itself has 43 variants. So it is very important to design the primer to identify those resistant genes. And we have designing the primers of these resistant determinants. We have also checked for the GC content, TM, and length of the oligos. So we have divided these 16 resistant genes in 4 different groups. In the first groups, we kept 4 different beta-lactamase enzymes like NDM, CTXM, KPC, and TEM. NDM, just now I have mentioned. CTXM and TEM are also comes under beta-lactamase genes, which is extended spectrum beta-lactamase. And these are the primer sequence that we have developed that has the specificity to bind almost all the variants of these resistant genes. Similarly, in the second group also, we kept 4 different resistant genes, which are showing resistance towards chloramphenicol, aminoglycoside, sulfonamide, and fluoroquinolones, which are one of the major antibiotics that has been used clinically. So in the group 3 and group 4 also, we kept the resistant determinant. And all the primers, all the oligos that we have designed were checked in silico to bind to their all the resistant determinants, whether they are binding to all the resistant determinants or not. So to validate, after designing the primers of all the 16 genes for in vitro validation of these oligos, we have taken these 5 different MDR isolates that have already been reported and characterized in the lab by NGS, and which were obtained from the stool samples of different patients. So, and all these isolates are carrying all the 16 resistant determinants. So with these organisms, PCR was standardized for amplification of the 4 resistant genes from each group, and their identification with maximum sensitivity and resolution in agar-rose gel electrophoresis. So in the first, this is the electrophoregram showing the first amplification of all the 4 beta-lactamase resistant genes in uniplex and multiplex at their specific amplicon size. Similarly, the second gel image showed the amplification of a second multiplex PCR, and where we have targeted 4 resistant genes, CAT, ASA, OQX, and SAL2. Similarly, in the group 3 and group 4 also, we have standardized all the resistant genes. In the group 4, we have taken 4 resistant genes, ERM, and among these, ERM, B, and MPH are the cause of macrolide resistance, and DFRA, which causes the trimethoprim resistance, and QNRS is the quinolone resistance, cause for the quinolone resistance. So after followed by the standardization of this MPCR assay, we try to translate this assay in dipstick. So the same oligos that we have designed and standardized in the MPCR assay. So the forward primer of all the resistant genes were linked with the tag linker sequences, and the complementary of those sequence was imprinted on the dipsticks. Similarly, the reverse primer was also labeled with biotin that which is the affinity to bind with the streptavidin. And this streptavidin was attached to the link coated with the latex blue beads that is present in the labeling reagents. So after performing the PCR, the amplicon using these tech primers along with the buffer were mixed with the latex beads. And during lateral transfer, it provides blue color signal so that we can identify the presence or absence of the resistant genes at the specific positions. So this technology was developed by Dipstick TVA, which is a Japanese-based company, and this is the overall representation of the technique, and which is named as STH pass. So this is the here is the analysis after doing the dipstick assay of where we have targeted all the beta-lactamase resistant genes in uniplex and simplex. So here we can, based on the line location of the resistant genes, we can easily identify the presence or absence of the resistant genes, and where in negative control there is no band. Similarly, in the multiplex also we have seen we can see the blue signal for all the resistance genes, all the 4 resistant genes what we have targeted, and in negative control there was no band. So similarly, similar studies we have also done, and recently we have completed a study on with similar technology where we have standardized a rapid point of care test for differentiation of SARS-CoV-2 variants in COVID-19 patients. And this work was selected for the Bill and Melinda Gates Foundation, and the same was presented in International Congress on Infectious Disease, which was held in Malaysia. So to summarize my talk, I would like to tell that our gut microbiota is continuously exposed to antibiotics, and hence it is very important to prevent those things. And I think using this dipstick assay is an appropriate assay to at least which will select the antibiotics against these acute and chronic infections. So I would like to acknowledge my Executive Director Pramod Garg, my mentor Dr. Babatosh Das, and Department of Biotechnology, and all my colleagues and lab members. Thank you so much.
Deep Jyoti for this wonderful talk. Now the session is open for a couple of quick questions.
I have one question for you, Jyoti. So in your genomic analysis, so when you found signatures of antibiotic resistance in Salmonella, for example, did you also see any features of invasiveness? Because this particular organism, Salmonella typhimurium, is supposed to be a localized, it infects only the gut, right? I mean, it doesn't go beyond the gut area. But there are, cases as seen in, Southern African countries, wherein this particular, pathogen has evolved to also become invasive. So did you see any such features? Are you aware of any of these?
So in that particular study, we only concentrated on the AMR part. So we didn't look into the invasive features of Salmonella.
Yeah, my question to Tarani is, so some of the replicon prediction in your through sequence is very challenging sometimes, because it needs a lot of involvement of environmental factors like pH or EH, and then the carbohydrate concentration or amino acid concentration. That's what we conventionally using. So one challenge, probably Ramandeep also agree with me, culturing a Mycobacterium leprae is, we don't have any medium for the growth of Mycobacterium. Through your prediction models, it's possible to recommend some medium or media, whatever it is.
So that is exactly a very nice question, and something what we are trying to like me and a couple of my colleagues, when we sit and discuss, we do it like, so there are different species which have different growth requirements. They have different genomic profiles. So can we predict the ideal growth conditions of a species looking into its genomic profile or looking into its genomic signature? So this is essentially what we keep on discussing, like, can we actually build AI-based models to do that? Like, we have the protocols for actually isolating species, and then we have the corresponding genomic gene families, or like gene profile of each the coding sequences. Can we actually do that? We have not tried it, but we are currently like that is what we are like trying to do, like collect culturing profiles or culturing protocols for multiple isolates. Then we take their matched genomic sequence and try to predict with the requirements of, like you mentioned, pH and other functional aspects. We have not tried it,
But yeah, you will get a Nobel Prize. So
Hello.
we are concluding this session. We thank all the speakers and all the audience.
Sir, I do have one question. Can I ask?
Yeah, a quick question, only one.
So, Dr. Paul, do you have the AMR kit or is just still process is going on?
Thank you. Actually, it is an ongoing study, so we are going to simplify the technique, and then we are thinking to use the directly from the sample we are detecting. We try to detect these AMR genes. So it is an ongoing study.
So I have just one question for Taini. So Taini, you know that in the gut, what actually we observe that the commensal bacteria, they are also coming extensively drug resistance. So when you basically discuss regarding the role of antibiotic in the microbial dysbiosis, do you find a specific classes of antibiotic that has the maximum impact on dysbiosis?
Yes, for example, when we were doing this analysis, so there were some antibiotics which are targeted with a generic, like say for example, clindamycin or like macrolides. They are more general, like we tend to have more firmicutes having resistance for macrolides, for example. But amoxiclav and amoxiclav was like ampicillin based antibiotics. You have proteobacteria and bacteroides to some extent which are more resistant to it. So yes, depending on the antibiotic, we will see the different alterations in the extent and the kind of dysbiosis that we see in the gut microbiome. So it will depend. So the exact diversity goes down, but the exact direction in which the sample goes or the microbiome moves will depend on the duration as well as the kind of antibiotic, as well as the baseline microbiome composition.
Yeah, thank you.
I thank all the session chairs for the excellent moderation and our young investigators for sharing their inspiring works. I now request the session chairs to kindly felicitate the speakers. I invite Professor T. Ramamurthy to hand over the memento to Dr. Tarni Shankar Ghosh. Thank you, sir. I request Professor Ramandeep Singh to hand over the memento to Dr. Jyoti Verma. Thank you, sir. Next, I call upon Professor C.V. Shrikant to hand over the memento to Dr. Deepjyoti Paul. Sir, please. Thank you, sir. Now, I request Professor Shibnath Majumdar to kindly come upon to the stage to felicitate the session chairs. Sir, please. To Professor T. Ramamurthy. To Professor Ramandeep Singh. To Professor C.V. Shrikant. So please join for the group photo. With this, we have come to the end of the one-day conference on human microbiome in health and diseases. Now, we will have the much-awaited THSTI Yakult Best Poster Award declaration. Please join us in applauding the awardees. I request Professor Shinjini Bhatnagar, Professor Vineet Ahuja, and Usha Menon ma'am to kindly come up onto the stage to give away the THSTI Yakult Best Poster Awards. We have 10 THSTI Yakult Best Poster Awards. First position will receive a cash prize of rupees 25,000. Second position will receive 20,000 and third position will receive 15,000. We also have 7 best posters receiving rupees 10,000 each. So I'll announce the winners from the 10th position. We have the honor to have Professor Menon, Professor of Gynecology and Oncology, UCL, to be here with us today. Welcome, ma'am. So at the 10th position, we have Tuba Qamar, CDRI. I request Tuba to kindly come upon to the stage. Ma'am, please give the 10th position award to Tuba Kumar. Next, we have Tanshi Mehrotra. I request Tanshi to please come upon to the stage.
You should give the area of work also, no?
Title hai kya?
Title hai. So they can announce it, then they can tell it themselves, okay?
I request the winners to kindly tell their title also. Once they receive the award, please come over to the mic and announce the title of your presentation.
So my title of the poster is Genomic Insights into Antimicrobial Resistant Traits of Commensals and Pathogenic Bacteria. Thank you.
Next, Shivani, University of Baroda. I request Shivani to kindly come upon to the stage to receive the award. Next, we have Shweta, PGIMER. I request Shweta to come over to the stage.
Yes, good evening to all of you. My topic of the title is Development of Effective Symbiotic Consortium of Bifidobacterium longum subspecies BB536, which seems to be showing the good bifidogenic effect when we incorporate with the consortium inside the gut. So thank you.
Thank you.
And just one more thing, just I wanted to say that thanks for this whole community sessions of health events which was organized. By these events, we can do the collaborative work with different scientists. So I am very thankful to all the scientists working in the India.
so that we can do some collaborative work, as well as we can grow up. We can grow up in the scientific community, and we can grow up like that. Whole Indian can be a good level, at the good level. So thank you.
Thank you, Shivani. Next award, I request Meenal Chawla to come upon to the stage.
So a very good evening to all of you. So my title was to fecal short chain fatty acids as a biomarker of acute graft versus host disease in transplant patients. Thank you.
Next, the award goes to Ayushi Purohit. I request Ayushi to come upon to the stage.
Hello, everyone. So the title of my poster was Modulating the Levels of Master Regulators, PPGP and DKSA in Reducing the Resistance Potency. Thank you.
Next award goes to Ankita, NIBMG. Ankita, please come over.
So the topic for my poster was the gut microbial taxa that was linked with non-alcoholic fatty liver disease.
At third position, we have Taruna Aharodia. I request Taruna to please come over.
So the title of my poster was some contributions to the characterization of infant gut microbiome in early childhood growth failure. Thank you.
Thank you, Ankita. At third position, we have Taruna Arodia.
Hi, thank you everyone. So the title of my poster is Pancreatic Microbiota Associated with Chronic Pancreatitis. I would like to thank THSTI and AIMS for providing me this opportunity. Thank you so much.
At the second position, we have Bharti Kandial. I request Bharti to kindly come over to the stage.
Good evening, everyone. I'm Bharti Kandial, currently doing my PhD under the supervision of Dr. Babatosh Das. So the title of my poster is The Role of Gut Microbiome in the Pathogenesis of Non-Alcoholic Fatty Liver Disease. Thank you. Thank you, ma'am.
At the first position, we have Ms. Komal Sharma. I request Ms. Komal to kindly come upon to the stage.
Hello, everyone. Thank you so much. And my title was Vaginal Microbiome Signature Associated with Preterm Birth. Thank you so much.
Now, I request all the award winners to kindly come upon to the stage for a group photo. So we have a request. I would request all the winners to kindly once again tell their title. Okay, number 10, we have Tuba. Yeah.
My topic is deciphering the role of M1, M2 polarization in visceral leishmaniasis.
I request Professor Bhabatosh Das to also come upon to the stage to have the group photo. I would like to thank our judges for the poster evaluation.
Okay. So I would also like to thank all the judges. So what actually I want to mention that we intentionally keep all the judges, those are working outside THSTI, particularly Professor Binit Ahuja, then Dr. Ranjan Nanda, and Dr. Nita Wadha, and also Dr. A.K. Pandey. I thanks all of you. Basically, they have taken really huge amount of time to evaluate the posters, and I hope everybody was very happy with their questions. Thanks to all of you.
Can we have a big round of applause for all the winners, and congratulations to all winners and the participants. I now call upon Dr. Dinesh Mahajan, Principal Scientist, THSTI, to kindly come upon to the stage to deliver the vote of thanks. Sir, please. Sorry. I would like to call upon Dr. Nitya Wadhwar to the stage for deliver the vote of thanks. Ma'am, please.
That's okay. Good evening, everyone. It is my immense pleasure to close today's conference on human microbiome in health and disease with a vote of thanks. On behalf of THSTI, we would like to express our gratitude to Professor Gokhale, Secretary, Department of Biotechnology, Government of India, for his gracious visit today, his encouraging words, and unwavering support. Thank you so much, sir. We thank our distinguished dignitaries, Professor Sudhanshu Rati, Professor Debasis Mohanty, Professor Partha Majumdar, Professor Vinit Ahuja, and for their constant guidance and support. We are thankful to Professor Shinjini Bhatnagar, Professor of Eminence, THSTI, for the welcome address, and Dr. Sushmita Chaudhary, Assistant Professor, THSTI, for wonderfully explaining the objectives and expected outcome of the conference and workshop. We sincerely thank Professor Pramod Garg, our Executive Director, for his guidance and supervision, without which this whole program would not have been possible. Thank you, sir. We thank all the respected delegates, the session chairs and speakers from various institutes and THSTI for taking out time to participate in this event despite their busy schedules. We thank Professor Takeda, Okasa University, Japan, for joining us online. Our sincere thanks to all students and their mentors for their participation in the conference and making the discussions vibrant and interactive. Our heartfelt thanks to all our collaborators and to all the invitees who have joined virtually. We thank all the committee members involved in organizing this program today, and especially the staff and student volunteers who have worked relentlessly for the last days. We thank Mr. Santo and his team as well for all his support. We once again congratulate all the winners of the THSTI Yakult Best Poster Awards. Lastly, on behalf of the organizing committee, I thank the entire THSTI family for making this program a grand success. Thank you.